welcome you all here, both the people that are here in the room, as well as the people that are dialed in via the Zoom, and as well as people in the future that are going to watch the recording. We held this UCSF Rare Disease Symposium 6 years ago, and as well 2 years ago, and now we plan to every other year. And we plan to hold it in February, because the last day of February is Rare Disease Day. I just want to thank Rupa and Sohaila and Mike and Clea for all their work organizing this meeting, and especially Nate Prorak for all his work on the logistics. The numbers around rare diseases are dramatic. Over 7,500 have been described. Over 350 million people on the globe are affected. Last year, the FDA approved 58 new medicines, 58 new therapeutics, and over half were for the treatment of rare diseases. But obviously, the unmet need still remains very great. Of particular note, that a treatment for a familial, an inherited genetic form of a rare disease may in fact have utility for the sporadic, common, non-genetically inherited form of the same disease, whose numbers might be much, much, much greater. Obviously, it depends on the pathophysiology of the disease and the mechanism of action of the drug. But without further ado, I want to introduce James Levine, the president of Fondation Ipsen. Jim, who's going to lead the first session.
Morning, everybody, and morning, everybody out in Zoom land. And a massive thank you, of course, to the UCSF team, Charles, and the whole crew, and for all of the team out in Paris. A massive thank you to them too. So I really, the reason I came to Fondation Ipsen is it's very rare in medicine that there are absolutely massive, critical, unmet needs. And not only was every single syllable uttered by Charles true, 350 million people living with rare diseases, 94% of those individuals do not have any cure, any available therapy. And with the invention of new therapeutic technologies, the advancement of biotechnology, this is a massive, massive health opportunity. And exactly in line with that, it is my unbelievable honor to introduce via Zoomland, if you like, David Fajgenbaum, Professor David Fajgenbaum at the University of Pennsylvania. I've got his bib written here. I could actually spend probably the entire symposium telling you about him. The only thing I'll tell you is I mean, the man is a superstar. He's a scholar, he's a true gentleman, and he's a true advocate for medicines for rare disease. He's received multiple NIH and FDA awards. He was the first youngest tenured professor at the University of Pennsylvania. He's been profiled by every news media outlet you've heard of, and many you haven't. And he's received degrees from places that they won't even let me into. University of Oxford, University of Pennsylvania, Wharton School. He even has an MBA. I mean, as you will see, he's not only a true advocate for rare diseases, but his personal story will change how you look at the world. And it's my honor to click and share with you what he's going to teach us. Thank you all.
Dr. Fajgenbaum, how can I begin to say thank you? It's slightly cooler here in Philadelphia compared to San Francisco, but we're warm in this room.
That's right.
So hearing all of your accolades, degrees, and accomplishments, the question I have that really predominantly in my mind is, I've never interviewed somebody called the Beast.
Well, we should clarify. This is a former nickname. Well, first off, I just want to say how honored I am to be able to have this conversation with you and to be a part of such an important event and also such a great foundation. But yes, we should preface that as formerly known as the Beast. Now no one would nickname me the Beast.
Could you put that in context, please?
Sure. So, I played college football here in the States, American football at Georgetown. I was a quarterback there, and I trained a lot. This is well before I was in medical school or dealt with my illness, and trained a lot. And because of how much I trained, my nickname was the Beast. And like I said, no one would call me the Beast anymore. But,
none of your students.
That's right. That's right. But my classmates in med school called me the Beast.
Seriously, I've read your book, and I think, being serious, I think the Beast is actually quite important in all of this, if I may. This sense of unlimited capacity to win, I mean at all costs, because the costs in your case were the greatest that can be imagined. And that beast inside of you, I suspect, is this incredible force of nature. And I'm so happy that we can get to share a little bit of this with the audience. But to start with that, I'd like to, as you know, in the rare disease space, the diagnostic odyssey is where this all begins. And interestingly, it's sort of a terrible story, but you describe becoming ill in college.
That's right.
And originally you were diagnosed with lymphoma.
Yes.
And a nurse comes up to you and she says, good news.
Yeah, it's not lymphoma. That's right. Yeah, so I was a third-year med student at the time, and you're right. The doctors thought it was lymphoma, and then the nurse came in and said, "Good news, it's not lymphoma. It's Castleman disease." And of course, I was thrilled. I was a med student. I still didn't know what Castleman was. But great, it's not lymphoma. And then I started to look on Wikipedia of all places, and I got some really frightening statistics. I mean, basically, the Wikipedia page said that no one with Castleman disease lives more than a few years after diagnosis. And here I was, 25 years old, sitting in the hospital room. But I will say that at that stage, we just didn't know what this disease was. And so, as you know all too well, in the rare disease space, we fight so hard to get a diagnosis. We want a diagnosis so we know what it is that we're up against. But sometimes getting the diagnosis doesn't actually give you more answers, it just gives you more questions. Mm-hmm. And that's certainly the case with Castleman's.
And we know this, of course, again, just opening the lens a little bit, we know this, of course, internationally, that patients can be waiting multiple years. And in fact, many patients, don't get a diagnosis at all. Right. My father's wife, she has a rare disease, undiagnosed, progressive neurological condition when you don't know what it is. And that's, and sort of, as you say, when you get the diagnosis, is that a positive step towards the next step, which is ultimately, of course, looking for a cure? Or is it sort of such a destructive moment that it's sort of shattering?
It's a great question. I think it's so different depending on the rare disease that you're talking about. In the Castleman's case, when I was diagnosed back in 2010, there were no FDA-approved therapies. There were no promising drugs that were sort of around the corner. There were no off-label drugs, and so it wasn't very comforting to get it. But I think that there are, thankfully, some rare diseases where you get the diagnosis and all of a sudden you have a full explanation for what is it you've been going through, and now what we're going to do moving forward. And that's the goal for all of us in the rare disease space, right? We want to quickly and accurately diagnose patients, and we want to treat them as promptly as we can.
You were in this sort of boat of having a disease that at the time, I think there were only 6 documented patients who had it. You were the 6th.
Well, so this actually gets to sort of medical misinformation, and that was that according to...
Am I medically misinformed?
Well, no, and actually, so UpToDate, which at least here in the United States is a website that is like the ground truth for medicine. People often call it the Bible of medicine. It's sort of like, what do you do for every disease? How do you treat diseases? And UpToDate said that there had only been 6 cases ever of my subtype of Castleman's. And I make a joke in my book that UpToDate was not up to date, because I learned right around that time that there was a clinical trial that was enrolling patients just down the street in North Carolina that had just enrolled 79 patients into it. And so I was like, wait a minute, how could you have 6 patients ever, but you just enrolled 79 patients down the street? And this gets to this sort of issue with rare diseases. Oftentimes, you have a hard time with getting accurate information. And so what happens is that, you know, basically there's what we now know are 5,000 patients diagnosed each year in the US. So not 6, but of course, when you're less than 200,000 diagnosed or alive, which is our definition for rare disease, there's really limited information, and people just don't put in the effort to bring together cases.
So Dr. Fajgenbaum, open the lens again for me a little
Sure.
bit. So all of us around the world trying to help patients get diagnosed, but also to try and find cures. Part of the development of markets, you've also got an MBA, of course, right? Part of the development of market is knowing how many patients have a given disease and how many will have access to that treatment. Talk to us a little bit about international databasing. You talk, even in the context of Castleman's, that would be great, but can you give us, can you reflect on the power of that?
Sure, there actually are very few international databases that exist for any disease. Within certain countries, you can get really good estimates. So countries like the UK and others that have nationalized health systems with one centralized medical record data set, that's where you can really get a sense for how many people have this and who will receive the diagnosis. And then you sort of extrapolate. You say, well, if this is how many people have it in the UK, then this is how many people probably have it around the world. We all know that there's differences from one country to another, but we sort of make those assumptions. For Castleman disease, we do have an international registry, so we can get patients to enroll from around the world. But again, you can't expect that every patient from around the world is going to enroll into your registry. But actually, probably the best source of getting epidemiology for rare diseases is exactly what you said, and that's from drug companies. Because companies that are developing a drug for a given disease, they want to know what that market size is, right? They need to have a good sense for where are they, who treats them, you know, what's the total numbers, what can we project in terms of sales for our drug. And so they oftentimes have the best information on epidemiology, but unfortunately that information doesn't always get shared externally, right? Sometimes it ends up in a publication, but sometimes it's just used in a PowerPoint within that company, and there aren't really ways to share that information externally.
Just feeding off that microscopically, you know, your interest and many others is, of course, in using artificial intelligence. And one of the areas of growing interest is the capacity of AI to interrogate medical data record sets. And this can be done agnostic of language, this can be done agnostic of country, and different methods can be used in the United States. Claims language can be used. In Europe, the actual records can be scanned, all electronically using AI to pick up cases that have yet to be diagnosed. And sometimes, you know, I worry that even these sort of mind-boggling estimates, what 300 or 400 million patients, can be massive underestimates. And how about the issues of underserved countries?
It's a great question, and I think that the challenges within a country like the United States or the UK or others, you know, in Europe or North America, are massive. I mean, there's huge challenges to get the right diagnosis, to get the right care, to get access to those medicines, and then to your point, now move outside of North America and Europe, and the problems get exacerbated even further. And so there's sort of challenges at every level, and I think that, you know, as you correctly described, you know, the first step is find the patient, get them diagnosed, and I do think artificial intelligence is going to be helpful. Actually, there have been AI companies for more than the last 7 years that have been doing this kind of work. They use EHR data, they apply AI to them to find patients that have patterns that look similar to patients who go on to get the diagnosis, and then they try to figure out, based on this patient's pattern, where might that patient be, what hospital might they be in, what's their zip code, and then can we get a sales rep to that area and maybe get them to learn about this disease, get them on the right drug. That's done in a very bespoke way by specific companies for specific pharmaceutical companies. That's not done systematically, and it would be a dream if that sort of work could be done systematically. So AI can be really powerful to help to identify patients, potentially get them a diagnosis. Where we're really focused is once you have a diagnosis for a given rare disease, can you use artificial intelligence to ask the question, among all the drugs that are FDA approved today, what drugs could potentially be useful for the disease that you're dealing with? So using all the world's biomedical knowledge and all that we know about drugs, diseases, targets, genes, proteins, what drugs that already exist might be useful?
So let me bring that right down to brass tacks. What's IL-6?
So IL-6 is interleukin-6. It's an important pro-inflammatory cytokine that is very important in Castleman disease, but actually it's important for all of us.
How about in your life?
Oh, in my life. So interleukin-6 is.
I have to say, this is an extraordinary story. This comes back to the beast. You know, for those of you, just if I may put this in context, here's a young man, everything to live for, wonderful family, what I understand, relationship, incredible career, incredible skills, and you get this diagnosis of a disease at the time that you think is almost unheard of, and you start ferreting around in the medical literature, and IL-6 pops up, interleukin-6.
That's right. Yeah, so to continue that, I mean, basically that's the only thing that was known about Castleman. So, okay, in Castleman disease, you get really sick and you die, and the reason we think that you get sick and you die is because your interleukin-6 levels are too high. And that's sort of the extent of things. And so I guess I'll share a couple anecdotes related to that. So first off, a dear friend of mine, Kazu Yoshizaki, he's a physician in Japan. He was the one who actually discovered that interleukin-6 was elevated in Castleman disease, and he actually went on to develop a drug for Castleman's called tocilizumab, blocks the receptor for IL-6. And I was recently speaking with Kazu and our friend Makoto, who's a mutual friend, and Makoto said, you know, David, Kazu, he didn't just develop tocilizumab, he actually gave it to himself. He was the first patient ever. And I said, no way. And so I went to Kazu, Makoto just said that you gave it to yourself. And he said, I didn't give it to myself, the nurse gave it to me. So I said, okay, Kazu. So Kazu, he actually discovered IL-6 was important. He developed the drug that blocks the receptor for it. He gave it to himself, and he survived. And then he went on to develop it and get it approved for Castleman's in Japan. This is really important because this is the first ever, not FDA approved, but the first ever approved drug for any form of Castleman disease. Happened thanks to Kazu's work in Japan. Now that drug never got an approval here in the United States because it was a rare population, and the drug company decided that they didn't want to pursue it in the US. So they repurposed it for rheumatoid arthritis, one of the most common autoimmune diseases. It got approval here for RA. Then it got repurposed again for individuals that have what's called cytokine release syndrome after you get cellular immunotherapy. And so these patients get a treatment for their cancer, it ends up putting them in the ICU. So you take tocilizumab, you save their lives. And then more recently, during the pandemic, the COVID pandemic, tocilizumab was the first drug patients would receive when they're in the ICU for COVID, and it saved many, many lives. And so I share this example because tocilizumab was made for Castleman's, repurposed for a bunch of others. In my case, I ended up surviving and being here with you today, not because of a drug that was made for Castleman's, but I'm alive because of a drug that was made for something else that we repurposed to save me.
And also repurposed an mTOR.
That's right, exactly. So that mTOR inhibitor. So I guess I'll fast forward a bit in my story to basically say that I got an IL-6 blocker. We thought it was going to work. It helped many, many other patients with my disease, and it didn't work for me. I relapsed on this drug, and my doctor explained to me that we were out of options. There were no more drugs in development, no more promising leads. And so I went on a journey. I was a third-year med student at the time to see if I could maybe find a drug that already existed that could be repurposed for me. And I knew that was possible because, you know, as I shared with this example of tocilizumab, many diseases share the same underlying problems and therefore be treated with the same drug. You know, toci could be useful for RA or Castleman's for COVID. And so I thought to myself, maybe there's something out there. And so I got to work. I started storing blood samples on myself in the lab. I started performing lots of experiments on myself. I got a lymph node cut out of my neck so that way I could work on my lymph node. And I thought maybe I could make some progress. Unfortunately, I relapsed, so I clearly didn't make enough progress. But then when I got out of that last flare, which thankfully now has been my last flare, that's when I guess I went into a bit of beast mode for any American football fans. There's this great football player named Marshawn Lynch, and they say he's in beast mode because he sort of like never goes down. And so anyway, I got out of the hospital and it was like, I barely survived that fifth relapse. I mean, there's no reason I should have survived. Said goodbye to my family, said goodbye to my friends, said goodbye to my fiance. I mean, there's no reason I should have survived. But when I did survive, I got a lot of chemotherapy. When I did survive, thanks to the chemo, I was just relentless for the next about 4 to 6 weeks, where it was like, I'm just going to run every experiment that I could ever think of and ever possibly run on those blood samples I'd been storing, on the lymph node I'd been storing. And from within all of those samples and the data, and between not sleeping and working around the clock, this signal emerged that a communication line in the immune system called mTOR. So your immune cells use mTOR to basically communicate with other immune cells and to say, this is something to attack, something not to attack, let's turn on, let's turn off. Turns out that was in overdrive for me. And that was like the most exciting moment for me because there are drugs that block mTOR.
What was the moment in the lab when you saw that?
It's hard to even put into words what it felt like.
What did you see? What happened?
So basically we did, so first off, we had a sense that maybe mTOR was activated from what's called serum proteomics. There was a signature of it, but I wanted to confirm it. And that lymph node I mentioned, I got cut out of my neck. So your lymph node is your home base for your immune system. It's where your immune cells go to basically come up with their plan of attack. And so I had that lymph node taken out, and so now I wanted to confirm that mTOR was actually turned on. And so I did this really inexpensive stain, immunohistochemistry for a protein called phospho-S6. When PS6 is on, that means mTOR is on. So I stained a bunch of other lymph nodes for PS6, stained my lymph node for PS6, and I kept looking at all these other lymph nodes and they came back blue. So if the pathway's turned off, the cells are blue. If the pathway's turned on, the cells are brown. And I keep looking at slide after slide from all these other ones. I'm looking at the microscope, they're all blue. And I'm like looking, are there any brown cells? I'm like,
please.
Yeah, well, in the normal, in the non-me, I was, I want to see, is there, and I would see there's a brown cell, there's a brown cell. And then I get to my lymph node and I throw it under the microscope and I'm just like, my jaw just drops because it's not like I'm looking around for like a brown cell mixed within the blue. It's like, it's all brown. It's just like every cell, and I'm getting like goosebumps just remembering it. Like every cell is just brown. And I was like, oh my gosh, the thing that we think is on, it's on.
Yeah.
And the best part about it is that thing that is on, there's a drug that can turn it off. Like sirolimus, rapamycin, this mTOR inhibitor, it completely shuts the switch off.
Crazy.
So this is the problem. We have a way to turn it off.
Unbelievable. And here you are.
Yeah, so within 2 weeks of me looking under the microscope, I went down to NIH to show the data to my doctor, and he looked at the data and he basically said, if I had any other options, I might try something, but we're out of options. I mean, I had relapsed and nearly died 5 times from this disease. No drugs existed that had been tried in Castleman's that hadn't been tried in me. So he said, I'm going to give it a try.
So if there's anyone watching this who doubts the potential of repurposing medications, they have to listen to what you've said, because if it's a patient, a parent, a researcher, this to me is, there must be a way of multiplying, expanding the capacity across 7,000 diseases to find medications, as you say, that have common pathways.
Yep.
I know this is like a true passion of yours.
It is.
Understanding that we don't have 3.5 days to discuss it, can you just sort of give us the high level, this is what's coming?
Yeah. Well, the good news is what's coming is here. And what that is utilizing artificial intelligence to look for the same patterns that I was looking for in this one particular, in my blood for my disease, but to look for those patterns across all diseases and to look across all possible drugs that could maybe revert or abrogate those patterns that we're seeing in the disease states. And so the reason why I say it's already here is because the technology is absolutely here. The artificial intelligence tools and approaches exist today and they're capable today. They just haven't been deployed at scale yet.
But Dr. Fajgenbaum, do we actually have also the big clinical trials we need to allow patients to enter these spaces? Do we have the FDA's understanding that this is a space that can be exploited for good for patients who would otherwise have no clear options? I mean, are those pieces in place as well, or are we in this discovery phase?
You're right. It's early days for that other stuff. So what is here is the technology to be able to connect a drug to a disease at scale. So all drugs versus all diseases using the world's biomedical knowledge. I launched a nonprofit called Every Cure about 1.5 years ago, and we are literally using what the world knows about everything, medicine, science, technology, to ask artificial intelligence algorithms, what is the likelihood from 0 to 1? What's the score for every drug versus every disease? So all 3,000 drugs, all 20,000 plus diseases. So 60 million possibilities. If you tried every drug for every disease, you'd do it 60 million times. So we generate a single score from 0 to 1 for the likelihood.
It's just an Excel spreadsheet.
It's just, interestingly, it does actually spit out a giant Excel spreadsheet. It's 60 million rows long. So, and so you get this rank ordered list from 1 to 60 million for every drug versus every disease.
What's caught your eye?
Well, a lot of things have caught my eye. I mean, a few that I'm excited about: Bocitinib for ALS, arginine for sickle cell disease, folinic acid for autism. These are all diseases that are, that are challenging, that they're debilitating, and these are drugs that exist and they're right around the corner.
Also with good risk profiles.
Yeah, exactly.
In other words, if this sort of thing was to go forward within the FDA, it's sort of risk benefit. This is it seems to me a very, very good option.
Yeah, because so basically we can look all versus all, we can come up with these top hits, and to your point, we can filter and say, we just want to use the drugs that have a really good safety profile. Let's eliminate all of the sort of stuff that might be controversial or unsafe. I mean, they're all FDA approved, but even among the FDA approved.
That's the beauty of repurposed drugs.
Exactly.
Correct.
Exactly. They're all FDA approved, and so that's one piece. So we are nailing that. That is what we are totally focused on right now. To your point, coming up with a match and saying that I think arginine would be a great treatment for sickle cell disease, that's one step. Once you've said, okay, it looks promising, the next step is, what's the evidence that you need to generate to be able to really convince yourself or anyone else that it really is going to work? You can't just say, hey, this AI algorithm says it's going to work, right? That doesn't work, right? The AI algorithm can say it might work.
Hypothesis generating.
Hypothesis generating. And then the next step is to go into the lab potentially to confirm it, and or go right into humans, clinical trials, off-label drug use. That can then happen sequentially.
And I think we're at a pretty literally in this space, a thrilling moment in time, because for the first time, FDA is now using natural history data, biometric data.
Yeah.
I mean, this is a unique opportunity to start real-time testing.
It is.
And I think the audience needs to have in the back of its mind that we're talking about illnesses that 95% of patients with rare diseases have no therapy.
That's right.
So just as you were describing in your own case, I mean, these aren't doors opening, but these are doors creaking open.
I agree with that. And, my case is one example of repurposing, and I'm obviously a little biased because I'm alive thanks to this repurposing example, but there are many other really incredible examples of repurposing. I shared tocilizumab, this Castleman's drug for RA and for cellular immunotherapy, and then more recently for COVID. But the reality is that of those 3,000 FDA-approved drugs, about half of them are approved for more than one disease. So about half of drugs that are already on the shelf have already been studied in one more disease than they initially were started in. And what we're arguing for is not that someone should start doing repurposing. We're already repurposing. What we're arguing for is that we shouldn't stop repurposing once the drug's no longer profitable. Because that's the world we're in right now, is that we already repurpose, we just stop if the drug isn't profitable because it's generic, or if the disease isn't profitable because it's maybe too small.
I'm getting goosebumps now. I mean, one of our real passions is creating the profit formula for repurposed drugs with the pharmaceutical industry, because that is actually one of the key stumbling blocks, as you know better than me. I want to move on. Our time together is like flying by like a rocket. So we're talking about cures in rare diseases, but, and again, please excuse me, and please feel free not to answer my questions, but looking after people with rare diseases isn't just about medicine. Caregivers, support staff. In your book, your acknowledgements, instead of being like 4 lines, are 7 pages, right? It's almost like a tenth of your book. No, it's about 5% of your book is acknowledgements, and it's just lists of these people who've helped you. And if I may, please say this. Kathryn appears to be, and your family and your friends and this extended medical family, but she seems to be at the center of all of this. Are you able to talk about the sort of the central role of caregivers in I mean, the beast can fight, but I got a feeling the beast needs a pretty beastful support team.
I totally agree, and I love that you noticed the length of the acknowledgements, because I didn't think that that was enough. That was me, like, that was like my short version of the acknowledgement.
Yeah, and there were actually like sort of 45 names in a paragraph.
Yeah, and it was like, you know, I would have 45 names and say, thank you for doing this thing. And I think that's why I often say that, you know, the book's called Chasing My Cure, but that's exhibit A for why it should have been called Chasing Our Cures, because it wasn't my cure, it was our cure, right? You know, and then cures plural, because we're continuing to look for more and more treatments. And so I guess, you know, what I would say is absolutely, Caitlin has been the champion of all of this, supporting me in every way possible. And one thing that I'd also add to that is that I think this sort of sense of how do you get out of a really difficult situation, how do you emerge from a really big challenge, I think that there's 3 fundamental ingredients to do that. I think that the first is that you have to have a vision for your future, what you're fighting for. If you're in the middle of a fight and you don't know what you're fighting for, you're not going to win that fight, especially if it's a tough fight. So one, you got to have your vision for the future. And Caitlin actually was part of that vision for the future. I wanted to have a family with her. I wanted to marry her one day. I dreamed of that. Plus, I also dreamed of treating patients in memory of my mom, who of course passed from cancer about 20 years ago. And so that vision for the future, that helps you to keep fighting during tough times. The second thing, which is also really relevant to this, is to have support by your side. If I didn't have Caitlin, my dad, my sisters, literally like holding my hand, encouraging me to continue to fight, again, I don't think I could have fought for the amount of time that I did. And then the third piece I really believe in is the importance of one step at a time. You know, if someone said, David, you're going to, so if someone told me when I first was admitted to the hospital, which is, you know, just down the street, they said, David, you're going to be admitted to the hospital for over 6 months. And during that time, you're going to suffer pain that you didn't think was imaginable, and that it's going to be living on the brink of death for months and months. It's going to be horrible. And then when you get out, it's going to happen again and again and again, and you're going to get close, and then you're going to fail, and it's going to be miserable. There's no way that I would have had the strength to get through that, right? I mean, like if you gave me the whole picture and then said, oh, and by the way, the only way you're getting out of this is if you find a treatment for yourself, I would have said, you know, where's the white flag, right? I don't have the strength, I don't have the fortitude, I don't have any of those things to do this for years and then have to be the one to get myself out of this thing, right? But because I took it one day at a time, and not even, maybe even more than, maybe 1 hour at a time, 1 minute, one breath at a time, that sort of helped me to get through those really tough points.
Coming back to the beast, I wasn't going to mention this, but you did. I noticed that after you lost your mum, I'm really sorry about that.
Thank you.
You know, I think a lot of people, of course, go correctly through mourning and you want to do her proud. I completely get that. But that isn't quite what you did. You actually founded a whole organization about, just explain, please.
Sure, yeah. I started an organization called AMF. My mom's initials, her name was Anne-Marie Fajgenbaum, so her initials AMF, for grieving college students. And the reason—
And you wrote a book about it.
I did, yeah, I did as well, called We Get It. You know, my mom was the most amazing person in the world. She was so supportive to everyone. She had this like ferocious work ethic that that I think I inherited a bit of, but she just was the most kind person. And so when I lost her to cancer, and I struggled and coped and grieved with her loss, I started to observe that other people were also struggling with loss during college. And what was so interesting is that none of us were talking to one another about it. We were all struggling and suffering, and we might even be on the same floor in a dorm.
And I also think it's difficult, as doctors, we're not good at opening up about ourselves.
For sure. And so, you know, AMF was this dream that turned into reality, which was, can we create a resource for all of us to actually be able to share how we're feeling? And then secondly, so one is, can we help a lot of people by creating a place to share, which again, I think embodied my mom, because my mom was always the shoulder to lean on when she was alive. But also, it was a way for me to channel my grief towards something positive and to say, okay, I'm going to put my time, effort, and energy into this thing that can help people and keep my mom's memory alive. I mean, people still talk about AMF. There's chapters all over the country of AMF. My mom sort of, her legacy continues.
Yeah, for those of us listening, again, I don't know if other people have noticed this, but you basically deflected the question, which was that this was how you respond to things in this extraordinary capacity to rise up above it. And this is an extraordinary capacity. And I noticed, I read all the acknowledgements in the book, but there's one micro acknowledgement which I somehow caught my eye. Granddad Paddy.
Yeah. Sure. So Granddad Paddy, he's my mother's father. He well, I have a lot of things to say about Granddad.
But what's the bottom line story?
So bottom line is that he started as the mail person at a business. He didn't have a college education. In the small island of Trinidad in the Caribbean. He started as the mail person, and within about 20 years, he rose to become the CEO and the executive chair of the board. And he retired 20 plus years ago in that role. And he, I guess a couple other things I'd say about Granddad. So he was this incredible leader who led with such compassion. People just loved to work for him. They loved him so much that they did well in their work because of how compassionate of a leader he was. And the last thing I'll say is he actually just passed away a couple months ago, which is why you bringing him up here is actually special. So thank you for bringing him up. Of course, I had no idea you were going to.
But again, if I may, I think before the final comment, you basically described yourself.
I don't know.
No, my sense is I've read your book, I've read your work, I've seen the work you've done, and I've seen your, you got a business degree, so why do you start a non-for-profit for repurposing? That doesn't make sense to me. But your mum went early and you responded in this extraordinary way, chapters around the country, as you say. So I got a feeling you're somebody other people love to work for. And I think you're a person who other people look up to, and I think they'll continue to do that. And I just find that it has been an extraordinary experience to listen to you, not only to be inspired by you, but to understand that there are real opportunities in this space. There are millions of parents who are going to be watching this. There are going to be thousands of scientists going to be watching this. And you're going to realize the power of one man. And Professor Fajgenbaum, thank you so much for spending time with us. And I really appreciate it. Thank you.
Thank you so much. This was really special.
Thank you.
All right. Well, that was fantastic. Next, we're going to move on to the morning case study for rare eye disease. My name is Mike Papik. I work as part of the Innovation Ventures team, and we help organize events like this along with Fondation Ipsen. We primarily focused on advancing new technologies to market. Vision has been shown to be a significant component of human quality of life. And there are a number of rare eye diseases that impact many patients at an early age. So I'm excited to moderate this disease aimed at meeting some of those unmet medical needs. In today's session, we've really stacked the deck. I think we've got some exciting talks for you, and followed by an interactive startup panel at the end. First up, I'd like to welcome up Dr. Todd Durham, Senior Vice President of Foundation Fighting Blindness, who will speak about patients as partners on the path to treatment for inherited retinal disease. Todd. Okay.
Sorry. Just a second. Good morning. I'm Todd Durham, Senior Vice President of Clinical and Outcomes Research at the Foundation Fighting Blindness. Would like to thank the organizers, particularly Van, for the invitation to come here. I'd like to start out by telling you a little bit about the Foundation Fighting Blindness to set the context for three main areas where I think we're really doing well, partnering with patients on our shared quest and vision to find treatments and cures for inherited retinal diseases and dry AMD. Our story starts in 1971 with the foundation's founding by families who were impacted by retinitis pigmentosa, and that was the name of our original organization. Gordon Gund is still supporting us. He's still our chairman emeritus. In 1995, organization was renamed from Retinitis Pigmentosa Foundation to the Foundation Fighting Blindness. And today, our mission is really to drive the research to lead to treatments, cures, prevention for retinitis pigmentosa, Usher syndrome, and other inherited retinal diseases and dry AMD. And for all of these, there are still significant unmet needs. Since today we're talking about rare disease, I'm going to focus primarily on what we would call the monogenic inherited retinal diseases. And in our world, there are several, if we think of them as clinical classifications, but there have been at least 270 genes implicated in these conditions. Most common of which is retinitis pigmentosa, followed by Stargardt disease, Leber congenital amaurosis, Usher syndrome, and others that fall under our mission umbrella. In the United States, these are all considered orphan diseases. The combined prevalence is less than 200,000 Americans affected by one of these, where RP represents about half of these. And really, like rare diseases in the comments from the opening presentation and interview that Jim did, we had share the common challenge. That is, how do we estimate the underlying prevalence of these conditions when we don't have national databases, when they're genetically inherited, not always fully penetrant conditions. So this is a challenge that we all face together about rare disease. I'd like to share a little bit about our programs at the Foundation. In the community, many people get to know us through our volunteer chapters. These are ways for families to get to know the Foundation, establish community. We have educational opportunities there, but also, as a nonprofit, we have a large footprint in fundraising through vision walks, dinners, galas, golf events. And every 2 years, we have a Visions Conference, which is really focused on affected individuals and their families. This is a way to share information, have opportunities for networking and community. We have a very active effort underway for advocating for federal legislation to open the door for funding of research we would benefit from ultimately. And many of the researchers and scientists know us very well for our grants and awards program, which is really at the heart of what it is that we do. And something that'll be featured in tomorrow's program, we have a venture philanthropy subsidiary called the RD Fund, and our managing director, Rusty Kelly, will be here representing the RD Fund tomorrow. So I'm going to talk about two of the other areas of our initiatives, the Foundation Fighting Blindness Consortium and the My Retina Tracker Registry. But before I get there, I think would be remiss if I didn't say we believe the Foundation has played a major role in where we are today with over 50 clinical trials in the pipeline for inherited retinal diseases and dry AMD. And we've done this by funding research that's led to the identification of 40% of the genes that are implicated in IRDs. Funding research has led to the establishment of the non-primate large animal models. And we're proud of the fact that research we funded are still in the game today. About 90% of those who've been received funding through our Career Development Award are still active in this field. So we can't make progress when we have people who are dedicated and committed to doing that with us. So let me turn our attention now to My Retina Tracker Registry. Think of this as research of and for patients. This is a registry, global registry that we manage and operate. Its purpose is to accelerate the drive towards treatments and cures by identifying individuals with inherited retinal diseases. And this is an opportunity for us to engage with researchers who are also looking for subjects and participants in their own research. And that could be a clinical trial. It could be surveys. It could be somebody you need to find samples for your research. And really, this is an opportunity also for those participants in our registry to learn about those research opportunities. So this is for their benefit. When they sign informed consent to participate in our registry, they are also consenting to be contacted by us about relevant research opportunities, so they can make themselves available and pursue them at their choice. Today, we have 27,000 participants in My Retina Tracker registry, and over 17,000 of them have been genotyped through our sponsored genetic testing program. I think this is one of the largest genetic databases we have in IRDs in the world. This program, the genetic testing program, has been around since 2017. Its goal is to really understand the relative frequency of inherited retinal diseases, but also to serve as a mechanism to understand and classify these variants more accurately and comprehensively. This is offered to patients at no cost. They are also receive genetic counseling at no cost. One of the reasons why we think this is important, especially in rare disease, is removing the barrier of cost, increases access to genetic tests. I'm showing a slide here from a paper by Peter Zhao and colleagues from the University of Michigan. They did an evaluation of our program in their center. In the year prior to offering no-cost genetic testing at their center, about 51% of their IRD patients had genetic testing, but in the year after, that increased by 29%. And so that's not surprising, but I think the greatest demonstration of the value of this program is we also had a major impact by closing the testing gap among ethnic minorities. Before the program, 9% of Black African American patients received genetic testing in the year after. That went up to 61% of their eligible patients. I think this is particularly important as we all worry about representation in clinical trials and access to medicine, even if they are experimental. And of course, this access to a genetic test has profound benefits for patients. Many of them thought they had one condition and now they have another. Many of them are ready to advocate for themselves and pursue research opportunities. And so unlocking the details of their diagnosis really helps them pursue what's of most interest to them. So now let me talk about the second area where I feel like we're doing a good job of working with patients and working together as we tackle challenges, and that's in our scientific workshops. About this time last year, we had a workshop focused on PRPH2 and associated retinal diseases. This is a 2-day workshop. We invited researchers, academic, and industry representatives, but also patients. And we recruited through My Retina Tracker registry. And in this instance, this was the first time many members of the PRPH2 community even knew anybody cared about PRPH2. So some of them were meeting other affected PRPH2 people for the first time at this workshop. So we feature scientific meetings. We feature largely, and over the 2-day meeting, patient perspectives, both days starting out with patient perspectives. And then the key takeaways, I think the great value here is we are not shielding patients or affected family members from the realities of the difficulties of developing treatments. We're actually engaging them and sharing with them what the real barriers are. And in our case, in this particular case, this event represented the kickoff for a specific funding mechanism that we now offer, funded by the Nixon Visions Foundation. So there's a family who's partnering with us to help remove some of the barriers for successful development of treatments and cures. And I believe the Nixon family is featured on our website yesterday or today, as a matter of fact. But this wasn't the first disease-specific workshop that we conducted. This program started back in 2019 with a family member, a mom of a child who's affected by Leber congenital amaurosis associated with RDH12. And Sylvia, you may have met Sylvia out there. Sylvia was the first to come to us to say, we really need to bring patients into this. And she, like many others, are encountering the real challenges associated with successful development, from capital, finding the right capital, animal models, all the challenges of developing treatments and cures for inherited retinal disease. And we have had a number of these similar disease-focused workshops in CRB1, IQCB1, myosin 7A. We had a patient-focused drug development meeting in June 2022 for RPGR-associated X-linked RP. This is following the toolkit established by the FDA, where we very formal engagement where we have FDA representatives there. And we have another one coming up on March 1, this time for dry AMD. So I think the significance of these workshops, they really focus on patients' perspectives, but they also highlight what are the remaining challenges, where are the gaps, and we are asking for partnership with patients to help us take on some of those challenges and address those gaps. And then the last area I'd like to focus on is our natural history studies. In the presentation, there was a reference to the importance of this. And we are conducting our own natural history studies through the Foundation Fighting Blindness Consortium. Dr. Jackie Duncan here at UCSF is very much involved in our program here at the Foundation Fighting Blindness. In rare disease, we often don't have sensitive and reliable outcome measures with which to demonstrate the benefit of a new treatment. We also don't understand, with the variability of these rare conditions, they don't have a good sense for what we might expect, or understand how long treatment follow-up might be for a clinical trial. And so our natural history studies through the Foundation Fighting Blindness Consortium are intended to address and answer some of these major questions. And we do that with 42 clinical centers of inherited retinal disease specialists around the world, all trained using the same protocols, collection, devices, equipment, and we're really mimicking that process that's followed in large clinical trials. And so I wanted to highlight several natural history studies. The first was the RUSH-2A study, and Dr. Jackie Duncan here from UCSF is the study chair for this. This study is just wrapped up 4 years of progression, and we have learned a lot about retinal degenerations associated with USH2A because of this study, some surprising results. We have another study in EYS-related retinal degeneration called the PRO-EYE study. But I want to focus, highlight today how patients have really helped fund these efforts, help close the gap. In the case of the RUSH1F study, this is USHR1F associated with mutations in PCDH15, and we have the USHR1F Collaborative to thank. They came to the foundation after years of funding research to identify a treatment for USHR1F. They realized that they would need some natural history study to help design clinical trials, and they are partnering with us on this program. Likewise, we just issued a press release yesterday, I believe, for a gyro study for gyrate atrophy. We have significant funding from another family who will benefit from this work. And lastly, we launched a large platform type natural history study called the UniRare study. We have significant funding from families whose children are affected by Usher type 1B. And we couldn't do this work without them. So I'd like to close by saying patients are partners in research and strategy and funding, and we're so thankful. We couldn't do this without them. We're a large organization, but we can't do it all. So thank you very much for your attention.
Thank you, Todd. What a fantastic organization dedicated to rare eye disease. Next up, we have Dr. Van Duesterberg from UCSF to speak about Reclaiming My Sight, a patient-scientist journey in finding her cure.
What an honor it is to be here for me to share a personal story of my rare disease. I want to take a moment to give you guys an insider perspective of what it's like to be a patient, a researcher, and an advocate. My journey started when I was in high school, and my friends and I were going out to look for a meteor shower. And through audio cues, I could hear my friends viewing a spectacular shower that was happening in front of them. But for me, I couldn't see a thing. Could see bright stars in the sky, but that's about it. And that's when I knew that there was something wrong with me. But at the time, optometrists didn't know what to make of it. I'd go to my annual visits and they would tell me, oh, you know, just adapt to the dark a little bit longer and you'll adjust. Well, for 10 years, time and time again, my vision never adjusted until, let's say, around second year at Stanford, which is 13 years ago. I was told to see an ophthalmologist, and he diagnosed me with retinitis pigmentosa. And at the time, my vision was pretty intact. I could see everything. But now, this is what my vision looks like 13 years later, where if I'm looking at a book, you can see white blotches on the screen, missing pieces, and it takes me a while to process what I'm actually reading, seeing, observing so that I can dodge or navigate around. And recently I went to see Dr. Duncan, who's the UCSF ophthalmologist. And this is what my current field of view looks like on the right. The dark spots represent what I missed in the examination, and on the left is what a normal vision, a normal field of view would look like for a person. So as a patient, going to my ophthalmology appointment every year to measure how much my vision I'm losing is very frustrating. There's no treatment. There's no path of a treatment to even subside vision loss. I just have to go in and see how much I'm losing. So I channel that frustration and anger and combined it with my personal curiosity as a scientist. And I knew I wasn't going to pursue academia and high resolution microscopy again, I'll tell you that. But I wanted to learn how much it would take to study a rare disease enough to get to the point where we can find a treatment. And so after grad school, I started a company to track data, combining genetic data, which is at its nascent stages. Patients weren't getting genetic testing at this time. We were getting 23andMe data, raw data, and combining it with lab data, blood work, lab samples, and start digitally tracking for customers what a comprehensive health map would look like. This was the first time, at least for me and for customers at that time, this is before COVID, by the way, where you can digitize the data over time and see what behavior, what sort of things that you're taking that can alter your baseline. And this way, not only are you informing the patients, but the patients are informed about themselves and having a more educated conversation with their physician to find out what their ailments are and what treatments might be out there for them. During these 2 years was very informative for me because I started to learn to be my own personal advocate. So I actually met a friend of a friend who is a physician at the KCI Institute. And he introduced me to his work where he put together clinical trials for inherited retinal diseases and said, hey, why don't you come up and I'll measure your eye, give you an ERG. It was the first time someone did this to me. I was so excited. Look how happy I was. Look how much stuff is in my eye. No one's going to smile with that, but I was, because I get to quantify myself. I get to collect the data. And not only that, that year, around 2020, I found out there was a free genetic testing offered by the Fondation Fighting Blindness. And that's when I met Todd as well. And I found out two things, one of which, if you look on the right, this is what a normal ERG would look like for a patient with fully intact vision. Mine pops up and you see about 10% response of my retina to light stimulus. That was telling that I wasn't just, seeing things or not seeing things. There was actually quantitative data that's measuring my vision loss here. On top of that, I identified that there's 2 trans mutations in the EYS gene. The first mutation actually introduces an early stop codon to EYS, shortening the protein to be non-functional. The second one, which is an interesting one, introduces a mutation right where the spliceosome binds and actually removes the intron. And so that altering the spliceosome binding site can actually cause the protein to express in different ways, which we don't know what that looks like yet. But this is exciting to me. And I wanted to further understand, what does this mean? After all, I am pursuing a career in my rare disease. And so I studied a little bit more about EYS, which is short for eye shut homolog. And it is a 10,000 kilobase gene that's spread across 2 megabases of genetic data. It is not an easy gene therapeutic target. On top of that, we know that there are a human homolog, as you can see here, with all these interesting EGF and LAMG domains that signifies that it's a secreted protein. And it interacts with something related to the photoreceptor matrix stability. And you can see there's the Drosophila and the zebrafish, but guess what? This is the interesting part. The mouse gene is not represented here because mice don't have EYS. So when you're talking about looking at drug targets and modeling rare diseases, we do have some candidates, but if you're looking at each point mutation along EYS along the 10,000 kilobases, you want to be best representing your disease and your pathology. And so I asked Dr. Duncan, is there any way we can model this and using patient-derived stem cells? And she said, actually, there's a guy I know named Dr. Deepak Lamba here at UCSF that creates patient-derived retinal organoids. I was ecstatic. I was like, what? Right in our backyard, literally, Parnassus. I'm 5 blocks away. So I walk up to him, I talk to him, and I said, do you guys study retinitis pigmentosa? Like, no, not typically. Retinal organoids, we only grow them maybe at best 300 days. And so when you're talking about late onset RP, we're not sure if this will model that. And so, well, let us try. So Deepak and I got an NIH grant. And that day, I became a patient to a researcher, collecting my own blood to make stem cells to make retinal organoids, to see if we can model retinal organoids, use retinal organoids as a model for late onset retinitis pigmentosa. And these are my organoids compared to wild type. And I wouldn't be able to do this if it wasn't for obviously me as the patient, but also advocates like Dr. Deepak Lamba and Dr. Duncan, and also my intern Christine here, that's here in the audience today, because she is my hands and eyes to help navigate and make this happen. This year, we're hoping to publish a paper to see if this actually does a good job of modeling late onset inherited retinal diseases like retinitis pigmentosa. But my time at UCSF opened up other opportunities and made me learn that modeling diseases is actually quite important. It's the first stepping stone to not only understand the disease, but have a high throughput pathway to figure out whether new or old drugs could be a potential target. And prior to UCSF, I actually worked for a company called Polara. They hire cure guides, whether they're scientists or doctors, and recruit them to develop a roadmap for patients and foundations with rare diseases towards a treatment. And to this date, we've looked at over 24 rare diseases and have found a dozen potential drug targets that are over the counter for some of them. And some of them are actually nutraceuticals. Patients can actually get their treatment today. We don't have to wait further down the line because they're able to find hits on their avatars. We did everything from yeast, fly to fibroblasts for these patients. So we're really excited to demonstrate that there is a pathway for patients to find treatments today. We just need to find a scalable way to model their disease. But there's more to the patient journey than just patient and treatments. I learned the very hard way, actually, this past year when my brother was diagnosed with glioblastoma. And there's an aspect of patient care that no one talks about. And it took 3 sisters and my mom to take care of my brother in order to manage his prescriptions, which are about a dozen or so, his doctor's appointments, which is on a daily basis, as well as his day-to-day care to make sure he can eat, can walk, can get stronger, and to get to his treatments on time. So I just want to recapture the rare disease journey, because it's not just about the patient or the researchers, but also the caregivers that provide the whole holistic support for any disease, let alone a rare disease team. And I shouldn't even be my own researcher, to be fair, but here we are. And I hope today that I can inspire more folks to help patients in this journey. And there's many facets to do that. And I want to take the opportunity to thank my advocates, Dr. Jackie Duncan and Dr. Deepak Lamba, as well as Christine, who's my intern, who are my hand and eyes when I couldn't see, changing media to make my retinal organoids even. And I want to thank also my family and the Foundation Fighting for Blindness that got me here today. I want to also share my reclaiming my vision, both figuratively and physically, with a poem that I wrote for Rare Disease last year. It's titled From One to Many. You may not know my vision loss unless I use my cane to navigate dimly lit places and detect uneven terrain. During the day, when there's plenty of light, I navigate and socialize like those with normal sight. When the sun slowly sets, I anxiously rush home to prevent myself from getting injured, lost, or alone. As I only have 10% of my vision intact, I plan to use what's left of my vision to make an impact. Trained as a PhD, an engineer, and entrepreneur, I hope to develop a streamlined process to find a cure. For every mutation and every gene, I'll model using patient-derived cells of their specific disease by growing millions and to test dozens of drug cocktails. I hope this path of end of one I pave will be seen and heard throughout industry, academia, and regulatory, even though it seems absurd to develop a cure for only a few, but eventually these few will change the landscape, and then the few become many. Thank you.
Thank you, Van, and thank you, Todd. It's my pleasure to introduce Mike Ehlers. Mike was an undergrad at Caltech in chemistry. He went to Johns Hopkins University for an MD and a PhD. And he became a professor at Duke and a Howard Hughes investigator at Duke. After like 12 years, he went to the private sector. He went to Pfizer and then to Biogen, where he led a lot of programs, specifically in neuroscience, immunology, excuse me, and biologics. And about 5 years ago, he joined Apple Tree Partners, where he is the chief scientific officer and a venture partner. And he's founded a number of companies. I noticed that a few of them are in rare diseases and also ophthalmologic rare diseases. And today is going to be talking about one of them called ART-SCID. So thank you, Mike.
Okay, thank you, Charles, and thanks to the Foundation Ipsen for organizing this meeting. What I'm going to tell you about today is a little bit of a story. It's going to start with a bit of hardcore molecular biology for those who enjoy that, and how that kind of ancient biology has really inspired new technologies that have the prospect of treating previously intractable disease. First, I'll just provide my conflict of interest statement. You'll hear a bit about ART-SCID Therapeutics, where I've been founder, board chair, and CEO. So as an outline of the talk, first, I'm going to tell you about RNA at the origins and crossroads of cellular physiology. Then I'm going to give a general overview about new therapeutic modalities that are derived from or directly target RNA. I'm going to provide this one specific example on trans-splicing and the editing of pre-messenger RNA at kilobase scales. And then we'll link this to the clinical program that we're advancing, which is large-scale RNA editing of a gene called ABCA4 for the treatment of Stargardt disease, which you already heard about from Todd. So considered the primordial macromolecule of life, RNA in its many forms plays a central role in nearly all aspects of cellular physiology, from classic involvement as the primary initial product of gene transcription with pre-mRNAs to mRNAs, to structural mRNAs that deliver amino acids for peptide bond formation and transfer RNAs, to structural components of large ribonucleoprotein machines like the ribosome, like the spliceosome, to more recently discovered functions of RNA like microRNA, the left, using the dicer and argonaute systems to essentially regulate translation and different functions. And maybe more recently, things like long non-coding RNA, which can act as scaffolds or signaling or allow for the formation of biomolecular condensates. Many varied, diverse, and central roles of RNA molecules in cellular physiology. So perhaps it's not surprising that recent years have seen an explosion in the number of therapeutics that are either RNA molecules themselves or directly target RNAs or the metabolism and modification of RNAs. And this has really been more than a 20-year journey for therapeutics that are now approved in the clinic for a variety of indications. One of the first was antisense oligonucleotides, essentially sets of modified oligonucleotides that recognize target RNA, form RNA-DNA hybrid duplexes, and thus incur recognition by an innate immune sensor, RNase H, to degrade the RNA and thus reduce the expression of the encoded protein. First approved antisense oligonucleotide was in the late 90s, and there have been a handful of others that are now out there and approved for ALS, for transthyretin amyloidosis, etc. A little more recently, small interfering RNAs, which essentially apply this other ancient biology of microRNAs, originally discovered by Craig Mello and Andy Fire, a natural cellular system, goes all the way back to plants, that allows for translational control, amongst other things. A different category of antisense oligonucleotides, different chemical structure that intersect the ability of the spliceosome, which you heard about from Van, to effectively or alter the splicing of target pre-mRNAs, approved drugs like Spinraza for spinal muscular atrophy, which has completely changed the trajectory of an otherwise lethal disease of infants and children. Small molecules that are now being developed to directly target RNA, approved, again, for spinal muscular atrophy, risdiplam controlled the splicing of the gene target in spinal muscular atrophy. And of course, everybody, or probably nearly everybody here, has experienced linear messenger RNA in the form of the COVID vaccines encoding a slightly modified version of the primary antigen of SARS-CoV-2. So all these are RNA therapeutics that have recently been, or the last few decades, entered clinical practice, standard clinical practice. But this is really only the tip of the iceberg, because this massive ancient biology is being intersected in a variety of new, interesting ways. I'll just point out a few. Now out there in development are entities which intersect RNA editing, the ADAR system, the adenosine deaminases, which are enzymes that go back far in phylogeny. They're heavily used in cephalopods like squid and octopi to change their phenotypic gene expression and protein coding during environmental challenges, but exist in man in very important ways. Some classic work from Peter Seberg on glutamate receptors. This enzymes, what they do is they can change single bases at the RNA level from adenosine to inosine is the classic example, single base editing, out there being exploited. Type 6 CRISPR systems, so probably an even more ancient biology, back to CRISPR. CRISPR-Cas systems, not just DNA, for those familiar, but there are CRISPR-Cas systems that target RNA, likewise being utilized now to introduce genetic changes at the RNA level. Circular RNAs, these can exist endogenously in cells. We still don't really understand them very much, but confer structural constraints on the RNA and may allow for things like more facile translation from a circular RNA. Another example, tRNAs themselves, so being used in a variety of different ways or intersected to allow for things like nonsense read-through, for example, in mutations that are associated with nonsense mutations. Pre-mRNA transplicing, otherwise you may call it RNA exon editing, which is a different form where one RNA molecule can splice into a second RNA molecule to introduce large-scale changes of a hybrid RNA. I'll come back to this. This was really first described in trypanosomes in the 80s, and in some organisms is involved in 100% of all the transcripts across the genome. It's ancient biology. And another example is self-replicating RNA, which is nearly every positive stranded RNA virus needs to copy their genomes. They do that by using things like RNA-dependent RNA polymerases that can copy the RNA before the RNA makes DNA in ancient viral biology. I could talk at length about many of these, but I'm really going to focus here on pre-mRNA trans-splicing, otherwise known as RNA exon editing. All of those that I showed you before, they're not just random examples. Each of those technologies has led to the formation of a number of biotechnology companies. This is an outdated list. There should be others on here, and some that are on here no longer exist. It's biotech. Things change rapidly. But they all have been the basis for biotechnology companies. I'm going to be talking about specifically a company that I founded with some excellent scientists called Vasidian to tell you a little bit about how we've taken advantage of this phenomenon of RNA trans-splicing to bring a new form of therapy into the clinic. So I think when everybody hears about RNA splicing, they actually think about splicing in cis. This was the kind of unusual observation even back in the 80s at the time that genes are in pieces, as Wally Gilbert said. So when you hear RNA splicing, most people think about this, which is splicing in cis. You have a pre-mRNA with exons that encode the protein of the gene product, introns that don't. What are they doing there? And those introns are removed, the exons are brought together to produce a mature messenger RNA shown there, exons stitched together. That is RNA splicing in cis, and that's the canonical pre-mRNA to mRNA that translates into protein. And that occurs through a quite elegant, complex set of reactions and ribonucleoprotein complexes that are increasingly well understood. It's incredibly complex and highly conserved across evolution, involves this multi-megadalton spliceosome, which is a ribonucleoprotein complex. And it occurs in a variety of steps as components get added and removed, and then two specific chemical reactions happen. So it starts where there's a U1 small nuclear ribonucleoprotein that recognizes the 5' end of introns. Second step is that there's a U2 small nuclear ribonucleoprotein or a U2 SNRP that recognizes a point closer to the 3' end of the intron, which is called a branch site. There are two specific reactions that will occur. Use this here, which is, first of all, there's cleavage of the 5' splice site, which is coupled to the formation of a lariat structure where you've got a 2' 5' phosphodiester bond that forms between the first nucleotide of the intron and an adenosine residue, adenosine nucleotide at the branch site that creates this lariat structure. And then you've got ligation of the two exons that come together, and that's coupled to cleavage at the 3' site. Then that lariat structure intron is removed and degraded, and the spliceosome disassembles. And this happens over and over again at every intron of the 200,000 introns that are in expressed pre-mRNAs across the genome. Quite well understood. That is all on the left. That's all splicing in cis, which is pretty much what you ever hear about. Much less studied or understood is splicing in trans. So it's splicing in cis because it's all occurs within one molecule. Splicing in trans means that one RNA molecule splices into a second RNA molecule to produce a potentially hybrid molecule or chimeric molecule, collinear molecule like this. And it turns out across evolution, there are a variety of forms of transplicing. There's intergenic transplicing, there's intragenic transplicing. The most common, by the way, out there in biology is on the right. It's this splice leader transplicing, or S-cell transplicing, where a very common 5' sequence gets transpliced onto essentially every RNA or large population of RNAs. Happens in many organisms. I'll show you that. I always ask the question, but particularly if I'm in front of molecular biology, which is how many people have ever heard of transplicing? That's not too bad, because normally it's like close to zero, unless you're in some weird, obscure area of biology. So, but still not even close to 25%. And we're here at UCSF, so we've got so a ways to go. Across biology, this transplicing is a phenomenon that is known to occur in evolution. You see here the percent transplicing events across transcripts. So there are some organisms out there where it's 100%, like trypanosomes, which are on there in the yellow. And you see a variety of other nematodes like C. elegans, many genes undergo transplicing. A lot of it's for operon resolution for the aficionados. And then it's really not particularly prominent at all in higher organisms as you get to deuterostomes and vertebrates, with this very interesting spike right here around this thing, this organism, which is Sionia intestinalis, which occurs right here in the chain, in this phylogenetic genetic tree. Sionia intestinalis is a urochordate, so it's a primitive chordate, ancestor of vertebrates. It's a sea squirt or a tunicate. It's otherwise known as an ascidian. So that was the basis for this. Is an ancestor of chordates, which are like all of us. So inspired by this known biology that you can take one RNA molecule and splice it to another, and that can create this hybrid molecule, we envisioned that what if we got really good at being able to do that on demand as we wanted to, with designer RNAs that we would design toward therapeutic purposes. And the idea would go something like this, right? Which is you'd engineer a molecule, I'll call it an exon editor, that can conduct a trans-splicing reaction by recognizing a target RNA. Now you can imagine a situation where if you've got a gene where the thick bars are exons, the thin bars are introns, it gets processed during splicing in cis, and there might be patient mutations that can occur in a variety of different exons and so forth. If you are able to do this, you could envision replacing multiple exons at a time to potentially correct many different mutations or changes that you would want to introduce in a target mRNA. And that's what we set out to do. This process occurs by exactly the same or similar molecular mechanisms in spliceosome process. It's just that instead of splicing by cis, as I described to you, you now have splicing in trans, where you've got a different molecule that contributes either a splice donor or a splice acceptor, depending on whether you're doing a 5' trans-splicing or 3' trans-splicing. Why would this be something of interest therapeutically relative to the variety of different other editing technologies that are out there, including now approved drugs, right, ex vivo, and some other things that are in vivo out there? Well, a few things which I think are potential attractive features of it. It edits RNA and not DNA, so it's not a permanent change to the DNA. I'll show you it edits whole exons, not single bases. Our current most advanced program replaces 22 exons and over 3,700 bases at once in a target gene. No foreign enzymes, so it co-opts the endogenous spliceosome machinery. There's no need to introduce a Cas enzyme or kind of a Frankenstein reverse transcriptase, nickase fusion or something. You could conceivably deliver it in many ways, and because it's RNA, it maintains native gene expression, which may be advantageous in certain circumstances. So how do we do that? The way that we've done that is we took advantage of all the tools. This had been tried, by the way, 20-some years ago, before really the advent of contemporary high-throughput molecular biology, large-scale library generation and barcoding, next-generation sequence, and associated computational techniques. It was the synthesis of all those that we thought gave us the opportunity today to be able to do what could not be done 20 years ago. So each of these exon editors is itself an RNA molecule that's constructed from parts. So it's a multi-component. It contains things like a regulatory domain. It contains the coding sequence, splice site, linker domain, a binding domain that allows you to recognize the target pre-mRNA and some other things like a downstream region and terminator. So it's modular, comes in many modules. And by virtue of being able to take advantage of some bespoke discoveries that we've made, along with large-scale library screening, we can take each of those components and make like every combination of them or many combinations of them, barcode each one, do a single experiment, find the ones that work the best. You can find the needle in the haystack. And that's the fundamental platform technology that we've built at ART-SCID over the last 4 years of existence of the company. Well, 3 years and 11 months, it'll be 4 years next month. And so we've been able to develop learning algorithms in and around being able to make this process better and better. To give you a flavor for this, just a bit on the molecular biology side. So these take these components like the binding domain. What you're seeing depicted here is where we want to target an intron. And so we scan through that intron. Every dot here is an exon editor where we're recognizing that part of the intron. And the y-axis is the edited pre-mRNA. So the upshot here is not every site within the intron is equal in terms of its ability to infer or trans-splicing. And we can identify those hotspots. We screen through many. We couple these target-specific components like binding domains with target-independent components, like I'm showing here, terminators, which are structured RNA sequences, which likewise can have impact trans-splicing efficiency agnostic to target. So this is pretty cool because here, this original terminator was a classic hammerhead ribozyme terminator for those who are in this field. This is our own designed bespoke proprietary terminator sequence, which is already 20, 25-fold improvement. So that's an example. Another flavor, just to give you how we do this, is that we can do all this in combination. So we can do small-scale library screening where here's one where we just combine 3 different splice sites, 3 linkers, 4 binding domains. We did every combination. We see which one works the best. And there's a lot of variability because, as you know, RNA has its own structure. So it's not just a linear sequence. So each of these can be quite different in structure. That's true for the coding sequence. So it's not like we just take the known coding sequence at the base level and put that in. We test all codon optimizations using the degeneracy of the amino acid code, and they're not equal. So every dot here represents a different codon variant. They all code the same protein, but and you're looking at trans-splicing efficiency, you can see it's not all the same. So you have to screen those. And then even the splice site, like if you look at the canonical GU-AG splice donor splice acceptor, there are variations of this, and the immediately adjacent nucleotides, they're not all the same. So we can mine all that data. Every dot on there is an individual exon editor, and you can see that the splice site sequence likewise matters as we construct these things. So the question then being able to do that, what are the really good applications for this? Well, one really attractive application is to go after disease-causing mutations or diseases in which the associated cause are large genes with high mutational variance. Too large to encode in a classic vector like adeno-associated virus, and with mutational diversity such that if you'd have to have a base editor, you'd need dozens or hundreds of drugs. Now, there are other applications I'll mention that could be quite useful for this technology, like dominant diseases, where in one reaction we replace a toxic sequence with a wild type, but I'm going to focus on this. And you're probably already seeing where I might be going with this, which is because of this, we chose as our lead program the most common single gene cause of inherited blindness, which is Stargardt disease. You heard about this from Todd. Stargardt disease, this is a macular dystrophy. It's a progressive blinding disorder, 30 to 40,000 patients in the US. It's caused by generally loss of function mutations in a gene called ABCA4, which encodes a transporter. This is a retinoid transporter that's present in the outer segments of photoreceptors, and it's involved in this whole visual cycle of visual pigments in the retinal cycle. It's autosomal recessive, so you inherit one mutant copy from mom and one from dad, and loss of this leads to buildup of these retinoid intermediates, associated lipofuscin accumulation, and over time you get these degenerative changes that occur in the macula over time. Key things are ABCA4 is 6.8 kb, that's too large for AAV packaging, can't do that. And there are at least 900 known mutations across all 50 exons of the ABCA4 gene. So it's not a bad place to start. So that's where we started. I'm going to show some data about how this works. So one of the first things we did was engineer a very simple cell model system, a hex cell line. Hex cells do not express ABCA4. It's pretty much photoreceptor specific, although a little bit in RPE. So hex cells don't express ABCA4, this ABCA4 protein. So we knocked in a strong promoter in front of the ABCA4 gene in the hex cells to induce expression of the RNA in the protein. Then we took that knock-in cell line and we introduced stop codons in the first, early on in the sequence here. So that now, and that's over to the right. So now it's no longer expressing protein, but it's still expressing the mRNA, the pre-mRNA. It's just that it's got stop codons. And then we can screen and introduce our exon editors. Here's a couple of them, which what we're doing now is we're introducing RNA exon editors that replace 22 exons at once. And you can see that when we do this, we get rescue essentially up to wild type levels of the parent cell line. And we know that this is functional ABCA4, because ABCA4 is a transporter. It's a transport is coupled to ATPase activity. We can measure ATPase activity biochemically. And you can see that over here. You get a robust ATPase activity of this trans-spliced rescued ABCA4, and it's stimulated by ATR, that's all trans-retinal, which is the known biological function. So it's functional transporter restored to essentially wild-type levels in these cell lines. Well, we wanted to know what about relevant human cells for that. So to do that, we collaborated with the Lions Eye Institute in Tampa. We obtained donor eyes. These are usually eyes donated for corneal transplants, but you also can take the retina, culture them ex vivo, and then introduce our RNA exon editors, again, by an AAV transduction, directly into human photoreceptors. And that's what you're seeing here. So this is human photoreceptors where we're able to achieve 40 to 50% of RNA replacement in the relevant human cell type. We have taken this in vivo, and for these, by the way, the therapeutic, I know some of you will be familiar, the Envision therapeutic is itself an AAV that encodes the RNA exon editor. So an AAV episome expresses this exon editor and does so essentially permanently. And it's delivered by subretinal injection, much like Luxturna or RPE65. And just in case there are those who haven't seen, this is in a non-human primate eye, but this is just showing what a subretinal injection looks like. You're going in the back of the eye. The surgeon is placing there a needle to make a small retinopathy. The retina, of course, is transparent, and you see there right by the macula up there to the right of the optic nerve. You raise a bleb. The AAV virus gets injected into the bleb. It's going to transduce the photoreceptors. The photoreceptors are going to express this RNA exon editor, and that's going to trans-splice into ABCA4. That is the therapy. So showing some of that data in non-human primates. We've dosed over 50 non-human primates at this point. This is just showing by OCT what that looks like in a tangential view. Again, you're here close to, you're in the macula, close to the fovea, you raise a bleb. That bleb settles down over a few days in the monkey eye. And then we're able to take the eyes from the monkeys and measure the degree of transplicing of ABCA4. These are not mutant monkeys, not human primates. They are wild type, but we can nonetheless measure it. I can tell you how we do that in more detail. But in the non-human primates, really at least 50% replacement at the RNA level of ABCA4 and 30 to 40% at the protein level. We've developed a mass spec assay where we can measure the human-specific trans-spliced ABCA4 protein that we have. And it's nice because we know a couple things. One, we know is that ABCA4 mutation carriers are essentially phenotypically normal. So that would argue that a 50% correction should be phenotypically indistinguishable from carriers. But we also know from knockout mouse experiments that as little as 10% expression of ABCA4 in the knockout mouse will reverse phenotypes. So we think we're in a therapeutically relevant range. Importantly, I'd say with that is we've taken this forward. We've had discussions now with IND, and I'm really pleased to report that our IND was cleared and approved for us to be able to initiate trials in patients beginning in the coming months. And we are beginning enrollment for these phase 1/2 study in Stargardt's patients. This received a bit of attention. This is last month, by the way, because to my knowledge, I think it's only the second in vivo editing technology of any sort for which the first jurisdiction cleared for human testing was the US. And I'm very proud of that because that really indicates the rigor that we've put into this program and intentionally wanted to go through FDA first off to make sure that we were meeting the bar for this. And that dialogue has been incredibly productive. I'd say we're not stopping with Stargardt's. I'll just say we've got a whole other pipeline of stuff. There are lots of different types of diseases and targets you can go after in a variety of ways. And I think there's a lot of potential for this, but what we've been able to show, at least to date, is very consistent reproducible RNA XNA in vivo, in vitro, and ex vivo. Whether it's cells, mutants, patient-derived cells, they didn't really show, human retinal explants, exon editing non-human primates. I'll preempt a question because we've gone out to 6 months in the non-human primate. It doesn't change. It's going to be a single dose therapy. And we think that this could be potentially useful for a broad number of diseases, including a number of inherited retinal diseases, right, which you heard from Van about another good example, because there are a number of these inherited retinal diseases that are large genes with high mutational variability that could potentially be targetable by this approach. So what I've told you, RNA is the origins of life and intersects nearly all cellular physiology. This very basic RNA biology that at the time seems like extremely obscure is unlocking novel therapeutic modalities that are derived from or directly target RNA. I've told you how trans-splicing holds the potential for large-scale RNA editing at kilobase scales. And we believe that exon editing by trans-splicing holds promise for genetic correction of ABCA4 and Stargardt's disease and has broad potential in IRDs and other disease areas. So thank you very much for your attention.
Thanks a lot, Mike. That was spectacular. Okay, it's my pleasure to introduce Gary Hammer. Gary holds an MD and a PhD from Tufts, and then went to the University of Michigan, where he's been a professor since 1999. He's an expert in endocrine cancers, and in particular, adrenal cancer. And he's gotten a lot of awards and written books on adrenal cancer. He's also founded two biotech companies, one of which he's going to talk about today. And the other thing I want to mention, he was a clinical resident here at UCSF, and as well a postdoctoral fellow in Paul Ingram's lab. And the last thing I want to say is his son, Max, is here. And Max is an MD PhD student here at UCSF right now, today, this year.
Giving class.
Okay, thanks a lot, Gary.
Thanks, Charlie. Are we lined up? Sure. All right, great. Well, Charles, thanks so much for inviting me here back home of sorts. You already gave half my introduction. So delighted to be here. Really hard to follow the first four amazing speakers from both patient perspective, patient scientist perspective, and then just an unbelievable talk on RNA splicing and its use in therapeutics. My 25 minutes will be a little bit of both, a little bit of science and a little bit of journey about how one might advance therapeutics for rare diseases. We'll see if this works or not, this advancer. Maybe not. Okay, my disclosures are the companies we're talking about today. Okay, most of you in the audience certainly will know David Julius, Holly Ingram's husband, who won the Nobel Prize in 2021. You may also know about the 2023 Nobel Prize for concerning nucleoside-based modifications given just this year to Weissman and Kiriko. You may, if you're an endocrinologist, know about the Nobel Prize in 1950 given for the discovery of adrenal steroid hormones. But I'll suspect that only a few of you might know about the Nobel Prize in 1948. Anybody? So this was given to Paul Muller for the discovery of the pesticide DDT. Now, DDT, as you know, was sprayed on everything, including our food and our children, to prevent mosquito-borne illnesses. The challenge was, however, that early pathologic and tox studies showed that the DDT and its derivative DDD was toxic to dogs. And it was quite toxic, actually, despite its use, from semen quality to miscarriages, thyroid function, autism, cancer, etc., and had one particular toxicity, which was that it caused adrenal failure in dogs. So that becomes the beginning of our story today. And it's about thinking outside the box. Because how can we turn a toxicity into a therapeutic asset? And that's precisely what some folks, Bergenstahl, did in the late 1960s, where they thought about the fact that DDT and its derivative DDT, called mitotane, was toxic to the dog adrenal, but they thought about using that toxicity perhaps to treat adrenal cancer. Okay, and the reason they thought about that was on the next slide. But DDD, or mitotane, became the first ever approved drug for adrenal cancer in 1970, approved by the FDA, despite the fact that DDT ignited the environmental movement, founded the EPA, and was banned from the earth in 1972. So that's a problem for therapeutics. That's our drug. Okay, and in 2023, it remains the only approved drug for adrenal cancer. So we have to do better than using a toxin like that for treatment of a disease. Very difficult to dose, extremely toxic, and it works in about 10% of patients. Okay. So that's the problem. So I'm here to talk about solutions. Adrenal cancer is rare. When I say rare, it's not an N of 1 disease, but there's about 500 cases per year in the US maximum in terms of incidence. Most patients die within a few years. Challenge is that there's no understanding of the disease, or at least there wasn't 15 years ago. There's no standard of care. There's no incentive to study because there's no return on investment for big pharma up until recently. And government funding agencies, quite frankly, not enough lives saved to make it valuable. So therein lies the problem for the rare diseases we're all talking about today. Monkey see no evil, do no evil, hear no evil. Doctors didn't know what to do when they saw patients. There's nothing to do. So what's the solution? The solution, as you've heard already today, is all about leverage. And so what kind of leverage? You've heard some of that today. It's about the team. Now, I don't put up Michigan's football team here because we won the national championship, but I do put it up because my journey starts with a coach. The advocate I had was a football coach, Bo Schembechler, not because he was the football coach at Michigan, because his wife died of adrenal cancer. And Bo, pissed as hell that there was nothing to do for this disease, raised a modest amount of funds, a couple $1 million, and said, we need to build an adrenal team at Michigan. So I was in Holly's lab at the time, and Michigan already had some adrenal history. Conn syndrome, which is primary aldosteronism, an endocrine disease of too much hormone aldosterone. Conn was from Michigan, and he had studied adrenal stuff over the years. But as you know, at all of our universities, things come and go, trends happen, people die, diseases stop being studied. But in the modern era, we had other champions, including Bo, the head of our cancer center, and others, who together with our thyroid team built one of the country's first true endocrine oncology programs, multidisciplinary programs to focus on diseases like adrenal cancer. So I came there in '99 to be part of that growth, and I'm so fortunate to say that now as the director of that program, we've grown from 2 people to over 25 clinicians engaged in the true multidisciplinary care required for any rare disease, quite frankly. And the adrenal team at Michigan, what we have done is build international networks that you've kind of heard about with repositories and databases, 2 large international cooperatives with over 100 universities, 5 continents, where we all share genetic databases, and finally collaborate together to both do science, leverage ourselves with funding agencies, and then ultimately try to, of course, develop standard guidelines and practices. The actual adrenal team has grown from just a couple labs to all these labs at the University of Michigan, focused exquisitely on adrenal biology and disease. 1, understand biology, 2, exploit it, 3, develop therapies and partner. So that's what we try to do. My lab is a small lab, quite frankly, anywhere from 6 to 14 people, undergrads, grad students, postdocs, scientists at various levels of their development. And we utilize a variety of techniques, some of the things you've heard today, from mouse modeling and cell-based work to human studies looking at genomic variants in various adrenal diseases, using everything from single-cell technologies to big data analysis. But what we've learned, and you've all heard today, this is an iterative process. One day at a time that we heard earlier today is absolutely critical, and it takes a team. Because what we learn, of course, this is a bit of a tautology, but what you learn in the lab helps you think about what questions to ask in your patients, and what you learn from your patients helps you think about what studies you need to do. And this iterative process of discovery continues again and again, and hopefully you hone things to a level of understanding like some of the stories we've heard earlier today. But it takes teams. You can't do it alone. Everything from basic scientists through clinical practitioners, of course. So when I got to Michigan, that was me. I was a basic scientist doing science over here, and then I was the clinician seeing patients over here. But I never ventured into that valley of death, which is trying to understand how my science may have import for my patients. Slowly that changed, but this was a long process for me, a decade. I've been there 25 years now. But the valley of death is a hard valley to cross, as we well know from all the stories we've heard today. And what I'd like to talk about is how do we avoid the valley of death or cross that valley of death, where classically, of course, academics do science and the industry translate our science. So what I want to explore in 15 minutes or so is how do we avoid the valley of death? And I'm going to start with this video. It's about how to avoid being the stereotype who can't cross the valley.
But Dad, you'll never make it.
Don't you think I know that? Goodbye, son.
Wait, Dad! Don't do it! I won't jump anymore. I promise.
Oh, thank God. Thank God. Thank God.
I love you, Dad.
I love you too, son. You know, boy, I don't think I've ever felt as close to you as I do right. Oh?
Valley of death. How we all feel?
I'm gonna make it. This is the greatest thrill of my life. I'm king of the world. Woohoo! Woohoo!
So science is hard, and translation is hard, and we fail all the time, right? So let's talk about how we can avoid that, or at least one way to avoid it. Discovery is iterative. So the first quick vignette for 3 or 4 minutes is the first company I founded, Milendo, which actually stands for Millie Schembechler, Bo's wife, and endocrinology. Okay. This company was founded by a patient. Riley Crapola was a scientist in the rare disease space at Pfizer. Her husband, Tom Crapola, is a colleague and biochemist at the University of Michigan. Riley came down with a very bad case of adrenal cancer that was rapidly progressive. And Riley, like Bo, was pissed off that there was nothing but a toxic DDT drug to give her. And she came to me, I was her doctor, and came to me and said, I need to work in your lab. And we need to find something more effective for this disease. And she started scouring the literature. And what she noticed from her days at Pfizer was, remember, that when atherosclerotic plaque forms from a macrophage, as it's becoming a foam cell, it sucks up cholesterol, turns it into cholesterol ester, and the foam cell initiates the atherosclerotic plaque. So she knew that from her work at Pfizer, but she also knew that Pfizer and many companies were trying to develop inhibitors of that esterification process. ACAT is an enzyme, ACAT inhibitors. And there were many ACAT inhibitors on the market, and Pfizer's ACAT inhibitor had one toxicity, curiously, and it was that it caused adrenal failure again in dogs. Riley was, and all the ACAT inhibitors were put on the shelf because the statins came out and the whole field was lost. But Riley was interested in the fact that, well, not only does a foam cell use cholesterol ester, but the adrenal that makes steroid hormones, all steroid hormones are derivatives of cholesterol ester. The adrenal stores more cholesterol than any cell in the body, but the liver and a macrophage in its process of becoming atherosclerotic plaque. So Riley thought about exploiting that with cholesterol ester, determining adrenal identity. But she also realized, and we figured out the biology of this in my laboratory, of course, molecular biologists know that, cell biologists know that cholesterol and cholesterol ester are the mediators of integrity of all of your membranes, your Golgi, your ER, your cell membrane. The ratio of cholesterol ester and cholesterol, and then perhaps by inhibiting ACAT, you can do two things. You can inhibit cholesterol, you can inhibit cholesterolification and therefore inhibit steroid production, but maybe you can also kill a cell by messing up the ER and inducing a cell death. So we patented that idea, not a composition of matter patent, and method of use patent back in 2012, and Riley single-handedly, as she was succumbing to the disease, raised $15 million and founded the company with a Series A with colleagues she knew. You have to know people. Networking is critical. She was well connected to the infrastructure. And we founded Melendo Therapeutics with Series A finance led by Fraser Health. And it was supposed to be a quick and dirty trial. Laugh if you like, a quick trial for adrenal cancer. And we did that with a 3x3 design. We started with a low dose based on some dog studies and realized that dogs aren't human beings aren't dogs. The dose was too low, but the FDA doesn't let you change your dosing regimen. It's still a 3, then a 3, then a 3, doubling your dose every 3. And after about 2 years and 63 patients, 21 cohorts of patients, we were just now at the right dose to start seeing effects and seeing tumor shrinkage, and time and money ran out. $15 million gone after 2 years, which is how money is spent. So the company had to pivot. And this is lesson 1 for me. You have to pivot when you have to pivot to stay alive. So we did that, and we raised a Series B of $62 million now with a syndicate to not go after trying to kill cancer cells, but let's block the steroid production that happens in adrenal cancer and other steroid diseases. So easier perhaps than killing cells. So we did that, and then did a phase 2 trial in a hormone excess syndrome called congenital hyperplasia, this time an intra-patient dosage titration scheme, enrolled patients using our big network now. We have 100 universities around 4 continents for these rare diseases, and we were getting consistent, fantastic, said naively, responses in individual patients. Drug blocks steroids, take patient off drug, it goes back up, down and up in the sawtooth pattern whenever we use the drug, but it just wasn't robust enough to get anyone excited, let alone the investors, and had to pivot again. So 2 things didn't work. So this time we engaged in a reverse merger for another $85 million with a company that had failed, unfortunately, sadly, in their quest for a drug for other diseases. So reverse merger, we subsumed that company with some capital and went after other diseases. So you see how I had to let go of my adrenal cancer dream and just keep alive, but still working on helping patients in other diseases. We did that, new asset, new diseases, and fantastic data for a disease I won't go into. And then we had a liver toxicity. As you know, if your liver toxicity is great enough, your drug is dead. So that happened, and the drug died, and Melendo had to pivot. And this time the pivot meant close shop after 10 years. We were now reverse merged out with Tempest Therapeutics, where Melinda was taken over by Tempest, and that story was done after spending $200 million and 10 years, 8 years of time. That happens in biotech, and you have to be resilient. So that was story one. What I did was claw back my patents that we had, got them back to Gary Hammer University as opposed to Gary Hammer Company. So I had the patents in my hand to pursue this later, and we'll see what happens there. So time will tell about the adrenal cancer story. The real story is about Sling Therapeutics, because this goes back to really 15 years of our work in my own laboratory, where we had known for a long time that adrenal cancer occurred in 3 primary familial genetic syndromes, monogenic diseases of Wnt pathway activation, IGF pathway activation, or loss of p53RB. So Li-Fraumeni syndrome, familial adenomatous polyposis coli, colon cancer, and Beckwith-Wiedemann syndrome in children who have IGF amplification due to the imprinting defect in the IGF-2 locus. All of these folks get adrenal cancer. So we knew that these pathways were involved, okay? And this is about IGF-2. So we studied it for years, just grad student PhD theses, postdoc theses, where we basically show that, yeah, IGF is important. One, we can overexpress IGF in cells and they grow faster. We can make mice that have IGF overexpressed in their adrenal. They get adrenal tumors. We combine it with Wnt activation. These 2 familial syndromes, we make mice with those gain of function mutations, we get cancer. So we kind of knew it was important. And then we started studying human patients now that we had our database and all of our consortium. And whenever we did a study looking at the genetics of adrenal cancer, IGF-2, the big red line, was the most upregulated gene in every study ever done in adrenal cancer by 100-fold. So correlation, but seemed to be important, the IGF-2 expression. Okay. And then companies, as you know, started making drugs for every single growth factor and growth factor receptor out there. There were drugs for all of them. Matter of fact, 26 IGF receptor antagonists were made. Small molecules, antibodies. And as you know, in the early days, companies took their drugs and studied every common cancer, looking for the big bang for the buck. Breast, colon, prostate, for example, with IGF inhibitors. We had studied it in cell lines, and this just shows you, yep, we can use small molecules, lincitinib, the drug we're talking about today, monoclonals, and we could block activation of the pathway. We can make xenografts, or look in cells and block growth of adrenal cancer cells, and we can do xenografts and show that when we block the receptor, boom, human adrenal cancer shrunk to nothing in mice. But once again, mice aren't human beings. So that's where we were until, as I developed an adrenal cancer, we developed an adrenal cancer program at Michigan. We now see about 25% of the country's adrenal cancer at Michigan, see patients from around the country now. Like many rare diseases, there happens to be centers that have resources to study. So as we started studying patients, we were able to start thinking about maybe actually doing a clinical trial. So I begged and pleaded with Big Pharma, and as you might predict, there was no interest at all in a disease that affects 500 patients a year. I got no traction. Finally got Pfizer to lend me some drug for an investigator-initiated trial, and after begging and pleading, and did a trial and saw this kind of waterfall plot. Below the line means tumor response. We saw some biologic response, albeit pretty modest compared to what we saw earlier, pretty modest responses in patients. But I saw a signal, but only 4 months duration, maybe 10, 15% of patients. Then I begged and pleaded, that was an antibody, begged and pleaded for a phase 2 trial, did a phase 2, same thing, 10 or 50% of patients. But I just told you the IGF pathway is disrupted in 100% of patients. So there was no interest in this. But then my grad student published our preclinical papers, and a company called us saying, we've been watching you, and we have the same story. We have a small molecule against the IGF receptor, and we want to do a phase 3. Are you interested? Well, yeah. So now we had our consortium, or we had our consortium of 100 universities. We said, we can do this for you. And so we did a phase 3, industry sponsored this time, 100 or so patients with adrenal cancer. And this time we got the same result, but with more patients. So under the bar are the responses, but this time it's still just 15%. This is placebo controlled. So in the placebo controlled trial now, you'll see all placebos are above the line. Under the line, there are a number of responders. But this time, we had 15% response, 80% tumor shrinkage with response time of up to 3 years. That doesn't happen in adrenal cancer. These kind of responses, complete resolution of lung metastasis after multiple doses of chemotherapy. But again, 15% of patients. So the return on investment was not there. The FDA said, nope, your response, even though the only therapy we have, by the way, is the pesticide DDT. This was not good enough for the FDA, right? 50% of patients, when the mutational sequences in every patient, it works in 15%, and the company just couldn't do it, bye-bye. So there's no return on investment. So all 26 IGF receptor inhibitors were put on the shelf. The adrenal cancer was the only one of the Ewing sarcoma where there was any biological signal, but they're all put on the shelf for every cancer for about 7 years. So what do you do next? Well, I didn't want to stop doing work, so we had to pivot. So about leverage. So because the program we had developed in rare endocrine cancers now had this consortium, when the TCJ project at the NCI also was just studying common cancers, we went and they were having trouble even getting 500 colon cancers because people didn't want to collaborate with each other. We said to them, we'll get you 100 adrenal cancers tomorrow. And they said, no, you won't. We said, yes, we will. No, you won't. No, we will. So we got them to do their first 3 rare cancers: thyroid cancer, adrenal cancer, pheochromocytoma. This is about the adrenal cancer TCGA. This is years later after that the project. What we realized was that adrenal cancer came in 3 flavors. This is a genomic profile. These studies cost the NCI tens of millions of dollars, so this can't be done clinically. But we were able to genomically classify adrenal cancer into three kinds of flavors, let's say, with different signatures. We then did just a simple data decomposition. This has nothing to do with the 3 flavors. We just took all the data, put it in a blender, and said, what are the 3 main pathways perturbed? And it was depressing because we didn't learn anything. Guess what it was? IGF signaling, Wnt signaling, HIP-3R cell cycle. We learned nothing after a $10 million project. But we asked the question, these are the same mutations as in the germline mutations, but this is all sporadic adrenal cancer. Well, did these biological processes at all underlie those 3 flavors? And if so, how? So back to IGF. So we did what's called this unsupervised clustering analysis of all the patients in the TCJ project and said, when we do unsupervised clustering with IGF signaling, do we get 1 group or 3 groups, or how does it stratify? And we get 1 group. Again, we learn nothing. Every patient has IGF dysregulation. This is an IGF signature we developed. Okay, we then did the same thing with the Wnt pathway, and we said, let's do unsupervised clustering with what we call a Wnt score, looking at a Wnt activation, and what kind of clustering do we get? This time we got 3 groups. These are not the 3 clusters. These are just 3 independent groups by unhierarchical clustering. 3 groups. But in this time, when we looked at the actual Wnt signature, only 2 of these groups had any activation of Wnt activation with the gain of function mutations in those 2 groups, not the other group on the left. So we then did something different. We went back to our 3 now genomically defined clusters and said, let's do a network analysis and see what transcription factors are in each of these 3 flavors. And let's look at the Wnt transcription factors. And lo and behold, only the 2 clusters had Wnt activation, saying that the unsupervised clustering mirrored the true genomic classification of adrenal cancer, and you have Wnt activation now in those 2 more aggressive flavors on the middle and the stage, whatever this is, stage myeloid. Okay. So then we went after the p53 pathway and did the same thing, and we again got 3 different unsupervised clusters out of groups out of the analysis. When we looked at E2F activation or cell cycle activation. And this time, though, only 1 had significant cell cycle activation. So we again went back to our 3 clusters and did a network analysis to say, where is cell cycle engaged in our 3 now genomically defined clusters? And you guessed it, it's only in the last cluster. So what that tells us, to summarize that, is we were able to distill the biology of the genomic clusters to biology where all 3 have IGF dysregulation, 2 have additional Wnt, and the most aggressive has additional cell cycle dysregulation. That's where a grad student comes into play. Deepika Mohan, very talented student, who was able to distill down this months and months of work of genomic analysis, 3 flavors, down to the biology, and then down to an overnight PCR assay that was able to distinguish cluster 1 from cluster 2 from cluster 3, with a 2 PCR step, which I will not go into. And she had the good fortune of having an entrepreneurial spirit, and she said, let's patent that first. And so for the first time in my career, I said, that makes sense. Yes, so we patented first. The academic dilemma, patented 2 things. One, the companion diagnostic to stratify patients pre-treatment for possible pathway engagement in the 3 flavors. And then we patented a diagnostic for treatment to utilize those for treatment strategies. 2 patents. And then she published a series of papers for her PhD, which were just wonderful. So we had the biology down. Let's talk about the IGF then. So she had this patented diagnostic. We had it. With the idea that that 1 flavor of adrenal cancer, the 1 flavor here was IGF dominant. The hypothesis was that may be where the responders are. It only has IGF mutations. Maybe those are where those responders to the IGF therapy lie. So we went back to the trial, and I have to tell you, negotiating with Japan is hard. Astellas, the company, God love them, took 2.5 years to get the DNA from the sample for which I was the academic PI on. We had all the tumoral DNA. We finally got the DNA, ran it through our assay, and look at the yellow box, which are the responders. When we tested them for cluster 1, 2, 3, for 1, 2, 3, after 2 years of negotiation, every responder was IGF dominant and IGF only, and the non-responders had additional Wnt mutations and p53 mutations. These were long-lived responses, as I mentioned already. So with that, I was very excited and a bit naive, and went back to Astellas and said, gosh, you should do another trial. We figured out how your drug works. And the question was an appropriate one from their standpoint. Well, what's the ROI? And back of the napkin, based on TKIs, was about $300 or $400 million, which to me was astronomical. And I got a chuckle. From Big Pharma saying, well, 500, 800, maybe. No, thank you. So I asked them if they would license the drug to me. And they chuckled again and said, do you have a company? So I said, yes. Founded a company with some help from colleagues. And then went back to them and negotiated a license of their drug for this cancer that they wanted nothing to do with anyway. So that took about another 2.5 years. Gary Hammer, U of M, was easy. That was the license where we licensed patent. And then licensing from Astellas took quite a bit of time. 2.5 years of negotiation with a lawyer who volunteered her time, about $400,000 of her time to help me. You need champions to help you. Whether it's a foundation, patient support group, a lawyer, $400,000 that she was being yelled at from her boss not to spend on Hammer anymore, but she did. The terms of the license, after negotiating down from a much, much higher number than this small number, it was a $350,000 upfront payment that my wife was going to leave me for, because that would be, we'd have to mortgage our house for that. But they were able to defer that until I raised $10 million. But they only gave me 6 months to raise $10 million and then pay them the $350. Well, COVID hit. I could not raise money during COVID. Couldn't raise money. The ROI was again low. 6 months ran out. I begged and pleaded for 3 extensions for that deadline, and was finally given a drop dead deadline of April 21st, 2021. Hammer, if you don't raise 10 million and give us 350, you're done. We're taking the license back. That's where we were. And that's when a pivot happened. I had gone to all the people I know. Wasn't going to work. Lesson: luck comes to the prepared mind. Only. Louis Pasteur. So while I was studying IGF in adrenal cancer, my friends down the hall were studying IGF in a different disease called thyroid eye disease. You've seen the ads on TV by Horizon. Eyes bug out during Graves' disease, completely IGF dependent. Okay. And it so happens that not only do my colleagues, but Horizon Therapeutics bought one of those failed IGF drugs from the cancer trials, an antibody this time. They bought that drug from Vision not for $350,000, but for $140 million. And they tested it in thyroid eye disease. And it worked like a charm. 85% response rate. So that happened. They made a few $1 billion their first year after they were approved. And they published the results in the New England Journal of Medicine. I had about 90 days left to go. And then my phone ran off the hook. People knew that I had the only oral IGF inhibitor out there. All the rest were monoclonal antibodies. And I had to learn to be flexible yet again. And when TPG called me, a private equity firm founded by Fred Cowan, here, my old boss at UCSF, they were interested in thyroid eye disease. And I have to tell you that as I had about 10 more days left before it ran out, with a drop dead date, with days to spare, they were so interested in Horizon's story and what I had that they invested $35 million, and we were given a valuation of $100 million because of Horizon's explosion on the scene. So that's how I founded Sling Therapeutics. And what I had to learn, though, was to let go. And this is critical for academic founders. I stayed on as chief medical officer that year, which nearly killed me, because I was also a professor at UCSF running an adrenal cancer program in the lab, and I was the president of the International Endocrine Society, and I was trying to do this, and I couldn't. I mean, I got the IND written, phase 2b/3 trial written, 50 sites enrolled across Europe and America, wrote 3 protocols and 4 additional patents, and then I said, no, you need a thyroid, you need an eye specialist. I'm an endocrinologist. So you have to know when to step away. So I stepped down. Who knows the Peter principle? The Peter principle is when you rise to your level of incompetence. So I had surpassed my level of incompetence. And I realized that it's time for me to step down, and you have to know when to do that and bring on the team that you need. So I did that. I stayed on. I'm a board member. I resigned. Stayed on as board member with an amazing team. If you know Fahim Hasnain from Gossamer and others, he's our head of our board with TPG and others. Hired a CEO, and then we hired away from Horizon Jeff Kent, who was the CMO for Horizon's trial with TED, and we hired Ray Douglas, who is the founder of IGF's involvement in TED. Now we have the dream team, and I'm happy to say that we have a data readout coming in Q2, Q3 24, and I'm hoping for a nice year for this company. Series B, we just raised another $80 million with a new valuation of $200 million, and we're looking forward to this year being a real banner year where we could really make a difference to these patients. I again had to claw back my patents because they're not doing adrenal cancer anymore. So you have to learn to let go of that too. So I had to let go of adrenal cancer. The only way to get the IGF drug approved is to go after the other disease. What I hope to do then, I got the patents back, clawed them back to Gary Hammer, Michigan, that we will ultimately do an ITT, investigator trial for adrenal cancer, and I'll get that done eventually. So we're making progress, but you have to know, even though I started out with adrenal cancer, I ended with thyroid eye disease. Success has lots of failures. The quick lessons learned: be flexible, leverage, surround yourself with a team, as I mentioned, always. By the way, we're always imposters. None of us know what we're doing this stuff. We're always imposters. I'm like continually imposter. I mean, you have to hire people to help you. This is the first time for everything. It's hard. Magic always happens outside the company. Always. Fear less, risk more. I always feel like a kid on a boogie board on a tsunami about to fall off. But if you follow the science, you'll realize that success is never straightforward. Ground your questions in the best science, ground your goals in the best patient care, and you'll hopefully ultimately get to the promised land. And know how to tell your pitch and tell your story and pivot. Like, what's your ROI? So listen to this guy's pitch as my closing slide.
Being diagnosed with low adrenaline levels was devastating. No one ever warned me about adrenalitis, so I never saw it coming. There's times when you need adrenaline, and I didn't have it. That's when a friend who had beaten adrenalitis recommended the all-new 245 horsepower V6 Toyota Tacoma with the TRD package. Knocked it right out. That truck saved my life.
The all-new Tacoma. Now that's moving you forward.
All right. Thanks very much for listening.
Okay. Thank you, Professor Hammer. That was very excellent talk. So now we're going to transition to the interactive panel discussion section. We're going to bring down a few chairs and some microphones. I'd like to welcome up our panelists. So first we have Dr. Daniel Chung, Chief Medical Officer of Sparing Vision, company focused in developing gene therapies for inherited retinal diseases, and former head of medical strategy at Spark Therapeutics, which established the first FDA-approved gene therapy in ophthalmology. And Dr. Lukas Scheibler, Chief Research Officer of Apellis Pharmaceuticals, which recently approved the first ever therapy for geographic atrophy using complement therapy, former EVP of Acucella and VP of Innovation from Alcon Novartis. While they get ready very quickly, we'll ask the audience to prepare or to, if you guys have any questions. You both have microphones, so thank you guys for being here. Excellent. So while they think about their questions here, let me start by asking you to tell us a little bit about your story. You've both been part of startups that have continued to succeed and grow, and maybe just give us a few minutes of your thoughts on what that journey looked like and your experiences from that. And we'll start maybe with Lukas, if you don't mind.
Yeah, thank you very much. Thanks for inviting me and thanks for everybody coming here. So, Apellis has about 20 years history with Catalyzing. We started off as a little startup called Potential. And I was not there. I joined some of the Apellis. And Apellis started with some experiments done. And it's been out actually from Harvard on price as a business case for so it all started with the broken trees. There was a patient experiment with trees that go Christmas the usual story. So then the founders of the company walked down the hallway and this paper and post license it and that's the start of the story. And what you just presented sort of the ups and downs and left and right and looking into this. I mean, Apellis has its fair share. We have to say we never let that go. So we started as a company with complement to originally called the geographic atrophy and part of the outcome and was in the early days as an hybrid work in I worked a lot on together. And so I got involved in this and the outcome report I know is in the one state plan. It's a long story. Ultimately we developed the doctors, the treatment doctors, we improved their management 3 or 4 years ago. And so we were commercializing and we went back and then also developed it for the treatment of geographic atrophy form of and so we originated from the rare diseases, developed it in rare diseases, and now we pivoted into really large diseases. But again, we never let go of the crisis just last year. Finally got approved to launch the world types of those events before. Not very frequent. Put us into the crisis. On your already public we're back, we're recovering, and so here we are. We are still focused on the main immune system. We are excited, and we all learn in the course is the confidence system with 50, 60 proteins involved. We learn every day. We learn much more. All of us, my age, probably learn in school, in university, where you went, you learned like confidence system, equals membrane plaque complex, and equals, defense against pathogens. And just where we are, and so we are kind of focused, we continue to be focused on technology. With a big effort in this always understand anyone from new opportunity. And we continue to go proof of the rare diseases that we can non-genetic shows excellent proof of the disease. And so the excellent proof of the kidney constellation is used so we are exciting times in front of us.
Wonderful. Thank you, Lukas. Dr. Shah, please.
So great to be here. Thank you for the invitation for UCSF and the Foundation. So my journey is a little different because I was never always enthralled with industry. I was an academic and I still consider myself someone an academic. And the worlds are much less divided down than they were many years ago, and I think that's very important. So for my journey, it was one of those things that I'm totally in retinal gene therapy. I started in 1994 when I did a retinal gene therapy fellowship at the NEI, with the NIH. We were still using adenovirus at the time. So back then, gene therapy was going to cure the world. We had, I think it was Rosie the sheep or something, I can't remember. But the founder came, spoke at NIH, and it was supposed to cure cystic fibrosis. Everything was going to be just great. Well, nobody really thought about the thing called the immune system, seemed like, because that started raising its head up in a way that we are still dealing with the immune system now. The eye, luckily, is somewhat immune privileged, so we are a little bit fortunate with that. So, spent about 12 years at University of Pennsylvania after my clinical training. I trained as a pediatric ophthalmologist with some in ocular genetics. I was fortunate enough to work with a group, Gene Bennett and Alan Dwyer. And they had this idea where could we put a normal copy of a gene back into these inherited retinal diseases and restore some of this function. So with that, I was very fortunate to be part of the preclinical work to some degree and really heavily involved in the clinical development of the program, and then went on to Spark Therapeutics, where we became the first to have an approved gene therapy drug in the United States. And to the talk that came before me, that is such a great illustration of what it takes to get a drug going, because there are just so many steps. And as an academic, you're always thinking, if I could just get a drug approved, it would be great. Well, there's a whole nother thing after that. That's called reimbursement and commercialization and ROI. It used to stand, I keep thinking it's RO1, but it's not RO1. It's an ROI. But anyhow, so we had some success with that, being with the whole Luxturna and Spark Therapeutics, where I led their ocular group towards the end of my tenure there. They were bought by Roche, and a lot of times when big pharma comes in, they like to leave you alone for a little bit, but then they start kind of doing their own thing, and it was the idea that maybe ocular wasn't going to be quite as prominent at Spark. Now I joined a French genomics medicine company called Sparing Vision. We are a genomics medicine company only in the eye. Our main area of concentration right now is in a gene-independent way of slowing down the degeneration of your central and rod cone dystrophy, which the primary example is retinitis pigmentosa. We are currently in phase 1 trials. We just finished our second dose of our dose escalation, and we hope to be fully enrolled. Depending on who you ask, as I like more time, but if you talk for investors or others, hopefully by the end of the year or early 2025. We have have a second program that will hopefully be in the clinic in 2025, which is actually going to hopefully restore function in cone cells that are what we call dormant. They can't do this thing called phototransduction, which is the idea that the light coming in is translated to an electrical impulse and goes to the brain because, believe it or not, the eye doesn't see a thing. It's the brain that translates everything for you. The eye is this huge relay system, however complex it is, but the reality is the brain is the one that really kind of sends the message. And without that message, you don't get that. So those are the two main things in the gene therapy space. And then we're also in a partnership with Intellia Therapeutics to be kind of their ophthalmic arm for gene editing. So this is kind of where we are. We have an office also in Philadelphia in the United States, but we have executives here in the Bay Area and San Diego and Boston. So we truly are somewhat global. So we'll be going to Asia to do some talks there very, very soon. So that's kind of the idea, but the whole idea, of course, I'll just say one thing. When we were at Spark, there were a lot of times when we didn't know what we didn't know because there was no blueprint to follow when it came to gene therapy. And so the development took 10 years, but we had results at 30 days. And so now it's not, we can't take 10 years for gene therapy if at all possible when you have results at 30 days. But let's face it, there's not a lot of that going on right now. Some of these diseases are very slow. So you're taking an endpoint that's going to be 2 or 3 years potentially. And now I won't say that tomorrow to investors, but the idea is that it is going to take some patience. The ocular space has had some challenges because we don't have clearly defined endpoints for all these different diseases. We had to make a novel endpoint at Spark Therapeutics. We had to do our own natural history studies. So the idea is that, and this whole thing of ROI for these rare diseases, we're not talking 100,000, 200,000 patients in eye disease. We're talking tens of thousands at the most. But a lot of these genes, you're talking a couple hundred around the world. So we're in the ultra low population. That's where we're thinking maybe the gene independent approach may go as well. We can get into more detail, but I want to make sure we have plenty of time.
Wonderful, thank you. Do we have any questions from the audience? Over here. Do we have?
Yeah, I think it depends on your approach. If you're doing obviously gene specific, then you're going to go for the folks that test positive testing. But if you're doing gene independent, one of the determining factors is you still have to have some kind of preclinical data, even if you're trying to do gene independent. So really, what's out there when it comes to animal models is that you can do some type of proof of concept, and whether that's animal models or in vitro models, it has to be somewhat robust that the FDA is going to take that. So that really determines a lot of where we're going. And then the idea is to leverage that data, and hopefully you'll have a good safety profile when we go through your clinical development. But then you can start adding other genetic etiologies that might be very similar or the pattern. And that's one thing about RP. For the most part, all of RP is going to start with, because the rod cone dystrophy is going to start with this peripheral field loss, night blindness, and then go to the central vision. And our mechanism of action is really key on that sequence. So it's not addressing the underlying genetic mutation or genetic cause, but it's really one of the pathways that really happens in all our eyes, and that is that there's a neurotrophic factor that's secreted by your rod photoreceptors that keep your cones alive and functioning. So the reality is in RP, the rods are gone, so we just supplement that, but this is a process that's made. So we're trying to take advantage.
Great. Yes.
Yeah, I had a question about wonderful story, how the challenge between your indication for rare disease, switching to a common disease, the different risk tolerance for the rare versus the common disease, and the setting of price point and the challenges of that.
The rare disease is a systemic disease, it's a systemic immune syndrome, and it's true, it's enormously expensive. We also those pricing. The ocular disease is an IVT, and weekly injected we inject once a month, once every other month, and for 15 years. So you're really not exposed to this pricing challenge. However, you're absolutely right. So when you look at some of the rare disease development approaches, you develop a drug for rare disease, and then you want to switch it to some sort of a larger indication. And that is in general an enormous challenge, especially if you have already approved in the rare disease, and especially if you're already selling it and you make good money. Because the minute you lower the price to go into the new indication, you lose a big chunk of your revenue, and you're not entirely sure that the new indication will really be that. So that is a big challenge in our industry, and there are ways to go around it. But it's in general, it's much easier to go from a broader indication into a rare indication. You may not get the full revenue potential there, of course, but you convince them to do that. It's in my experience.
I didn't realize though you have two different compositions of matter, two different routes of administration.
We have two different routes of administration. We have two different products, but one API in both products.
I can comment on that as well, because we're doing the same thing with our new product, because the mechanism of action is also cheap, disease independent. So the idea right now is we're looking at dry AMD as well, because there's similar mechanisms of deterioration.
Anyone else in the audience? So one question I have, and this is really kind of thinking about this from the standpoint of, we have some young innovators in the audience. What advice would you give, to somebody sitting in the audience today that's, thinking they may have a compound or the aspiration to start a company around some of the research that they're doing? What advice would you give them that could, help them in their journey, either through the process of creating a startup and specifically in rare disease?
So in some ways, I had to go through that process, being an academic and kind of then being a part of the clinical trial team, and then being a part of the sponsor. You have to kind of shift your mindset. And I think the advice I would have really been already voiced is that you have to be patient. You have to know when to step out. Because sometimes a founder is so passionate about their product, I found that, and they don't always understand what it means to be in industry. And even writing a paper, when it's an industry paper, it looks a little different than an academic paper. And so that's probably the biggest thing. And, I've had a very positive experience currently with keeping founders engaged and things of that nature. And I think in some of my other experiences, I was in CMO and those experiences, I saw that it didn't quite work as well. And I think that's probably the company, but that was my biggest thing. You just have to be patient. It's a long, long process. You have to be willing to fail.
I would add to that, and from my experience, I was larger companies, and now again, Paris is coming off. And so what this does is it brings us a lot of startups, a lot of innovators come to us. And I can maybe share my impression I have from these pitches. Often we see a pitch which says, I can use this technology or this ability at a disease. And there is often good rationale that this indeed can work. But what is regularly missing in sort of the broad picture is just because it works doesn't mean there is a market for it, doesn't mean it's competitive in its positioning versus competition. So if you have an anti-inflammatory, you say, hey, it works, it's an anti-inflammatory, it's not evidence that who cares? It means that it's $2. So, this type of what I see from the academic world is you sort of have a technology and you try to find an application. what we do in a large company is we're in many ways technology agnostic. We have a problem. We see a market, we see an unmet need. We try to address this unmet need. And if it is a device or a drug or a injector or whatever it is, it's in many ways the question is how can you address the unmet need? So I encourage all of you to start with the unmet need. Go from the patient perspective and start there and say, what is this patient experience? Don't go from my drug could work. Start from how could I address this problem, the patient experience? And then go around and say, what are the technological solutions? And it may include where you are at the moment in your research. You need to come from this angle to convince everybody that this is the best approach to address this particular unmet need. And in my mind as a researcher, I would not worry too much about pricing, about all of this. I will leave that stuff out. In my experience, it's always, if there is an unmet medical need, if there are patients in need to get treatment, the money problem.
Models. How do you address safety concerns related to using a gene therapy for delivering something that is not directly the gene, so in a sort of a gene agnostic manner? And then also, once you get into the clinical trial, how are you going to measure that it's working?
So the idea of safety, I mean, there's two parts. There's proof of concept and then there's safety. And so safety a lot of times is done in wild type monkeys to make sure that the product itself does not have some type of inflammatory reaction that you're unaware of, that you may not see in a proof of concept model, whether that be in vitro in mice or some other animal model. So that's the way we usually do safety in kind of a farm tox manner. And then obviously POC is done with whatever animal model. And now with iPS cells and the second program when we were at Spark was choroideremia. And that was based on an in vitro proof of concept. So there are precedents out there to do that. When it comes to the clinical trial, what you're looking for us is the product is called rod-derived form viability factor. And my team has named our clinical trial Prodigy. We're naming after rock bands. I didn't even know Prodigy was a rock band. So, for that, we're using phase 1 is obviously the primary endpoint, safety and tolerability. Secondary endpoints, we're looking for some type of pharmacology. And therefore, what we're looking at is to see if we can slow down the progression of the deterioration of the cone cells itself. So you use basically the things that you would use in most trials. If you name the test, we're probably doing it. But we're highlighting basically structure in this case because the FDA has basically said that you can have structural decrease in progression, even if there's no clinical change, and that would be an approval product. We're also doing obvious functional changes because we want to see that there's an effect for these patients. So there's a whole battery of things that we'll be looking at, both structure and function.
Okay, excellent. I think for the sake of time, I think we're going to move on, but I want to thank our panelists here and really appreciate you guys coming and sharing your insights. I hope you guys will be around so that folks can ask their questions perhaps personally at some of the breaks. Okay.
All right, so now we're going to switch gears a little bit from ophthalmology to a rare disease program here at UCSF for disease known as Artemis-SCID. And my name is Sohaila, and I'm the program director of the Catalyst Program's diagnostic track. And in the last few months, I've had the privilege and opportunity to work with the clinical trial development team here at UCSF. And it is my privilege to introduce the dedicated scientists working on it, Dr. Jennifer Peck, who is joining us in person, and Dr. Cowan, who will be telling us more about the disease via Zoom. And I'm also delighted to welcome Dr. Cammie, who is the director for the Living Therapeutics Initiative here at UCSF. And together they will be telling us more about the gene therapy trial development, its advancement, and bringing this to life. Jennifer.
I would love to have that bringing to market kind of discussion, but we're not there yet. Not even close. But I'm very happy to be here today just to even think about it. So let's see. Diseases of the immune system, single G immune disorders, are individually very rare, but you can see there's been logarithmic growth in the number of these identified since 1980, of course, because of the advances in sequencing, but also advances in immunology and the ability to dissect immune system pathways. So now we have over 500 of these single gene disorders, and they're immune deficiency and also immune dysregulation, because the immune system has to know when to turn itself off, and when that doesn't work, we have big problems. So these are in great majority defects in the hematopoietic cell lineages. And so we have thought about approaching these through treatment based on the hematopoietic stem cell, which then differentiates into different lineages. So the most severe of these is severe combined immunodeficiency, or SCID, which got popularized by the boy in the bubble in Houston years ago. And I think medical students these days are too young to remember the bubble boy, but he actually, it was someone I took care of as a fellow when I was at Baylor. So this is a complete absence of adaptive immunity, or T cell and B cell immunity. Very few or no T lymphocytes at all, and absent antibody production, either because the B cells are also missing, or they don't work if they don't have T cell help. So infants born with this type of problem start to get sick very soon after birth, around 2 to 4 months, when maternal antibodies wane, and they develop more and more severe and recurrent infections. They actually collect infections like a toxic bouquet that they never get over because they can't overcome these pathogens. And this is eventually fatal unless we can establish an immune system, which has been done by a transplant from a healthy matched person, or allogeneic transplant. We now know that SCID is not one disease. There's at least 18 different ones, and more to be discovered. And another thing that we know is from the Primary Immune Deficiency Treatment Consortium, big mouthful, this is a group of 47 centers in the US and Canada that treat these diseases. That's funded by NIH, and UCSF is the lead institution. So we collected patient data to form a basis for treatments. And you can see that this is the distribution of age at hematopoietic cell transplant rate, or CT, and it's a biphasic curve. Why is that? Well, some had a family history, and everyone knew there might be a SCID baby coming, and so they immediately took precautions and treated the babies early. The other ones, the bigger group, were discovered because of infections they developed later in life. And guess what? Survival of the early treated group is much better than later. And this is shown here a little bit. You can see the very top, very excellent survival if treated under 3.5 months. That was that inflection of the biphasic curve. And on the that orange curve, even in modern times, 50% survival if you have to have a transplant when actively infected. So one of the ideas I had was, we've got to get this early. We've got to get every baby the same advantage that the ones with a family history have. And by the way, those are maybe 10%, maybe 20% of cases. So we can't count on that family history. We need to get a test that we can do on the dried blood spot that every baby gets in the nursery to test for PKU, sickle cell, and thyroid. So we've got one, and that is something called TREX, or T-cell receptor excision circles. These are the leftovers that are excised from our T and B cell receptor genes when they do their VDJ recombination to make specific receptors for antigen. And these leftover products form circles because they have cohesive ends, and you can do a PCR reaction across the circle, and that shows you whether T cells are being made properly. So this has turned into a newborn screening test. Now every baby in the US gets this test. So we're getting SCID patients early. That's great. And you can see in this PIDTC data, the green line is diagnosis by newborn screening over this period of years. And the diagnosis by infection in red has gone way down. So this has been a game changer for SCID and opens the door to new kinds of treatments, even though the individual genotypes are ultra rare. Now we want to be able to approach them and treat them with better means. So this is actually the PIDTC data showing the direct benefit of newborn screening, which was adopted in the decade between 2010, or 2 decades between 2010 and 2018. You can see that in the prior decades, survival didn't budge for SCID. It was stuck. And once we had this newborn screening, we had improvement. So, but what we've also learned, and I already said this, SCID is not one disease. And this PIDTC has collected enough cases to see that genotype matters. And some of the genes, like IL2RG, have very excellent survival, and this is with allogeneic transplants. But the worst one there is called DCLRE1C, or Artemis, and this is the disease that Morton, Cowan, and I have launched a gene therapy lentivirus program for. So compared to every other genotype, it has significantly worse survival. And I think Mort is on Zoom, and he's going to tell you why. So, Mort.
Okay, can you hear me?
Yes, we can. And do you want me to advance slides for you?
I think you're going to need to, yes. So I'll do
Great.
it. Yeah. So this slide really tells you everything you want to know about Artemis deficient SCID, or ART-SCID. So as Jennifer said, the gene involved is a DCLRE1C, which encodes for the Artemis protein, which turns out to be a very important component of DNA double-stranded break repair. And in fact, that's the main problem with these patients in terms of VDJ recombination that Jennifer mentioned, because without Artemis, then these patients don't develop T and B cell receptors. And so these patients don't develop T or B cells. And so they have this T minus B minus SCID phenotype. And in addition, for Artemis in particular, they have increased sensitivity to alkylating chemotherapy and ionizing radiation because of the abnormality in double-stranded break repair. So ART-SCID is clearly it's an ultra-rare disease. It represents only 2 to 3% of all SCID, although there are certain populations like the Navajo and Apache Native Americans, the Athabascan speakers, and also in the Middle East, it's an increased incidence. But in the US, North America, US and Canada, we only see about 3 patients per year. And we've estimated that there may be 30 to 40 patients per year worldwide. And as Jennifer said, it's the most difficult form of SCID to treat with an allogeneic transplant. Even with the gold standard, the HLA matched sibling donor, T cell immunity is often incompletely restored. And in less than 20% of patients do they get B cell immunity restored. So in most cases, alternative donors like unrelated donors or mismatched related donors have to be used. And in those cases, there's increased rejection, there's poor immune reconstitution, there's increased graft versus host disease, which I don't have time to go into, but it's a reaction when donor T cells attack the tissues from the recipient. And then overall, there's poor survival, as you can see, compared to other genotypes. When you can overcome some of these problems with high-dose alkylators, but because of the increased or the abnormality in DNA double-stranded break repair and increased sensitivity to alkylating agents, these patients will develop short stature. They have poor permanent tooth development, endocrinopathies, and increased mortality. So that's why we decided years ago to focus on autologous gene therapy. So this is the clinical trial. It's a phase 1/2 toxicity and feasibility study. This just gives you this is a picture of the self-inactivating lentiviral vector that we're using that carries the human Artemis cDNA and along with the human Artemis promoter. Patients are treated with a non-myeloablative, very low dose exposure targeted busulfan conditioning, in which the cumulative area under the curve is 20. And just to give you a frame of reference, the ablative dose of busulfan for a standard transplant would be 70 to 80. Then following that, we infuse the gene corrected bone marrow CD34 hematopoietic stem cells. So we're looking at safety, survival, overall survival, event-free survival, and T cell immune reconstitution. This just shows the process. The patient undergoes a bone marrow harvest. The bone marrow is taken to the lab, and the CD34 cells are isolated and then cultured over 3 days with the lentiviral vector. The end of 3 days, aliquots are taken for release criteria, and then the rest of the cells are cryopreserved over liquid nitrogen. Once the cells are released, then the patient undergoes 2 days of conditioning with the busulfan, and the cells are then thawed and infused. Next slide. So this is just a summary of the trial. We started the trial in May of 2018. We got the IND in late 2017, got funding for the trial to start in May of 2018. We've enrolled to date 14 patients, 12 of them diagnosed by newborn screening. And you can see that 5 were of the Athabascan speaking Navajo or Apache Native Americans. The median age at treatment was 2.8 months. The one outlier here at 13.3 months came from Canada, from a province that hadn't started newborn screening yet, and so was diagnosed by symptoms. The cell source is bone marrow in all patients, and we hit our target quite nicely of the busulfan, and we've seen no unexpected toxicity related to busulfan or the cell infusion. In terms of immune reconstitution, we have 11 of the 14 patients with at least 1 year of follow-up. The median follow-up for these patients is 48 months. 10 of the 11 have full T-cell immune reconstitution. The one patient that doesn't has adequate T-cell immunity, is living a normal life at home. In terms of B-cell immunity, all of the patients have developed B cells, although 2 have low B-cell numbers, and 7 of the 11 have a full B-cell reconstitution between 19 and 32 months. So just as in a preliminary analysis, we looked at a historical control database in which this came from the PIDTC, in which we identified 26 patients who had ART-SCID since 2000, which is when the recognition of the gene mutation was published. And 19 of them met enrollment criteria. That is, they would have been eligible for the clinical trial. And 10 of the 19, interestingly, were diagnosed either by newborn screening or family history. So it became a subgroup that we could compare to our to 10 gene therapy patients. So the overall survival for the gene therapy patients is 100% versus 63% for the historical controls. And you can see that that's statistically significant. T and B cell immunity was also superior in the gene therapy versus the allogeneic controls at 24 months. And that's also significant, statistically significant. And then looking at event-free survival, you can see that whether we looked at the entire group of 19 controls or just the 10 controls that were diagnosed by newborn screening and family history, the event-free survival was significantly better in the gene therapy cohort. Next. Then in summary, I think to date, we can say that infusion of gene-inserted autologous CD34 cells appears safe. For patients with ART-SCID, we've seen no rejection. We clearly need longer follow-up to determine any potential late effects from the busulfan exposure, but this low-dose exposure seems to be well tolerated, certainly acutely, and we've achieved multi-lineage engraftment and development and T and B cell immunity with that approach. And we've seen no problem with insertional mutagenesis, so no malignancies to date. I think we can say that outcomes are quite good compared to the historical allogeneic controls, certainly with respect to overall survival, event-free survival, and immune reconstitution. And there's a lot of people to acknowledge, but I think the main point I would make here is that without the funding from the California Institute of Regenerative Medicine, from CIRM, we wouldn't be talking to you today. They have funded all of the work that we did in both the preclinical, the IND, and now the clinical trial. Thanks.
Okay.
I wonder what we just shared. We could just share the card. We get more on. Can we get more on the screen?
Yes.
We don't see more. We have to pin more.
Danny, can you help me project?
Well, while they're figuring it out, let me just introduce myself. My name's Cammie Edwards, and I work with the Living Therapeutics Initiative here at UCSF. It's a 2-year-old initiative with the goal of actually advancing cellular therapies to patients. We actually are involved in not only the preclinical work, but also that valley of death that was spoken about, as well as the clinical trials. We certainly can't do it alone, so he's been a really terrific partner in that. But as part of those efforts, we are interested in those diseases for which there is no, we are interested in diseases for which there is commercial opportunity. We want to out-license those. But as a university, we also are in position to take care of those diseases for which there is no commercial partner. And that is the lead-in into how we got involved with, I guess we see us, oh, I hate seeing this. How we got involved with this ARTEMIS-SCID program of the rare, it's the ultra rare. Uh-oh, what did I do? Uh-oh. The rare, it's the ultra rare. And we believe with the ability for us to actually do gene therapy like I've been able to do before, with the possibility of CRISPR potentially allowing for simpler ways of having platforms that could be approved versus single, going by drug by drug by drug. I understand it's a long-term goal, but that's something we think about. We are interested in seeing how we can long-term develop a program here that we can actually support these cellular therapies for these patients for which there are solutions, but without us, they wouldn't get, that wouldn't be available to them. So this is, their story is so interesting because with 3 patients a year, they fit the ultra rare, not commercially viable. So I thought maybe you guys could start with where you are in the regulatory process and in the clinical trial process, just to give people a flavor of what you've been through and what's coming up next.
Yeah.
So,
oh, go ahead, Jennifer. Are you gonna?
No. Well, I would just say that nobody ever trains us to apply for an IND or do any of this stuff, and we were deer in the headlights completely. So I think I'm a pretty good immunologist, and I think I'm a good doctor. I'm not really cut out for this, but fortunately, we had help from colleagues who have done gene therapy, and they sort of helped give us their INDs, that things that had worked, and gave us a lot of hints. But I think another thing that has happened is there's been a lot of attention trying to make rare disease treatment developments accelerate. And the idea of warp speed from COVID vaccine being imported to rare disease new treatments is something that we hear lip service about. But I think, and I think Mort would probably agree, we've had a few meetings with the FDA. And at the last meeting, we did notice a difference. There was a thaw, and there was more flexibility, I think, on their part, recognizing this is not going to be something where you can have a randomized double-blind trial. No, that's just never going to happen. And then they wanted simultaneous controls, and of course, everybody wants gene therapy for this now because it works. So we can't do that either. And at this last meeting, they were actually willing to consider these historical controls, even though they weren't collected the way the FDA would want them to be for a standard kind of trial. So I think that we are seeing more flexibility. I don't know if you'd agree with that, Mort.
Yeah, I mean, I think the FDA is constrained by the, regulations and laws that have been established by Congress, mostly for, I think, standard drugs, treat hypertension and diabetes and so forth. And they're kind of right now forced to apply these same regulations to cellular and therapy and gene therapy. And I think it's pretty clear that, some of that has to change. I think they know that, but it's made it hard for them. But I do agree that there seems to be a somewhat of a thaw. I mean, we had our second type B meeting with the FDA in October of last year, 2023. And that was a very positive meeting. So basically they said that they did agree that we had done our due diligence in terms of control of controls, and that they recognized that it would be impossible to do a randomized control or even contemporary control study. So they have allowed us to use the historical control data set. I think they have told us to complete the efficacy study, which will involve probably enrolling maybe 5 to 8 or 10 more patients. And they also have told us, and what we have to do is to demonstrate in vitro and then in vivo comparability of a commercial vector. So that was another thing that that we learned that nobody taught us about. And that is, when we started the trial, we were using a very good vector from Indiana University Vector Production Facility. So we have great data and we're very happy with that vector, but that vector is only certified for phase 1-2 studies, and it can't be used for phase 3 or for commercial use. And to do that, we had to find a manufacturer of commercial vector. And of course, to do that, we have to not only, once we get that vector, we have to show in vitro comparability and then get FDA approval to treat a number of patients, 3 patients, to demonstrate in vivo comparability. This is all going to add more time to the study, but we think that we'll probably be able to have that completed by 2026, maybe late 2025 with a lot of luck, or 2026. And then we have to finalize the historical control data set. And the other thing that we have to do is validate a potency assay. So potency assays can be very difficult when you're dealing with some of these diseases, because you have to show that the protein that is made is in fact functioning. And it's hard to do that because Artemis, in our case, Artemis is not expressed at a very high level in hematopoietic stem cells, because it isn't needed to any great degree in hematopoietic stem cells. So it's hard to measure that activity. And we have fortunately developed an assay in the lab that we think will function as a potency assay, but it takes 7 weeks to get a result. And as I think we've made it clear, early diagnosis and treatment is critical for these patients, and we can't wait 7 weeks. So we have gotten the FDA to agree that we can use a surrogate assay and then still do the potency assay, but still treat the patient before that potency assay becomes available. So that's kind of the big picture, and in terms of where we think, where the regulatory issues are for us.
And I guess I would also add, in the big picture, we're still waiting for an Artemis baby whose parents happen to be billionaires or even multimillionaires. We have really cute kids from the Navajo reservation who live in hogans and who don't have running water. And we don't have a corporate angel. We wish we did, but we are limping along trying to do this without corporate backing. And so if anybody wants to step up, that'd be great. But meanwhile, we really appreciated UCSF stepping up and saying, look, there are other ways to do this. If this is not a good time for venture capital, let's see if we can do this in an academic setting. And we're hoping that we can make it work.
We have time for one more question. You can hear me. We have time for one more question. Does anybody have a question for them? Because I do have one thing that I thought might be kind of interesting, and that is the fact that we are an ultra rare indication. It's part of the reason why UCSF is getting so heavily involved in it. And the FDA is much more geared towards much larger markets, their rules. And I thought, Mort, you might share some of the challenges you've run into as we're trying to get it closer to be something that could be commercial, where we could actually treat patients. The figuring out reimbursement is something down the line for us, but I thought maybe some of your insights on that might be kind of useful to people.
Yeah, I mean, again, as Jennifer mentioned, our initial hope pre-pandemic, I would say everything now is pre and post-pandemic, but pre-pandemic, we probably interviewed 5 or 6 companies that were interested in taking on this project and developing it. And we chose one, and we're about to sign an agreement with them, or UCSF was, and then the pandemic hit and everything was put on hold, although we did continue treating patients. And then after the pandemic, the economy slipped, and there was the Silicon Valley Bank closure and failure, and all the interest from companies dried up. And since then, the interest is only, they say, yeah, we're interested in rare diseases as long as there's 200 patients a year. So 3 patients per year isn't going to make it. And so we've had to go to a plan where we're going to try to do it academically at UCSF. And of course, the problems there mean that our GMP facility is good for phase 1/2 trials. So we're going to have to upgrade the GMP facility, and that's going to be costly. That's going to cost money to do that. We talked already about the commercial vector, and I think we'll be able to do that with CIRM help, as well as UCSF help. I think those are sort of the biggest issues. The requirement for a financial investment. We have to develop a solid business plan. UCSF wants that too. And we have to develop a way of maintaining the GMP facility. And I think one of the probably most important things in sort of achieving these goals is to identify other uses, the use of like the GMP facility for other diseases, because I think this is the only way that this kind of approach can remain viable. And I think there is that possibility at UCSF because of all the research that's going on. And there may be interest as well from other places, because we're not the only ones that are having problems when we're dealing with these rare or ultra rare diseases in terms of finding a way of making them permanent. Because now that we know that this treatment works for these patients, we just have to find a way to keep it going long term. We can't stop. We can't go back to sort of the current or the standard therapy of transplantation when we know that this is so much more effective and safer.
Thank you, Mort, Jennifer, and Cammie, and thank you everyone for staying with us. I now welcome you all to join us for lunch, and we're hoping you can be back by 12:45. We will have a nice musical treat for you and continue with the afternoon session.
Truly amazing person that I got to meet got introduced to 2 years ago when he agreed to be at our remotely at our rare disease meeting, and his TED talk and his song. And he's a singer, songwriter, rapper, inspirational speaker, philanthropist. He has osteogenesis imperfecta, or brittle bone disease that causes a lot of fractures. He's had 160 plus fractures. And despite all of that, he's raised $2.25 million for charitable causes. And he's truly amazing. And y'all will all become his fans too when you hear him. So let me just play his recording.
So before anything else, thank you, Dr. Ramamurthy, for bringing me back here. And hopefully one day I can come and meet all of you beautiful people in person. Speaking of Dr. Ramamurthy, when I first participated here in 2022, I did a poetry workshop with her where I got to explore my experiences with osteogenesis imperfecta, or brittle bones, in a new way. Fast forward to December of 2023, and as you may have heard already, the compilation of poems that participants like me with rare diseases wrote rose to #1 on Amazon in Poetry of Specific Places, #2 on the Kindle store for poetry anthologies, and #3 in British poetry. So can we all give a huge round of applause to Dr. Ramamurthy for putting that compilation together, and to all the other participants in that collection for their imagination and amazing skill. So how would you like to hear one of my poems from the collection? Yeah? All right. This is a poem about perspective, worth, and freedom. Here is Doors and Windows. Whenever I talk about my condition to people, I used to tell them something like this. When in life one door starts closing, one more door will start to open. But on closed doors, when we focus, we can't see the ones that open. Then I tell them how this proverb applies to me. See, God may have closed the door on my ability to walk, but he gave me a mind to speak and then a voice to sing and talk. And by the end of the conversation, people would say things to me like, oh, what wretched men we are. We don't have so many woes, but look, life beat him black and blue, yet still he strolls, onward he goes. But then I learned about neuroplasticity, and here's how I've come to understand it. Our brains are wonders to behold. So adaptable are they that when some senses can't be used, the parts of our brains that convey messages to these dead senses, these parts take on other roles. Now you know why Stevie Wonder could find a mouse at a few years old. So I submit to you today, what if we were always wrong? Just because we look the weakest doesn't mean we aren't strong. Everyone has strengths and flaws, good at some things, others not. So why do we haste toward labels like disabled? Why these thoughts? What if disability means dissing our abilities and acting not on our potential because we're trapped in the circumstantial? We're not underprivileged, even though we're underserved. So let us put aside our bias. Treat us as we're deserved. So now, whenever I talk about my condition to people, I tell them something like this. Sometimes when we do compare ourselves to others, we conclude that life for us is never fair because what they can, we cannot do. So we think the doors are closed, but what if we thought differently? What if really these closed doors are windows of opportunity? Thank you, everyone. You guys are so kind. Now, spoken poetry is not the only way that I've used words to talk about my experiences. I mean, I'm speaking to you right now, and this is not poetry, but in fact, I think if you've ever heard me before, it's either an inspirational speech or you've probably heard one of my rap covers. In fact, I'm just curious, has anyone heard of my cover of Not Afraid or maybe Rap God by Eminem? Raise your hand if you have. Okay, that's cool. Well, how would you like to hear one of my own rap songs? Yeah? All right, cool. This is a song about overcoming identity and vision, which, funnily enough, I released last year amidst a painful series of eye surgeries that left my right eye with drastically less than ideal visibility. Here is This Is Me, the Rap Prince. Yeah. For everyone who wants to know the real artist, the person behind the Eminem covers, this is me. I was a broken boy, 40 fractures coming out of mommy's tummy. 48 hours to live, I was an unspoken dummy. Barely could breathe like a kid's seeds in his mother's milk. I was bound to die, ain't no lie. Parents cried, I found the thread of silk, but I survived that trial. The first of many cases. I revived a young child already going places. I got a gift from God, that was music so decide. The notes fell in my head, the notes I said, a wild journey those notes led. Started with piano, broke my arms once or twice. Couldn't play, had to switch lanes, brittle bones for my vice. So I started with North Indian classical music and on the western side. On my wheelchair, I was singing rhymes. First performance when I was 7 was my staircase up to heaven. Competitions, trophy cases, no ignorance, no scared faces. Wrote my first song when I was 10. Since then, I've been writing with no end. I am not meant to society, that's not what I was meant to be. I'm a bit brave with Not Afraid, and I put out a cover album. I was the six with every show I made, harder than the average could fathom. Globetrot, never stopping, ere my death arrives. It's too confident with the wings of a dove and the roar of a lion, but I'm still realizing. Wanna sing this with me? With the power of pen, the power of pen, the power of pen. So my rise from pain and my purpose lies. When my heart beats boom, bars leave from surprise. This book never ends with the I know it's raised with the ghost of my words. Yeah, I'm shining this light. Ripple like water, my fire gets hotter. The beat rock and running, breath taking me further. Cleaning up from start to the rocker. I'm out of a poetry, got a music is my water. The complexity of my trap and let out 670 back and swim. Cause I truly, fully am a tour de force to be reckoned with. Rap blacksmith, summertime to sip. Paradigm ships, med bitches, I make rips. I make grooves and twos, but move a tooth for me and you and keep it 110%. Twinkle eyes, cause about my soul, my work, my goal, my here to kill, just sweat the blood, so peace still, machine skills up, still, even on the field with seal, because this is only inception, hip hop turning succession. I'm here to help reclaim the lost kingdom, fitted and regression. So here arrives the next step to separate the weak from the tear. I'ma stay bold and I won't be scared. But even after I my mission will stay clear. I'm rushing in a new everything. Right across from 6 armaments, that's why I'm pure rhythm. Clean the wrist, I may not be the right God, but I am a right prince. Thank you. Mama, Papa, Mama, shine with love and we'll rise on top. Shine with love and we'll rise on top. like gods, but we shall die like men. And though I'm not a king, my voice will still ring, a prince with the power of pen. Thank you so much, everyone. Thank you. So would any of you like to hear one more? All right, lit. This last song is definitely easier to follow than the previous one because it's not a rap song. This is a slower ballad that I wrote after winning the Young Voice of New York City competition in 2015 and being crowned a youth ambassador for St. Jude Children's Research Hospital. It's a song of encouragement, dedication, and hope. Here is There's Always Tomorrow. Have you ever felt alone? Or maybe not at home? Did you want to give up? 'Cause maybe it was too much. Or you might not have wings, but you can still fly and you can sing. Because who you are doesn't matter, 'cause you're a superstar. There's always tomorrow, no need for the sorrow. We will make it clear if we can come back from where we began. If you can believe it, you can achieve it. Life's a mountain with steep and bumpy edges. Oh, and you have to push yourself all the way to the peak of the ledges. Well, you might not have wings, but you can still fly and you can sing. Because who you are doesn't matter, 'cause you're a superstar. There's always tomorrow, no need for the sorrow. We will make it if we can come back from where we began. If you can believe it, you can achieve it. So don't ever believe that what you have will go in vain. You can still follow your dreams, no matter how much the pain. There's always tomorrow, no need for the sorrow. We will make it if we can come back from where we began. If you can believe it, you can achieve it. Yeah. Thank you so much. My friends, art has always been a healing medium for me, so I just pray that my art moved and soothed and will continue to move and soothe you. And I hope it never ceases to remind you that if you can believe it, you can achieve it. Thank you again for having me.
Yes. Oh, I can try that. Actually, give me 1 second. Okay, so Dan Lowenstein wants the slide and present review. So since I'm sharing screen, can I just
So he wants what?
presenter view? So on my computer, he wants to be able to see the notes and everything.
Oh, I
And
see.
then have the slides up there. It's a little tricky. Right?
Can we?
Should we test it out?
Back on this. Is there any audio? Is there any audio?
Oh, maybe switch out.
Just keep that in there.
But no, first I have a video.
So first, we want to go to circumference. We want to make that extended.
I need to activate this. Okay.
Okay. Yes, please.
Mm-hmm.
I want to know that. Is it? Oh, it's a little loose, I think. You just ran it. It just worked. There it goes. Yeah. So who? Okay. Is she on the weapon right now?
No, it's a video recording.
Oh, it's being
So
recorded. Oh,
I know if you just have to hit play.
so that means there's audio.
This is a presentation.
Oh, is it?
and then
Okay,
I'll
no
put-
problem.
I'll handle it.
Perfect.
Mhm. And then Dan will just make-
Oh, no.
No, this one has audio. but We're doing it. through Zoom.
Oh, it was through Zoom right now?
Yeah.
Awesome.
Hold on. If you go on my screen. I can- As soon as you tap, I don't know if you. If you tap that- If not, I can do this. We're already found it.
Yes.
All right, we're going to get started with the afternoon session. So thank you everyone for gathering for this really important topic today, all day long, and I'm honored to be here. My name is Einor Sawyer. Clinically, I'm an orthopedist at UCSF. I started out in pediatric orthopedics, and I run the skeletal health service. So I'm going to take a moderator prerogative to tell you that if you didn't see the prior presentation, watch the recording of Sparsh Shah, who's my new teacher. He reminds me and reinforms me that people are not defined by their physical conditions, because that's certainly not what I'm going to remember from him, even though it's in my specialty. But he brought so much more to that just now. So please take a moment and watch that recording. So it's my great privilege, because of my role in innovation ventures, aside from my clinical role, I get to support frontline innovation. And in doing that, I get to meet fantastic people. And I get to introduce to you today a colleague of mine who's a friend and a long-term colleague, Dr. Kathryn North. Professor North is a pediatric neurologist and also a clinical geneticist. For 10 years, she's been the director of the world-famous Murdoch Children's Research Institute in Melbourne, Australia. And I've had the great good fortune of working and collaborating with them over the past 10 years in innovation and precision medicine. She's also was appointed in 2014 the vice chair of the Global Alliance for Genomics and Health, and co-chairs the clinical working group. So we get to see her often in the United States. When she's here, she always stops off at UCSF. She's a good friend and collaborator to our institute. And really excitingly, she's quite well known in her country, so we sometimes get to refer to her as Dame when we wish. She's received the Order of Australia in 2012, and higher than that, the Companion of the Order of Australia in 2019. And this was really for eminent service to genomic medicine internationally, as well as neurology and child health. So we're very excited to continue our work with her. We're starting new discussions with Kathryn and the MCRI in terms of working with UCSF on specific projects. If you have an interest in things she's saying and you feel like from your vantage point, you'd like to connect with her, please let me know. So I'd like to proceed now with this incredible presentation by Kathryn.
Good afternoon, everyone. Lovely to be with you from sunny Melbourne, Australia. I really wish I was in San Francisco, one of my favourite cities. My name is Kathryn North, and I'm director of the Murdoch Children's Research Institute. And today I really look forward to contributing to the Rare Disease Symposium by giving you an overview of how we're implementing genomic medicine within Australia, and to draw some parallels with what's happening internationally. To provide some background, I'm a pediatrician by background. I trained as a child neurologist and then as a clinical geneticist. And my clinical practice and my research is particularly focused on kids with neurogenetics disorders. So my research for many years was focused on gene discovery and how we increase diagnostic rates and how we ultimately turn that into better treatments for our kids. When I moved to Melbourne just 11 years ago, I changed my focus to then look at how we can take what we're doing in the research laboratory, funded usually by grants, and take this disruptive technology into healthcare to improve diagnostic rates and to make it accessible to all the children and the families that need it. That's what I'm going to talk about today. This audience will be very familiar with the impact that genomics can have across diagnosis and therapy and prevention across the life course. So already genomic medicine is transforming the delivery of healthcare, providing rapid diagnosis, particularly for kids and adults with rare disease, and in directing the diagnosis and therapies for cancer. It's involved in prevention, particularly with reproductive carrier screening and prenatal testing. There's increasing number of research programs looking at the application of genomic newborn screening. And then excitingly, the prediction to try and contribute to decreasing the frequency of chronic disorders in adult life, in particular applications with cancer and cardiovascular disease. So with these many uses for genomics, to be able to use it across all of these different areas will require and is requiring a revolution in approaches in our healthcare system. Where we've started in research, there's going to be increasingly a virtuous cycle of immediately translating research findings and discoveries of new genes into clinical practice and evidence-based healthcare. But along the way, we need to be growing and improving our ability to interpret and capture data, the ability to translate health technologies and health technology evaluation into policy and practice. How do we fund these testing, particularly as it goes to a population scale? And also at the same time, how do we create a genomic literate workforce? In Australia, in 2013, 2014, we held a series of roundtables to look at the current state around the country and also to look at what we really needed to do to take a cohesive national approach to the delivery of genomic medicine. Now, Australia is a federation. We have a number of different states with state governments where the hospital systems are funded. But at the national scale, there is a national healthcare system similar to the NHS. So we've got a number of different sources of funding we need to navigate across the healthcare system and to keep it all cohesive. So what arose out of these consultations was a targeted call for research called Preparing Australia for Genomic Medicine. And the simple goals of this were to demonstrate how patient benefit can be maximised in one or more diseases using genomic technologies, building evidence for cost effectiveness, the practical strategies for implementation, but at the same time, building upon our research translation capacity. And I have led since 2016, and still to this day, the national approach, Australian Genomics, that has developed this cohesive national approach. Our approach was to start with where we were in clinical practice, with the patients as they presented with rare disease or with cancer as a starting point, and then to build the infrastructure and the expertise to deal with the diagnostic pathway and the translation into clinical care. And this involved a number of cross-cutting programs, including the development of a national diagnostic and research network, developing and implementing a national approach to data federation and analysis, building the expertise and creating the resources for the evaluation of the cost effectiveness in health economic data, the translation into policy, the engagement across government, state and federal, and addressing the ethics and legal issues. And then at the same time, making sure that we're growing this genomic literate workforce to deliver this into healthcare. So this program worked has involved over 450 investigators in over 100 institutions around the country, including all the clinical and diagnostic genetic services in each state and territory, and they're listed here. Very importantly, we've taken advantage of pre-existing national infrastructure, but also very importantly have strong relationships internationally so that we continually can work with other countries around the world, very much with the USA, with Canada, and with Genomics England, so that we're adopting best practice in Australia as we're developing the resources. We've also, and I'll talk a little bit later about the Global Alliance for Genomics in Health, which has been a great international partnership. So to summarise our approach, we actually developed national coordinated networks across all of these different flagship disease areas in rare disease and in cancer. And we prospectively enrolled patients as they came into practice and then took them through the diagnostic process obviously getting quicker as we went along, but at the same time, as they came into practice, using the different technologies that were cost effective, initially with exome sequencing, moving into whole genome sequencing, and ultimately adding in proteomics and RNA-seq as we started to try and increase our diagnostic rates. Over this first series of pilot projects, over the first 5 years, we provided testing across over 5,000 families across 20 different disease areas. And we also were able to use that pilot data in each of these different flagships to take the case to government for national funding for these programs of work. So, for example, with looking at the diagnosis of childhood syndromes, we were able to demonstrate that using standard of care back in 2014, we were getting 11% diagnostic rates at a cost per diagnosis around $25,000. Whereas if we, in the same group of patients, provided a genomic test early in their diagnostic journey, we're increasing their diagnostic rates to 55% with a cost per diagnosis of $6,000. So 5 times the diagnostic rate, a quarter of the cost. And that in 2020 was funded by our government so that that testing is available to all family and children with suspected genetic disorders and funded through our national health system, Medicare. To give you an example of some of our other studies that we have supported, this is one of the first cabs off the rank as well, called Mackenzie's Mission, which was recognising the value of reproductive genetic carrier screening, which previously was only routinely available for 3 disorders. And what we aimed to do here was to see how we could make this available at a national level in rural and remote areas, and we're very proud of all the different areas that we accessed with this study. And the goal of the study was to test up to 10,000 couples, and we used a whole exome sequencing approach at this stage with an analysis of over 1,300 genes that were known to be associated with severe disease or disability, where the parents would be offered the choice to make a decision to do prenatal diagnosis. We also, as part of the study, provided access to IVF for those who wanted it. What we found out of this study is that over 1 in 50 couples were found to be at increased risk of having a kid with a severe childhood disorder with a genetic cause. And that 80% of those were disorders that were not part of the 3 that are routinely screened: cystic fibrosis, fragile or spinal muscular atrophy. So we're now currently working with government to design a publicly funded, expanded reproductive carrier screening across the country. At the other end of the spectrum is how quickly can we provide diagnosis for very critically ill children. And when we started this work, again, it was back in 2018, we were able at best to deliver genomic testing in around 2 weeks. But this study really focused to engage all paediatric intensive care and neonatal intensive cares around the country, and to decrease that diagnostic turnaround time to less than 5 days. And in this study, which has been published in JAMA in a recent paper in Nature Medicine, we demonstrated that we could provide that 3-day turnaround at a national scale, with 54% of the children tested receiving diagnosis, and that this changed care in 75% of cases. We're now working with government to make this available nationally, but a number of states, including our own state of Victoria, on the day that the research study finished, funded this within our intensive care system across our state. So it's cost effective to do this. It can make a dramatic difference in terms of early diagnosis for children, particularly when there is a therapeutic intervention. In all of these studies, we found that genomic testing was highly valued by participants and accepted by the broader community. And particularly when we looked at consent in cohorts of patients, and then went back a year later to see what their opinions were around the consent that they'd given. We found that 92% consented to secondary data sharing, and they maintained that level of consent a year later. And so that all the data sets through our research studies are now becoming part of a legacy data set to inform future research. So that was the first 5 years of the study. And at that time, there was a move by our government to create a national genomics agency embedded within government. But then, as we all know, the pandemic happened, and so that was slowed down. And so over the last 3 years, our government has funded our collaboration around Australian genomics to not be conducting these pilot projects ourselves, but to be maintaining that connectivity around the country, which is forming the basis of a future government agency, which I'll talk about at the end of this presentation. So our government has also funded 500 million Australian dollars over 10 years to support a range of research projects in genomic medicine. And so Australian genomics is underpinning that research, particularly providing the infrastructure that's needed in terms of translating research outcomes into practice and making sure that our research is really targeted on providing the data we need to inform government or government investment in making that testing available. So our focus over this last 3 years has particularly been on that engagement across different health services and the translation into policy, the support of the researchers and the research projects themselves, the increasing support of workforce, but also making sure that we had outreach into the community, both nationally and internationally. And just to give you a couple examples here of what we've been focusing on, is we've supported over 150 research programs that are government funded through our Genomics Health Futures mission across a number of different areas. Each are targeted on how they can be delivered into clinical practice if they're providing evidence that demonstrates benefit and cost effectiveness. We've also developed a number of genomic support capabilities that are available nationally and internationally. Our website is very actively used, but what we've really found is that as we provide all the tools and resources that are developed, these are getting many hits both nationally and internationally. And it's also promoting the cohesiveness of approaches around the country. We've also developed national infrastructure to support genomic integration into clinical practice with dynamic consent platforms that are also being adopted nationally. We've collaborated with Genomics England to implement Panel App, which has an online platform for gene and virtual panel curation. We've linked together all of the public pathology services around the country so that we can share evidence about clinically curated variants, but also importantly to link that into international databases like ClinVar and ClinGen, so that we're building up that reference point for gene and variant curation. And we've also worked with government to develop a prototype for the ongoing national approach to genomic information management and its interface with the health system and research. As I also mentioned, the public engagement is incredibly important, both the involvement of consumers, looking at the ethical, legal and social issues, particularly now at the moment we're doing a lot of work with government to make sure that insurance discrimination becomes part, that anti-discrimination becomes part of legislation. And we're really then starting to get into schools and high schools and medical schools to make sure that genomics is taught across the curriculum so that it's understood, particularly when it's going to be used increasingly in primary practice. We've also been progressing a number of different projects advancing health genomics implementation across many different areas that I won't go into, but all of this towards building a national cohesive approach And let me say, all of the tools and resources that we developed are freely available nationally and internationally. Internationally, I mentioned earlier the Global Alliance for Genomics in Health, and I've been Vice Chair of the Global Alliance for the last 10 or so years. And it's really been invaluable. Now a partnership of over 100 countries. We work together to develop the tools and resources that are necessary for the safe and responsible sharing of clinical data. And Heidi Rem from Boston is now chair of the Global Alliance, and Richard Scott, who's currently the acting CEO of Genomics England, and myself share what's called the National Initiatives Forum, where we're bringing together currently over 40 countries that are implementing genomics into their healthcare system, so that we can learn from each other, but also make sure that our data is interoperable internationally, so that the knowledge that's gained from one patient can be used for the benefit of all. For those of you who don't know, the Global Alliance is the website that you can go into and have a look. But it is really, I think, sort of creating the end. I see it would be great to have Google Genome. So if you've got a gene variant or a particular disease, you can just Google the variant and come up with an evidence-based data set which to interpret and provide rapid diagnosis for the patients that you're seeing. And to get there, we really need to make sure we're federating approaches and that we're sharing knowledge. So where are we going with all of this? As I mentioned, the whole goal of this Preparing Australia for Genomics Medicine was for us to establish and really entrench within our government and within our healthcare system a national approach to genomic medicine and the establishment of a national genomics healthcare initiative. So Genomics Australia is actually in the process of being formed as we speak, where the goal is to have the legislation for the creation of this new government agency finalised and in place by July 2025. And so we're currently just developing the details of that legislation based on really all this work that we've done over the last 8 years in trialling and testing what works within our Australian healthcare system. Overall, this government agency will be looking at what works best if we do it on a national scale. Australia is a small country with a population of 26 million people, but if we function as a single cohort of individuals and of patients, then we can make great contributions internationally. But as well, we can avoid duplication of effort, fragmentation in our approaches, and ensure equity of access of genomic healthcare delivery. So Genomics Australia will continue the work that I've outlined in Australian genomics as a critical connector across research and translation into practice, ensuring cohesive data management and genomic healthcare delivery. It's also a great point of contact for our international partnerships, for our engagement across consumers, and for the development and influence of evidence-based policy, and continually assessing and implementing what is international best practice. So the priorities of this genomics initiative, I won't go through in detail, but it does recognise the importance of continuously funding and supporting research and its effective evaluation and translation into practice. And I think that it's a really different model in genomics, is that we're constantly learning as we go along what we don't know. We're still in many rare diseases only hitting at best the 50 or 60% diagnostic hit rate. So there is so much work to do to get that extra 40% so that all patients will be able to get an accurate diagnosis. And then how do we work with the better understanding of disease mechanism and the delivery of appropriate therapies, and how do we deliver that into practice? Then there's all the elements of how we also work across in partnership with industry, with the development of different therapies. How do we do this with the preservation of public trust? And how do we ensure that what we're doing is most importantly of benefit to patients, but there is equitable access. And in a country like Australia, we have so many remote and rural areas where prevention is just as important as it is within the cities. So this is the goal of what we're doing. It's really the next building block related to the work that we've been doing over the last 10 years or so. So I'm obviously the mouthpiece for a wonderful cohort of investigators, clinicians, consumers and patients around Australia. And our managing director, Tiffany Walwood, herds the cats and makes this all happen. Again, I'd love to be there with you in San Francisco. I think I'm visiting later in the year. But just to let you know, we are hosting the Global Alliance for Genomics and Health meeting in mid-September in Melbourne, Australia. So if any of you are interested, please come on down. It's going to be a great program, but we're also making sure it's going to be a lot of fun. So have a great rest of your rare disease meeting. And I look forward to seeing you all when I'm next in San Francisco. Thank you.
I'm Rupa Ramurthy, Director for the Catalyst Program and Invent Fund. And it's my great privilege and pleasure to introduce Professor Dan Lowenstein, a distinguished professor of neurology at UCSF, an expert on epilepsy, clinician, scientist, who got his MD at Harvard, residency training at UCSF, and most recently was Executive Vice Chancellor and Provost at UCSF. He's an elected member of the National Academy of Medicine, and he's an avid hiker, climber, a canoer, and has done this all over the world. And my personal interaction when I'd had head jolts early in COVID, and my former vice chancellor introduced me to Professor Dan Lowenstein. And even though he was so busy with all the prep for COVID and planning, he still took my call in the evening, gave me advice when I had to go through the EEG. Luckily, the EEG came clear. It is his amazing achievements combined with his incredible compassion that has touched so many people. And it is my true honor to welcome him to give the keynote.
I did, but I'm also going to present more here.
That'll be fine. Great, thank you. Well, thank you for the introduction. That's one of those intros that you wish your mom could have heard, right? But a pleasure. Thanks for the opportunity to focus for the next short period of time on an area of health that is common. You've already heard that there's one person in the room who's already been impacted by the experience of having a seizure. And I'm sure that there are more of you in the room who directly understand what it means to have a seizure disorder or epilepsy, whether it's yourself personally or someone in your family or someone that you love. And so my assignment from Charles and the group was to talk about epilepsy, to give you an overview of this group of disorders, looking from the past, but also projecting to the future. I hope to have a little bit of fun thinking about what the future might hold. So let's start. A typical day in clinic. And this would be an 18-month-old girl from Redding, California. I emphasize far away to make the point that not all folks have access to institutions like UCSF or Stanford or the other places here in the Bay Area that can provide top-level care. And she recently had a unprovoked, meaning no clear reason why it happened, generalized tonic-clonic seizure or generalized convulsion. And she has a prior history of a prolonged febrile seizure, a normal MRI scan done locally in Reading, and a normal EEG. This is bread and butter pediatric neurology and epilepsy. And so in thinking about that common clinical case, it brings to mind what I call the 3 universal questions that we all ask if we're seeking the help of a healer. And those questions are, why did this happen to me? Can you help me? And what does my future hold? Yeah, I want you to think about that for a moment, right? Think about any time you or you brought someone to see a doctor or another healer. You have had to have asked at least 2, if not all of these questions. Otherwise, you wouldn't bother going, right? So let's think about that in terms of this, again, common clinical scenario for someone, a young child who's presented with a seizure. So the first question is, why did this happen to this individual? And we know quite well what the overall causes of epilepsy are. They look something like this. The most common used to be called idiopathic, but we now understand that that predominantly is, in fact, genetic. And I'll talk about that for a few minutes later. And then there are other causes that you probably are aware of: traumatic brain injury from car accidents and so forth, congenital causes, infections, stroke, and so forth. So we oftentimes know why an event occurred if it's provoked by the smaller part of the pie, but we still do not understand the majority of patients who present with new onset seizures and epilepsy, in which we presume that it's mostly genetic. And as you'll see, we're making great strides, but still, in most patients who come to see a doctor for a new seizure, we actually cannot define exactly why it happened to that individual yet. So the next question is, can you help me? And for sure, that's what we are going to try and do. And our goal, as you can see, is completely ablating the seizures and not having any side effects. However, despite the fact that a lot of new anti-seizure medications have been introduced, especially over the last couple of decades, the story is not so great. If you look back at a study that was done by Gowers in the latter part of the 19th century, when there was only one drug, bromide, about a third, 36% of the patients were not seizure-free. So the drug worked in 2/3, but not in a third. Fast forward to 1971, over 20 drugs had been introduced by that time. And yet studies, again, cohort studies and population studies showed that the numbers were similar. About 2/3 responded to the meds, 1/3 did not. Again, a number of years later, now over 30 drugs, the numbers are the same. And most recently, we have over 38 drugs. There's actually more now. There's probably 3 or 4. The numbers are still roughly the same. And these numbers also belie the fact that for the patients who seem to have their seizures brought under control, those 2/3, in my experience and shared with others, at least half of those individuals have side effects from the medication. So I think that our ability to fully control the seizures and the side effects of patients who have developed epilepsy is certainly less than a third of all patients that we see. Can you help me? It's also worth and very important to remind ourselves that not everyone has the access to true standard of care. And a whole host of studies have been done about disparities in access to care across all of medicine. Hundreds, now thousands of studies that have been done. And within neurology, for example, we know that a significant number of patients, people of color, for example, do not have access to neurology expertise. And for example, one aspect of treatment for epilepsy when it's refractory to medicine is access to surgical treatment, which can be effective. And we again know that there's a huge disparity in the ability for patients to have access to that higher level care. Now, what does the future hold? Well, the impact on the patient is more than just having the seizures. Certainly the worst thing that patients complain about in terms of having epilepsy, which is a tendency for recurrent seizures, is that it's not necessarily the seizure itself, It's the inability to predict when a seizure will occur. And that disrupts virtually everything in life if the seizures aren't brought under control. There's definite risk of physical injury and even death. There's a syndrome called sudden unexplained death in epilepsy that can occur in patients who don't have their seizures under control. There are comorbidities. Various psychiatric disorders seem to coexist with certain forms of epilepsy. It can destroy a person's ability to get around by driving if the seizures aren't brought under control, and of course, issues related to employment, schooling, and so forth. And then maybe most of all, carrying the diagnosis of epilepsy in most parts of the world, including many parts of the United States, is associated with a tremendous amount of stigma due to the fact that most people don't really understand what epilepsy or seizures are. So what does the future hold? Another issue that we face is the need for biomarkers, and we essentially don't have any biomarkers for epilepsy. We do have the EEG, which can let us know whether or not a person has a seizure or they have a propensity for a seizure, but it is definitely not 100, it's not even close to 100% predictive. And we don't have any circulating biomarker that would allow us to understand what the likelihood for another seizure might be, or what the current state of the person's epilepsy is, or the likelihood of them to respond to a particular anti-seizure medication. I can tell you, for those of you who are interested in this space, anyone who comes across a true biomarker for the kinds of things that I've just listed, it is going to be an absolute game changer in the ability for people to have their seizures brought under control or live with epilepsy. And it would just completely change the practice of medicine in this area. Now, that's the low point of my talk, okay? Because I do want to say that things have definitely been moving in the right direction. And if I go back 50 years, a number of very, very, very important translational advances have been made. I'm just going to list a couple of them. We do know what exactly happens in individual neurons during a seizure, like if we can measure what happens in a seizure focus, huge advance that happened many decades ago. We know one particular type of epilepsy called absence epilepsy. You may have heard it called petit mal seizures. The circuitry of that particular seizure type has been quite well characterized. We even know some of the specific calcium channels that are essentially involved in that. We know a lot about seizure-induced neuronal injury, both various types. Video EEG telemetry has been a breakthrough for clinical care, where we can actually bring patients into the hospital and monitor them with 24/7 EEG and video to really understand what the localization and the characteristics of the seizure itself might be. We have discovered both epilepsy and drug toxicity genes. I'll talk about that just in a moment. We know about autoimmune epilepsy, where there are certain syndromes now where a patient has a particular impact on their immune system, and the immune system is tricked into also targeting cells within the brain and then inducing the imbalance in excitation and inhibition that causes seizures. And then neuroimaging has definitely been a game changer, where we can now identify the cause of many either congenital or developmental aspects of a brain disorder that causes seizures. Let me just mention 3 areas that I would put right at the cutting edge that are incredibly exciting. One is genetic therapy. The second is stem cell therapy. And the third is neuromodulation. These are all very active areas of investigation and actual clinical practice now. Just as one example, here's the evolution of genetics in epilepsy. Very, very promising. I'll tell you, Kathryn actually mentioned the study where they were doing exome sequencing on kids who were really sick in the pediatric or neonatal ICU, and that they had a 50%, over 50% detection rate of significant causes of disease. The same things happened in pediatric epilepsy. So in infants and children who have severe epilepsies, 10 to 15 years ago, if we did any kind of sequencing, we might get a yield of roughly 5%. Now it's well over 50%, probably 70% for when the clinicians involved are able to identify those most likely to have, who have the phenotypic features. That's an incredible change. in the practice of epilepsy care. And as you can see, it's just continuing to rise. With those targets identified through genomic efforts, we now have therapies, and they're, I'm sure it's a theme that you've already heard about, very exciting, that using, for example, antisense oligos as a means of disrupting the presentation of mutated proteins. In fact, there is an active set of studies going on right now. Stoke Therapeutics is the lead company for this, in which ASOs have been designed to modulate the expression of a mutated sodium channel, SCN1A. And the translational studies shown in the upper right here suggest that the antisense oligo is in fact working as expected in terms of down-regulating the non-productive transcript. And animal studies, again using models of epilepsy, have shown that the introduction of the ASO decreases the likelihood of seizures in those models. That's now been actively used in humans, and Stoke Therapeutics hasn't yet reported their results, but what we hear is very, very promising. There's the second area is interneuron transplantation as a disease-modifying therapy in epilepsy. I'm not going to say basically more than a couple words about that, because you're going to hear about this incredible work. Oh, you're not going to work, not stem cells? Oh, what are you going to talk about? Oh, fish. Okay, sorry about that. All right, well, Scott is also the person who has led the field in terms of using stem cells as a means of correcting the imbalance between inhibition and excitation. What's that? Oh, it was ganglionic. Yeah, progenitor cells. Sorry, I got it wrong. You'll have to correct me. Anyway, introducing progenitor cells, which migrate and become inhibitory cells to offset the imbalance that occurs in models of epilepsy. And this has also been used not only in a model of temporal lobe epilepsy, but post-traumatic epilepsy as well. And then there's neuromodulation, in which there are a number of different devices have been introduced over the last decade and more, stimulating the vagus nerve, stimulating the thalamus, and most recently a responsive neurostimulus RNS system, which places electrodes on the surface of the brain in the region where a seizure is occurring, detects the seizure, and then in a closed loop system actually stimulates that region to stop the seizures. And that's also been a fantastic advance for patients who don't respond to meds. Okay, so let me finish by talking about what it might be like in the future going forward. And why not? Why don't we pick 2050? What's that, 26 years from now? 2050, 26 years from now. What will the care of the patient be like? Well, I should first say that we all recognize that it's a fool's errand to try to predict the future, right? And I have to admit that, I like thinking about the future, like I'm sure everybody else does. And my track record is not that great. So for example, I'm sort of thinking about some of the other people who have said things much more famous than I. This is Tom Watson, who was the CEO of IBM, who said, this is back in 1943. I think maybe the world will need 5 computers. It's a lot. This next one's my favorite though, and that's 1962. Decca Records rejected the Beatles because they claim that guitar groups are on the way out. So the Beatles had no future. They're off on that. So I also have a pretty dismal track record. So 2001, I was sure the Segway was going to change the world. I mean, any urban population, people would just be using Segways everywhere. That you can occasionally see them in San Francisco airport and some meetings, but otherwise I was wrong on that one. And then maybe even more embarrassing, Google Glass. Oh, I was, again, sure that we'd be all wearing them. That was way off. But I can claim, at least personally, being right on something. I'm sure many of you were right about this as well. And that is 2006, the iPhone. When the iPhone came out, I was quite sure that this was going to be revolutionary because you could quickly understand, building on BlackBerry and all the other things that were coming out, that this was going to offer instantaneous, ubiquitous access to information on the internet. It's turned out to be the case. I think about this. Can you imagine if an alien anthropologist came and visited Earth like in 2000 and then came back in 2020 and then reported back on a comparative study on the behavior of Homo sapiens? I'm sure that the study would say it's actually remarkable. Over the last 2 decades, Homo sapiens has evolved this rectangular, typically black object on the end of their upper extremity, usually on the right compared to the left. And they seem to always be either looking at it or holding it to their ear, right? So it's an example of, in fact, the way things can change amazingly. And with that, let's talk about epilepsy, because all of us know this. We are in the midst of an incredible revolution in our society on Earth, and that's AI. And in thinking about the way AI is going to change medicine, I find it incredibly exciting, and I'm quite sure that it's going to be happening. It already is, so I have no difficulty predicting that this is going to have a huge impact. In fact, I think that the iPhone likely has had as much of an impact on the behavior and the thinking of human beings as the introduction of the Gutenberg printing press, many centuries ago, in terms of the way we learn, the way we think. And the AI revolution clearly is representing the beginning of something as greater than the Industrial Revolution of a couple of centuries ago. So how is the AI revolution going to change the practice of epilepsy care in 2050? So here's the storyline. This revolution is going to allow us to have comprehensive AI-powered initial medical analysis when the patient is first presenting. There'll be new pre-visit diagnostic tests. There'll be a virtual reality epileptologist consultation with real-time AI documentation analysis. And then there'll be personalized data-driven diagnosis and treatment recommendations. So let's go back to the patient that we talked about who came into clinic recently, the 18-month-old. But now she's 29 years old, and she's referred by her primary care doctor because she had a recent unprovoked generalized tonic-clonic seizure. That first seizure when she was 18 months and the febrile seizures she never had anything else, never had to be put on treatment. But now she presents with a new seizure. And as you can see, she had this distant history, and that evaluation back in 2023-24 showed no abnormalities other than 2 what we call variants of unknown significance. So she had an exome done, but there was nothing that was identifiable, although there were some questionable changes at that time, and no other medical problems. So let's just go through what these steps might look like in 2050. So first of all, comprehensive AI-powered analysis. Well, all the information that's been collected on this individual from back in 2023-24 is available because of a universal medical record. All the lab studies, her genome has been done by now. Her microbiome is regularly done for all individuals. And all of this is undergoing an analysis that's giving us some initial ideas of what might the underlying cause be. She'll get pre-visit diagnostic tests. And by that, I mean a state-of-the-art, high-resolution imaging of the type we can't even imagine at the moment. But again, because of the presentation and the initial AI analysis, this is clearly indicated. And if the patient's willing to consent to these tests, they can all be done pre-visit. She'll also have the opportunity to be implanted with a subscalp self-powered nano micro electrode that is socially acceptable. Again, for those of you who are interested in things that are cutting edge and will completely change the nature of the care of patients with epilepsy, anyone who comes up with this is going to have an absolute breakthrough. Because at the moment, we do not have a good ability to follow what the actual electrical activity is of the subclinical seizure activity that our patients suffer from. And that creates huge problems in our ability to make clear diagnoses, but also know what our therapy is doing. So this has got to happen, and by 2050 it will. In addition, the patient's going to have a home-based neuro visit. No need for a neurologist, no need for a specialist, a nurse for this. It's all going to be done through AI. And the patient's going to be instructed to do the typical things that neurologists do, and that's all going to be well documented. Oh, and by the way, pre-visit diagnostic tests, they actually are already approved. Don't have to have pre-approval, because in 2035, the United States finally came up with a universal single-payer system. So everything's covered. I know that it's pre-authorized. I know that sounds like the most preposterous thing maybe that I'm talking about, but we got a dream. Okay, so then with all that information collected, the time comes for a virtual reality a consultation with the epilepsy expert. And in this case, of course, it's from a distance. The patient has been supplied something that's actually now common in 2050, which is image projecting contact lenses. The neurologist is able to project on a screen. They're both avatars, of course, is able to project the lab findings, the imaging findings, and is actually able to look at the patient and have a conversation. And there's a real connection between two people. And this image from back in the 2020s has become a vestige of history, where the physician or the nurse or whoever is constantly looking at the computer away from the patient, just trying to get that information in. And that's because the AI assistant is working in the background, recording the entire virtual visit. And after the first half of the visit, the patient and the epilepsy specialist turn to the AI assistant and ask for an interim assessment, which is then downloaded, reviewed, altered as seen necessary, and they can proceed with the discussion about likely diagnosis. Yet the AI assistant was using personalized data-driven diagnosis and treatment recommendations, including a scan of millions and millions of medical records and genomes and proteomes and microbiomes to see if there are any matches for this particular individual who's now presenting as a 29-year-old. And in fact, found about 5 or 6 individuals who have basically the same syndrome. And one of those VUSs, the variants of unknown significance for 2023, is added to 3 or 4 other mutations in the genome, which collectively are the cause of the epilepsy in this patient. And now that we have an actual diagnosis, we can think about the treatment plan. And the treatment plan will include perhaps an ASO. God knows what will be here in 20 plus years, but an ASO. It takes some time to make the ASO, maybe a week or 2, but we do have also immediately available therapeutics. This happens to be a drug with a chip, so we can actually monitor the ability for the drug to be absorbed. And we can get this to the patient very, very quickly because we'll just simply call it your own. So with that, following the visit, being put on the therapy, the patient will be able to have continuous monitoring on her own with the AI assistant, adding in the epileptologist and other members of the healthcare system as needed. So there you go. That's a vision of the future in 2050. My point is that the AI revolution that we're all part of is already having an incredible impact on the practice of medicine and the research that we do. The ability to integrate all of this information across millions and millions and millions of people in really complex data sets is a game changer. Virtual reality visits are going to finally do something to help remove the disparity gap that exists. Every individual who develops a seizure disorder or epilepsy should have access to the expertise that comes only from being someone or a group of people who understand epilepsy because of experience. Every member of society should have that. And it's not just epilepsy, of course. And then finally, the new therapeutics on the horizon are incredibly exciting. So with that, I'll say thank you. As you can probably tell, this presentation was a combination of my own limited experience, but very much ChatGPT 4.0 and Dolly. Okay, so I think now we make a transition. Great, so let's, thanks for your attention, and we're gonna call up Scott and Navneet, and we're gonna have a panel. With that? Sorry, I got it wrong. They didn't tell me. Yeah, I'm sure. Okay, so I think we're. So you're gonna. I think we're bringing down the chair.
Yes.
Just plug it down.
That might help.
It shouldn't be a problem.
Yeah, sorry. Yeah. Okay, so we're going to have two presentations and then a panel discussion. And I think this is good. I'll introduce both speakers now. First is Scott Barabin, who I've already mentioned, who's a professor and the William Bowes Endowed Chair in Neuroscience Research here at UCSF. And Scott's, as you can tell, pioneering work in our field has been focusing on translational discoveries and includes work on pediatric epilepsy in zebrafish. And the one thing I mentioned was the interneuron-based cell therapies for acquired epilepsy. He developed the first zebrafish models for epilepsy more than 2 decades ago, and he was the first person to use high-throughput drug screening using this zebrafish for a particular syndrome in epilepsy called Dravet. And they've led to clinical trials for this patient population, and Scott has started a number of companies around this. And then Navneet Mataru is the co-founder and chief scientific officer of Regal Therapeutics, and she's been pioneering the use of non-editing CRISPR-based platforms, such as CRISPR activation as a therapeutic modality for treating dosage-sensitive diseases. She's currently a visiting professor at UCSF, an investigator at the Innovative Genomic Institute at Berkeley, and recently received the Women Entrepreneurship Award from IGI-HC Child Foundation for Regal. So with that, Scott, come on up. Thank you.
Is there anything I need to do to? It's on my screen. I brought my laptop. It's on the screen. Sorry. I want to thank the organizers for inviting me and the Catalyst program, which has supported this work over the years. And of course, it's always a pleasure to follow Dan's talk because he sets up beautifully what I'm going to talk about, at least some of it, I guess. So I want to talk to you not about interneuron transplants, though if you are interested in that project, Google Cronut. He's an epileptic sea lion that we cured using interneuron cell transplants a couple of years ago. It's a fascinating story, so I recommend anybody interested in that kind of work to just Google Cronut and find him. But today I want to talk to you about zebrafish. So a little bit about me, as Dan mentioned, I've been a professor here at UCSF for almost 25 years and joined adjunct professor at Berkeley for about 10 years now. In addition to awards and other kind of details, I published over 125 manuscripts all on my career, all on epilepsy research, almost all of it translational. This has been the result of over $35 million in NIH funding to the lab, and everything I talked to you about has led to founding companies, but it was all done here at UCSF in my NIH funded laboratory. And intellectual property from the laboratory over these years has launched 3 different startup companies, Donio Labs, Neurona Therapeutics, which is pursuing stem cell therapies, and Epigenics Therapeutics, which is what I'm going to talk to you about. So as Dan noted, one of the biggest problems in the epilepsy field has been that many patients remain resistant to anti-epileptic drugs. And as noted, this hasn't really changed much since the 1860s. And despite addition of many new drugs, it's been a problem almost my entire career and longer than that. And so one of the things I did when I first started my laboratory was take a look at this approach to how we discover drugs for epilepsy. And what was really clear is that almost every drug on this list was identified in the same manner, specifically taking a normal, healthy rodent and inducing a seizure, which is an acute event, and then seeing if the drug changed the threshold for that seizure. Of course, this predates genetic information and genetic models that have spontaneous seizures, but most importantly, these are not drugs designed or that are identified because they're stopping epilepsy. Epilepsy, as Dan noted, is an unprovoked event in a not healthy brain that occurs spontaneously. And so when I started my laboratory research in this direction, I wanted to come up with animal models that had spontaneous epilepsy. We do this in mice. It's very tedious, and you can do video EEG monitoring. And that's how we developed the interneuron-based therapies. But for rare genetic epilepsies, we turn to zebrafish. And the reason we use zebrafish, I'll tell you in a moment. But the main reason is that we're capable of modeling genetic epilepsies and having a model that's a vertebrate model has spontaneous seizures. And that way we could look for drugs that stop the spontaneous seizures. Namely, we could look for drugs that stop epilepsy. And we think a lot of the reason that this 30% hasn't changed is that all the ways we've looked for these drugs or identify these drugs has never changed either. And so we propose this alternative approach to discovering new medicines to solve this 30%. And see, the approach is outlined here. Basically, as Dan noted, there are now, I can't keep track, over 100 genetic mutations, single gene mutations, largely de novo, that cause intractable forms of epilepsy, often occurring early in childhood. And these are almost always pharmacoresistant. That is, they don't respond to more than 2 or 3 available anti-epileptic drugs. And so our approach was to take this genetic information, which comes from actual patients, and then develop zebrafish models for those rare genetic epilepsies one by one. To do this, of course, we had to develop zebrafish assays to study seizures in these larvae. Larvae are about the size of your eyelash, to give you a little perspective. They're very small, they're only a couple of days old, but they are freely swimming, they have a highly developed nervous system, and they have complex behaviors that you can monitor by just videotaping them in a single well. But more importantly, you can do EEG recordings. So we established this about 20 years ago by showing you can do local field potential recordings from zebrafish larvae that are immobilized in agar. So our approach was to model these genetic epilepsies, do high throughput screening, and what we focused on was what we call phenotype-based screening. So again, not looking for drug modifiers, but looking for things that stop epileptic seizures. And we did this in 2 parts because the assays first for behavior are high throughput. You can do this on 96-well plates, but in any behavioral assay for epilepsy, there's potential false positives of identifying that behavior as a seizure behavior. Without electrical recordings, you don't really know what that behavior is. And even in patients, like Dan mentioned, EEG monitoring is critical in the diagnosis of patients with epilepsy. So drugs that come through the first pass stage are basically drugs that stop them from having these spontaneous seizures. But the second pass is to do electrophysiological recordings to look for drugs that normalize EEG back to normal. And that way we're looking for the most effective hits in these preclinical models. And then these move on to patients, as you'll see. So why do I use zebrafish? Zebrafish, first of all, are vertebrates. So they're genetically similar. They're actually 70 to 84% sequence identity with humans. For disease-based genes, it's in the 80 to 84% range. They have a complex nervous system, even very early in development, with both excitatory and inhibitory neurons and glial cells, all the components you need in the nervous system to generate abnormal activity, like you see in a seizure. They're inexpensive to maintain. We basically keep them in fish tanks. We have hundreds of fish tanks with adult zebrafish larvae, and you just feed them some flakes and some Artemia a couple times a day, and they're good. They're genetically tractable, and this is actually only improved in recent times with CRISPR technologies, allowing us to easily manipulate the genome and make stable zebrafish lines. More importantly, adult zebrafish, which we're talking about, are what you see in the pet store. They're about an inch or two long. If you breed adult zebrafish, you get about 1 to 200 larvae. And it's a larvae that we do the experiments on. So you can imagine even at a slow pace of doing this just once a week, you could do 200 experiments a week. Not possible, of course, with rodent models. And most importantly, larvae, which don't have scales, permeable to whatever you put in the bathing medium. So if you have a water-soluble drug, you can quickly test whether it's effective in a zebrafish model. Recently, with the help of Alicia Griffin, who's here in the audience, who's now in situ, we undertook to take on that genetic plot that Dan showed around 2010 or 12, when there were about 80 genes. And we generated, using CRISPR, genes for loss of function genetic mutations. We made over 40 zebrafish models. And actually, we can do more of this. We can actually make a fish for any gene mutation that comes up in the literature. And then we screened all those fish for epileptic phenotypes using our field recordings. So we screened almost 4,000 fish, and 8 of these lines actually have spontaneous seizures and therefore can be useful for drug screening. The others we're still studying. They might have some more subtle features. We made all of these publicly available. This is an NIH-funded project. You just send me an email, and then we send you the fish. So this is a large-scale project. Took us about, I want to say, 6 or 7 years. Shows the power of zebrafish to model any rare genetic epilepsy. But I want to focus on one quick story, and this is the story that led to the founding of Epigenetics. So our first project was to study SCN1, which is a sodium channel mutation seen in patients with Dravet syndrome, over 80% of patients. This is one of the most severe genetic epilepsies and one of the most common genetic epilepsies. There's over 1,200 de novo mutations in the sodium channel. And so we made a genetic model. This lacks this SCN1 gene, and then we started to study it. And for first, it has epileptic phenotypes, just looking at the behavior. This is a slowdown video of a fish having a spontaneous convulsive seizure. You can track the activity of that fish in a single well, and that's what allows us to do drug screening, tracking 96 wells of these fish simultaneously. But you could also see, more importantly, on the right side, that while behavior looks abnormal or convulsive, the EEG activity is what's really convincing. So this, a normal EEG in a fish would just be basically a flat line. But this fish, like patients with epilepsies, has high frequency burst activity followed by a burst suppression pattern. And this happens spontaneously in this fish. We don't put any drugs in the water. And it'll do this about once per minute. So there's very robust seizure phenotype. And it is a CNS-driven event. This on the bottom are 3D imaging using a light sheet microscope through the zebrafish brain, showing all neurons, and they're encoded with a calcium active, a GCAP, a genetically coded calcium indicator that tells you when the cell neuron is active. And as we'd expect from the field recordings, the seizure events correspond to generalized activation of the entire nervous system of the zebrafish larvae. And so this is what a seizure looks like in zebrafish. We've seen this not just in this model, but in other models of epilepsy as well. And interestingly, which is another side feature of using zebrafish for drug screening, it's not just seizures we could study. Working with Manisha Patel's lab, we've studied metabolic deficits in these larvae. We've shown behavioral assays on sleep disturbances and early fatality, all comorbidities that match patients with Dravet syndrome. Most importantly, when we give drugs that are known to be used in this patient population to these zebrafish, those drugs reduce the seizure activity, namely things like fenfluramine and stiripentol. And drugs that are not supposed to work don't reduce activity. So as a model for doing drug discovery, it's 100% predictive of drugs that are actually used in the clinic. This cannot be said for the mouse models, which so far to date in literature have only identified about 10% of drugs actually used by these patients and consistently failed to find things like fenfluramine. So what do we do with this fish? Well, we embarked on a large-scale effort over, I think now a 8-year period to screen over 3,500 commercially available drugs. We did this in a completely blinded manner. I just purchased commercially available libraries with no bias as to what the drugs we're looking for would be. We coded them and we put them through behavioral assays on these 96-well plates, looking for drugs that, again, reduce that spontaneous swim activity. They were tested multiple times. If they came out positive, we unblinded ourselves, purchased the drug from Sigma that the library indicated, and repeated these testings at different concentrations. If they went through all of those successfully, they moved on to electrophysiology. And again, looking for drugs that completely suppressed those electrographic seizure events. We screened about 3,500 drugs during this time on a minimum of 6 fish per drug, but usually a lot more. We found a lot of false positives of using the behavior, and this is important to point out that if you just look at behavior, you're going to find false positives on epilepsy readouts, specifically things like lidocaine or things that are sedative, or neuromuscular agents that like pancuronium that block activity. Those are going to stop the fish from having those behaviors, but do not affect the electrical activity in the brain. And so weeding out those false positives, we actually identified 3 new compounds, all repurposed drugs at the time, all serotonin activating drugs, lorcaserin, clemizole, and trazodone. With lorcaserin being, which is Belviq, a weight loss drug that was already in the market, we directly collaborated with Kelly Nupp, University of Colorado, and gave that drug to 5 patients. So it went right from fish to patients. All 5 of those patients responded with a reduction in seizure activity. We also did some pharmacology that I won't get into to develop novel chemical entities around clemizole and have 3 additional hits from that library. All of this work was done all of this preclinical work was done at UCSF. As Dan mentioned in the introduction, we first started using zebrafish back in 2005. We generated these unique zebrafish models, genetic models, more recently in 2021. To date, we've screened over 3,500 drugs, and with the OTM office here at UCSF, we developed strong IP around these findings. All the drugs identified in the Dravet model were then licensed, and I co-founded a company in 2016 called Epigenics Therapeutics to commercialize these findings and move these drugs into clinical trials. Epigenics has received over $35 million so far in private seed funding. We're a privately held company, and all 6 of the drug candidates have received orphan drug designation. All of them have already gone through pre-screening CMC data to show safety. And one of them actually, EPX-100, which is clemizole, has completed phase 1 studies and is currently in a phase 2 study. We have about 30 patients so far already enrolled toward getting to 100 patients. I can tell you almost every patient that completed this double-blind study entered the open label extension phase, and almost every patient in the open OLE phase is doing well on the drug with no side effects. We also plan to test this drug in LGS, Lennox-Gastaut syndrome, and eventually other genetic forms of epilepsy. So I think that's going to end there, so we can get to the next speaker. But thanks for giving me the few minutes to cover about 20 years of work in the laboratory.
Yeah. So I'll try to be short and sweet here. I think a lot of the introductions around epilepsies have been given beautifully by Dan, and thanks for talking about the sodium channel SCID already on this front. And as Dan mentioned, I'm interested in actually studying the dosage sensitive disorders, more genetic disorders that are related with either loss of function or gain of function. And this is the story we started almost a decade ago, and which actually led to a foundation of Regal Therapeutics, which I co-founded along with other faculty members, Nadav from Department of Bioengineering, and currently I'm a CSO and co-founder of Regal Therapeutics. So, sorry.
I can get it to do slideshow. Oh, because the slides are.
There is a PowerPoint presentation. You want to hide this panel? Or scroll? Okay. That's maybe okay. So everybody has heard about the editors, CRISPR being the new generation ones, which are actually zinc fingers and TALs be the earlier generation. What we are interested in the modulators.
using the same modalities, but deactivating their nuclease cutting domain, but retain the ability to target the favorite genome in the favorite gene in the genome. And what we are working on is actually using these deactivated CRISPR systems fused with some sort of a transcriptional activators or repressors to modulate gene expression. And I must tell you that there are actually more than hundreds of disorders, thousands of disorders that are caused due to alteration of gene expression or, dosage sensitivity disorders. And this modality that we are working on could be targeted, we call it gene expression modulation systems, and it can be targeted to gene regulatory regions in the genome. And by the virtue of actually affecting the gene expression by targeting any gene regulatory region, it can actually upregulate gene expression or downregulate gene expression, whatever is the pathological need of that therapeutic entity, the molecule. So we decided to actually follow haploinsufficient disorders and use the activation module here. Haploinsufficiency has a very strong burden in the genome. This is the exome sequencing database where I'm showing you almost like more than 60% of the genome actually, disease-causing genome has a huge haploinsufficiency burden. And there are around like 500 reported genes that are caused that are loss of function in those actually cause haploinsufficiencies. And I must tell you, these are one of the most neglected classes of disorders for therapeutic modalities. And around like 40% of those 500 are actually CNS neurodevelopmental disorders. And out of those, 100 are already in Simon research, spotlight genes associated with some neurodevelopmental disorders. And 1/3 of those are actually known to have a seizure burden. So haploinsufficiency, loss of function actually has been very clearly correlated with the seizure burden and neurodevelopmental disorder. So we used CRISPR activation module first to actually gain an understanding of this platform, and we used actually a neuroendocrine haploinsufficiency obesity model. We actually targeted MC4R with CRISPR activation, and you can see that the loss of function of MC4R gets rescued when you target with CRISPR activation, and these mice actually gain less body weight with their control counterparts. We also targeted SIM1, another neuroendocrine haploinsufficiency gene, and these mice were into remission. They never gained body weight even after 1 year post-injection, showing the durability and the long-term effect of this therapy. At the same time, the transgene expression was actually constant, and the SIM1 expression, the downstream target gene expression, was also maintained even after 1 year post-injection. We next actually focus our attention to a different sodium channel. It's a SCN2A, loss of function of it, clearly associated with autism spectrum disorders and neurodevelopmental delays. And with help of Kevin Bender's lab here in Neurosandler building, we actually generated CRISPR activation model in SCN2A mice and show that upregulation of SCN2A in these mice can rescue the cellular deficits. I'm showing you here the action potential velocity of these neurons compared to blue ones. If you see these purple one neurons, they actually are firing in the normal potential range as the wild type animals upon CRISPR activation. And some of the synaptic deficits recorded as AMPA and MDA get also rescued in these mice. What we are actually currently interested in how the clinical presentation of SCN2A loss of function could be answered by CRISPR activation. Most of the SCN2A loss of function kiddos have verbal delays, motor delays, and a subset of loss of function SCN2A kiddos actually have epilepsy burden, intractable seizures that are actually not under control by most of the anti-epileptic drugs. So we focus on modeling this epilepsy phenotype in mice, and we actually challenge these mice with 4AP, the proconvulsant molecule. And you can see that with the purple ones here, when we challenged with 4AP, the seizure susceptibility of these treated mice actually went down when compared to the untreated mice. And that was also shown in the number of ictal spikes that these mice, we can see in their EG reporting. So one of the other programs that we are interested is Dravet, where we can actually show that there is a temperature, hyperthermia protection seizure phenotype upon loss of function when we actually correct with CRISPR activation. So I'm going to compile my presentation on this node. Regal Therapeutics is focused on 6 active programs, cardiovascular and heart programs that are under Sarepta collaboration, and 3 of the main CNS disorders that are actually have a considerable seizure burden. I'll give acknowledgement to start with first patient caregivers and my UCSF mentors and colleagues and many of these people who have actually made it a reality, and other funding sources. Thank you.
So you guys have mics. Yeah, you guys have mics. So here we go. You're the guest of honor. So, so questions from the audience. I'll start. So Scott, let me the Clemenceau story. That's now been, what is it, a dozen years? Was it 2012? Yeah, the first 13. Yeah, so I know this is a common theme when you make an important translational discovery and the time it takes to get to the. Can you just talk briefly, and I need the same observations for you, about why it takes so long?
Yeah, great question. Actually, Alicia and I were talking about this earlier, discovery. There's 2 parts to it. One, on the preclinical side, obviously, we're publishing in the early years before it took about 3 or 4 years and lots of meetings to get into before we found the various iterations of how to start. That was about 3 or 4 years. From there, it actually went pretty quickly because we found the company in 2016. We had a new formulation, so it turned out not to be it. It was a FDA-used drug in 1950 that predates a lot of regulation. No one was manufactured. So we have to have the chemistry to make it work. The preclinical is great. The repurposed drug went cut that down. That was intentionally started. From there, it's pretty quick. I'd say about 2020 or so. When we started a trial. COVID slowed down. A lot of the progress in terms of bringing people up to that show. The trial is going slowly. It's picking up now. Should be hopefully. So while it's 10 years from publication to in clinical, I think that actually the real best we take that COVID and we took out UCSF Rare. And that was the company. So those things cut. So they're looking from discovery to clinical trial.
And I think you mentioned that Locarsin was a lot. I mean, it was much easier to implement because it's available and.
Right. So Kathryn, we tried to get a multiple of weight loss medicine.
Yeah.
In discussion with Kelly, neurologist who decided Kathryn was a safer bet to try compassionate use. It takes us over the years to get patients. As I'm a member of the Resurrecting Foundation for all time, patients who are there, we pick up the story. They actually funded some really personal things. But that year is that when it started, so we didn't get the patients in 2017. Kathryn, there was on A size, and there's also some real complications. He says that patented epilepsy. They've also done some clinical trials. In post-hospital. So that's what pursuing not pretty quick, but it turns out that this is another company involved. It's actually, it turned out that more complications.
And how much of an uptake has there been of Carson, given that it's been published and how much uptake has there been just clinically? Because physicians can pretty much, we can prescribe anything we want, even if it's not FDA approved,
Yeah,
right? So.
Anecdotally, for a couple of years after they published, I was hearing about use of it, but of course, patients don't cure. Patients are actually jumping on that. Also during that time, before we got approved.
Fenfluramine, yep.
There's a lot of moving parts happening. But I think the biggest thing on patients or other physicians are using it, is just the word that much information. And a lot of them are still doing it.
Yeah. It makes me wonder, and I'm going to be cautious in saying this, but I think that the early stages of development in these new therapeutics may go through the same problems that we've seen with anti-seizure drugs, which is, they show effect translation in a preclinical model, and then they show enough effectiveness, but they're not cures. And then they go to market, and then we see the kind of results that we've seen, historically. I fully am enthusiastic about alternative approaches, but I bet you the early years are somewhat like that. I'm need my question. I've got a couple, but yeah, good. Please, back there.
My son has a syndrome, and so he's got intellectual disability and epilepsy as well. And so our foundation is looking at kind of different research approaches. And you mentioned that Stokes has, what's it called? Stokes, the company that's doing the ASO approach, and Regal is doing the CRISPR-A approach. Can you say anything about like the likelihood of getting through FDA approval for the CRISPR-A approach? I'm not very familiar.
And is this on Zoom? Do we need to repeat the question first? It is on Zoom? Okay, so the question has to do with the haploinsufficiency approaches that have been presented. What's the likelihood that, or what's it going to take to get FDA approval for example, the CRISPR approach?
I think the CRISPR approach that I presented in there was a clear guideline for gene therapy products to FDA, through which we have to actually move forward with the clinical assets, actually clinical development program. And those FDA guidelines will also guide us to actually move forward with product development as well. And I can actually tell you very briefly that there was only first therapy CRISPR-based editing therapy that was used a couple of weeks ago in Kathryn by 4 years. So I think CRISPR-based therapies are definitely rising, and FDA guidelines will be actually, making product development
Is there a concern? Some people have concern of the CRISPR protein being a bacterial protein and expressed in the human, cause an immune response, and that's been shown in animals. How do you plan to get around that?
Can you repeat the question?
Yeah, so thanks, Bruce, for asking this question. Bruce, you're asking about CRISPR being the bacterial protein, and introduction into patients might actually lead to immune responses, and how we are thinking about navigating this. There is no doubt about it that CRISPR is a bacterial protein, and there could be actually immune reactions along, that could, we would actually end up seeing. But I must tell you that some of the safety and toxicity issues, the field has been collectively learning about it. The recent trial at Precision Biotherapeutics, where we delivered CRISPR molecule systemically in patients, those patients actually have immunogenicity well under control since almost 6 months. So I think there are actually mitigation ways to come around with the addressing of this immunogenicity. There are actually new technologies that are being made to make Cas protein less immunogenic as well, which I think will pave the way for these new genetic therapies.
I had a question about the SCN2A haplos insufficiencies. And just thinking about the gene itself, there's also a series of many reported gain of function alleles. So how do you think about that in the context of like in the future moving to clinic and patient stratification of the groups that are candidates?
Please.
So you're asking about, because SCN2A has been involved, some of the mutation involving gain of function and loss of function, and gain of function presented very different sort of clinical presentation than the loss of function. So these sort of precision medicine actually rely on the fact that these patients should be actually genetically stratified going after. So it should be very clearly loss of function mutation that we are actually taking. And again, the role of diagnosis, and what Kathryn was actually talking recently about, that the patient data about mutation stratification will definitely guide us. And we are, gain of function are different, disease, SCN will also function differently. And I do think that diagnosis act has to gear up to help actually design clinical trials in a very strong manner to go after patient stratification.
Yes.
I was curious about the gene expression modulation versus like the gene modifier. Like, is there a reason that you chose that approach?
Should I repeat the
Please
question?
do.
So you're asking why we chose gene modulation rather than gene modifier. If I actually ask you gene modifier, you mean the gene editing or?
Yeah, like changing whatever the mutation is.
Exactly. So I think a very valid question, and thanks for asking that. Gene editing approaches come with some burden of off-targets effect also, and we use non-editing forms of CRISPR to actually address that. There is no other off-target, there is no changing of the genome or editing the genome that we're doing. The second advantage here is when you're talking about gene editing, you're actually going after that one particular mutation. And if you look at the patient stratification, there are actually a number of loss of function mutations that are seen in patients. So our approach is to actually cover the whole footprint of loss of function mutation with a single modality rather than going and designing out for patient-specific mutation.
Maybe a follow-up related to that question about how to approach, in the epilepsy, the forms that you've been discussing, the genetic forms are mostly a broad network dysfunction, right? So we're thinking about cortical excitatory cells across large areas of the brain. Can you share some thoughts about how AAV approaches, like how do we know it's going to get to the appropriate targets, a very distributed target? And then related to that, Scott, maybe you can talk a little bit about your progenitor cells in terms of your thinking about how you target a broad network.
Thanks, Dan, for asking this question. There is actually a greater need now to actually make these AAV-based gene therapies as specific as possible, with less off-target, not just the overall, but also cell type specific off-targets as well, we're addressing. So the use of certain type of cell type specific drivers in the modality can actually guide the expression of these molecules either in individual neurons or excitatory neurons. And this will actually help us to off-target or mitigate the off-target effect of changing the circuitry by, like for Dravet, we have to actually go after the GABA neuron. And going after excitatory neuron might actually change that balance. And there are actually a bunch of studies that have shown. So these AAV approaches need to be actually more cell type specific. And I think the way we are actually thinking about it is by use of cell type specific drivers or by use of certain modalities like microRNA to
target cells that they can.
And what does the future hold in terms of the actual mechanics of getting the AAV in? Do you see standard like intraventricular injections into humans, or some new approaches that cross the blood-brain barrier but diffusely? Or what do you?
Yeah, I think Dan, you're asking very critical, futuristic questions already, because the kind of AAV capsids that we have were made like 20 years ago, for this. And there is a need for better biodistribution in CNS for AAV gene therapy. There are different, need to come up with a different, delivery routes, intraventricular or cisterna magna, how you're going to deliver intraventricular or not. And I know there are actually a bunch of companies chasing blood-brain barrier crossing capsids as well. And I think that's also pretty early. Right now, there is not like, strong data with, safety and toxicity yet with those capsids. Like if you actually overload systemically these capsids and they go into like, brain, what are the effects they have in the peripheral?
Yeah.
So, and I think a lot of safety data has to be generated in the next 1 decade to actually make the CNS gene therapy, much more stronger reality.
Great. Scott, anything to say about progenitors?
Yes, so Dan's referring to briefly about the slide in his talk. So what I talked to you about here in the short talk was genetic models of epilepsy, which are one side of the spectrum. As Dan mentioned, the other side are acquired forms of epilepsy, where it's a wide variety of reasons and circuit is disrupted. And so what my laboratory has also been engaged in for about 2 decades is what was just mentioned, which are inhibitory cells. So about 20% of our cells make GABA, which is an inhibitory transmitter. And they're a very diverse class of called interneurons. And we've been able to use the embryonic source of the subclass of these inhibitory neurons called medial ganglionic eminence, which is just the brain region where they come from when you're born. And so you can transplant them and get very specific integrated inhibitory cells that dampen excitation. And this goes back to a very ancient view in the epilepsy field that you have in these acquired epilepsies. The reason you're not seizing all the time is there's some form of surround inhibition. So the brain is basically clamping down on that epileptic kind of focus. Otherwise, with any of these epilepsies, you'd be seizing all day. It's not like your ion channel defect or your brain injury is different than Tuesday versus Wednesday. So that's the endogenous mechanisms. And one of the things that we know from anatomy studies for a long period of time is that these inhibitory cells are lost. So long story short, we're able to make these inhibitory cells using this fetal tissue and transplant them in animals that have epilepsy, including mice and Carla, who's an epileptic sea lion here in California because they ingested bolic acid and developed epilepsy. And the reason this works so effectively, it completely suppresses their seizures for the rest of their life. It's a one-time injection because the cells can migrate from where you put them, and they can make new synapses onto the existing excitatory cells. And therefore, they dampen the level of excitability. So they don't cure the epilepsy; they just stop the seizures from spreading and occurring. And so this approach is actually very useful for acquired forms of epilepsy. It's kind of agnostic to how the epilepsy occurs. So we've shown it works in genetic models of epilepsy, acquired models of epilepsy, etc. And pushing this to patients, of course, is a complicated translational kind of step. And not to rudely interrupt Dan earlier, but I'm not a fan of actual stem cells, because stem cells don't make this specific type of neuron that we use, at least not yet. And so to get to humans, or and even to get to product, we use cells that we took from fetal pigs. And pig cells, of course, we use for a lot of different trans-species, xenotransplantations. And we think that's a mechanism that you can develop these pig cell transplants that would work for really any type of epilepsy that's already existing in a patient, because these cells would simply integrate and functionally act like the cells that are gone. And so that's, in Dan's kind of year frame, I don't know what year that falls out. I can tell you, we started that project also around 2005. And I think we got to Corona, which is a large animal, in 2020. So 15 years to develop the preclinical work. And how many years that will taking that to the clinic, of course, depends Again, the same factors I said about zebrafish, depends on how a company is formed to approach that versus an academic lab. Academic research translate very slowly or costly. For example, if I wanted to do that here at UCSF, to be safe, I do monkey studies for safety. I was quoted, I think, $150,000 per monkey just to test that the cells are safe for 6 months. And so it's easily a couple $1 million just to get a safety study done. Again, that's not something an academic lab really can do with the type of funding NIH provides. And so those are some of the bigger discussions and barriers I found being here as an academic and working with the OTM office and Catalyst office for many years of the promise of preclinical work, not just in my lab, but lots of very talented labs around UCSF, getting that to actual patients. There's a very important business step that you need a partner in there somewhere. To be honest, I just don't think it's possible from a lab to people in terms of selling the product. For principle, no problem. But actually getting it where someone in the science is benefiting or family member takes another part, takes a business part.
I agree actually with you, Scott, on this. I think it's a rare disease symposium. I would like to share a bit of my experience with that, chasing rare epilepsy, rare diseases, and as you were talking about numbers like 150, and you need actually that sort of a funding and investment to be to make it actually from bench to bedside kind of approach, right? And I think according to the experience that I actually went through, it's been a little bit of a challenge to raise funds for rare diseases for sure. Although there is Orphan Disease Act, to promote industries and promote NORD is there, every bit of efforts are actually geared towards that direction. But at the end of the day, I think the capital is what will actually can help accelerate these therapies for rare disease disorders, which I find actually quite challenging because investors would like to actually put money in something that actually has a bigger population. And if you are talking about rare diseases, these are actually kiddos with high unmet need, with strong morbidities, and we need actually a social awareness around this.
And if I can jump in, this Bradford, Allie, Jan, one of the things I agree, I found very much, with business meetings, someone goes, well, how many kids have, I don't know, STXBP1 disorder? And we say, well, I don't know, 20,000 maybe, who knows, it's low. And all of a sudden it's like, well, we're not interested. And if we project it out, the second question becomes, well, how much are we gonna charge somebody for a gene therapy? You're gonna charge this family $5 million. And it's just to get the money back. I mean, this is how you guys know how the business world works, but it's like, what's the return on that investment? I think these things are, from a scientific point of view, I think they're clear. I think all these things are possible to help patients in terms of the science. I just don't know if they're all possible in reality.
Yeah, actually, that was one of my questions. So it just happened that yesterday I get this email from a master's student at the University of Chicago. And the email basically said, Dr. Lowenstein, a group of us are taking a class and we're doing a study on the possibility of a SYNGAP, a genetic modification therapy. And the goal of, it's a business class. And so we're studying how one would go about actually turning this laboratory finding into something that actually gets to patients. I wrote back and said, I really don't know much about any of that stuff. But she insisted. So I actually was on the phone with her today at 1:30. She was came across as very smart, asked some really good questions. But one of the key issues that she had not fully recognized, and I advised her to make sure it's part of her thesis paper or whatever the group is writing, has to do with just this. So what are your thoughts about and maybe this is a collective question, like how do we navigate the very brutal fact that it takes a lot of money to develop any therapeutic, and now we're talking about rare diseases. How do we approach the issue of the actual costs that are being asked? We're talking about hundreds of thousands of dollars a year, sometimes at a minimum to $1 million or more. What do we do about that? Yeah.
I get feedback.
Yeah. Has anyone thought about this? Yeah, please.
Well,
Oh, yeah, maybe we should use it like.
I've worked with companies facing this exact issue, and there's several things that need to be put in place. You really need to get buy-in from key opinion leaders, payers, other stakeholders, including the patients involved, and getting the right partner is a challenge, but they are out there, and if you work with companies and other individuals to find the partners that have other assets in a pipeline that are complementary, then you can really navigate that quite well. You also have to look at their competitors and what they're doing. So it is possible. And as far as the payer landscape, that varies, whether you're in the United States or in other countries. We have seen it with Luxturna. I mean, it was $1 million, but something that I didn't pay for. So, and with the Inflation Reduction Act and some of the new policy changes that are going in to examining this, both Medicare and Medicaid, and the changes in negotiations for drug prices. I think we're going to see some shifts in this landscape. So I would say everyone that's working on this, take heart. It is possible, as our one of the previous speakers said, if there is an unmet need, there is a way.
One of the things, like, I don't thought of the priority review voucher and how to use that to sell, to get it as an alternative. And the other quick question is, even if it can be afforded in the US, in most of the developing world, it is out of question. And how do we develop those kind of models?
Somebody over here. Yeah, that is a problem. And yeah, I was recently in Kenya, and there are 3 drugs that are available for seizures: valproate, phenytoin.
A couple of years ago, I think there was an academic paper from the University of Chicago and Columbia. The paper was titled "Financing the War on Cancer," and it was an analysis showing that for every year of life extension that a new therapy, would confer, the benefit to life insurance companies would total kind of on the order, just in the United States, about $6.8 billion. I mean, if you keep somebody alive for a year. I think one of the problems in this country is that there's kind of 2 rooms to the insurance house. There's health insurance and there's life insurance. And health insurance, the problem is that there's short-term policies, and the name of the game is like not paying. The payers hate to pay. Life insurance, however, and this might be true for governments, there is a financial incentive to keep people alive. So the thing about curing rare genetic diseases is that if you can take somebody who and transform them from being somebody who maybe has a shortened life or they die in childhood, or maybe they're not productive, or contracts, or maybe can't pay a life insurance policy, the cure actually in the context of the life of the patient, even if it's $1 million, may actually be a bargain to life insurers, or conversely governments that actually have a vested interest. So I think that's just one thought, is that I wish people would talk more to life insurers rather than talking to health insurers, because there's a real big conflict when we're talking about health insurance. And I hope smart people are thinking about that.
And one last comment in the back there, please. We're going to close it.
My name is Dan Lappin with Genesis Designs. I just want to give a shout out to the University of Chicago, where I'm a graduate of in economics. And I'm here because of the complicated medical condition I had, and looking at the infrastructure of economic concepts. And so I think one of the challenges that this segment faces is that it's focusing on a drug therapy ROI model, where the potential is that if you look at this rare disease segment, that there's other aspects that are rising in terms of our concepts of innovation, concepts what is the nature of genetics itself, and the kind of some qualitative aspects that can be translated into a arts, humanities, science, public health initiative that speaks to macroeconomic concepts of productivity and prevention, much in line with what the previous question asker asked about. What is the nature of risk reduction? Okay, and I speak as this as I've had conversations with the Federal Reserve Bank on these concepts of what is human capital, what is the nature of anatomy, physiology. So the potential, and so part of what I do is I'm on NASA's astrobiology science communication guild. So I work in translating origin of life principles in cosmochemistry and protocells into public health. And one of the things that NASA is famous for is going to the extreme and learning from that extreme and translating to benefit to health on Earth. And so I think there's a parallel that's happening here in the rare disease segment, that we're squeezing ourselves at the extreme. What are we learning from this that can translate and lead to the general population? Okay, so that's a different potential economic ROI from a macroeconomic perspective, different than this rare disease, this drug. And so that's why I'm here, looking forward to talking with folks on this, but that's another way of looking at how to translate that.
Yeah, thank you. That's actually, I've never thought of that specifically, but it makes complete sense to me. And I'm sure everybody else, I really love that analogy. Of course, we now have to weave in what the advances have been made since early on in space program of federal government versus private companies and the time it takes to get things done. But I think that's a fantastic analogy and a very good way to bring this session to a close. So I want to thank Navneet and Scott for excellent presentations, and thanks for your interest.
It's my pleasure to introduce Shah, who's going to introduce the next speaker. Do that.
While she's doing that, I work at the Baker Computational Health Sciences Institute here as Chief Technology Officer, and also with the NIH this year. Daphne deals with what I consider to be one of the most complete problems in AI and biomedicine, which is drug discovery. It is my honor to introduce her. I could spend most of my time telling you about her awards. I've tried to go fast, but I don't want to miss them altogether. She's CEO and founder of Incitro, an ML-driven drug discovery company, transforming the way drugs are discovered and delivered to patients. She was a co-founder and CEO of Coursera, which has reached 100 million learners worldwide. And she also was formerly Chief Computing Officer of Calico. These are all companies familiar to us. She is the co-founder of Engagely, which is an interactive digital learning platform, and was the Rajiv Motwani Professor of Computer Science at Stanford, where she served on faculty for 18 years, and is now an adjunct faculty member. Now, I could have gone on that much by memory. The rest is harder because it speaks of her awards, and I really don't feel like shortchanging them. She's authored over 300 referee publications in big name referee journals and has an age index of over 150. So please listen. She was recognized as one of Time Magazine's 100 most influential people in 2012, Newsweek's 10 most important people in 2010, has been honored with the Sloan Foundation Faculty Fellowship at 96, the ONR Young Investing, dredging up memories. Skip to the end, okay. Others include the Picasso Award, the MacArthur Foundation Fellowship, the ACM Prize in Computing. These are all things that I know about, they're worth calling out. And an ACM Newell Award and an Anita Borg Award. She was inducted into NAS, NAE, AAAI, and AAAS, and the International Society for Computation Biology. And her teaching was recognized by Stanford's Medal for Excellence and a Bayes University Fellowship. I consider her to be one of the greats in computer science and artificial intelligence. So please make it right what I said and welcome her.
Thank you. Well, I'm glad that's over. Thank you very much. So it's a pleasure to be here and tell you about the work that we're doing at In Citro. Some, but not all of which relates to rare diseases. I put a little bit more of an emphasis on the rare disease part in this presentation, but I also wanted to present a little bit of the broader arc of what we're doing. So you're going to hear about some pieces that are not specifically rare diseases, because I do think maybe following up on that last proposal that was made in the previous session, that those two are not unrelated to each other, and there are interplays between rare and common diseases that are worth us thinking about. Okay, so first of all, everyone I think is aware of the incredible time that we're living in terms of the emergence of AI as this force of nature almost. But if you think about why suddenly AI emerged, it's really actually an exponential curve that we've been living on for the past multiple decades. That it's just that when you're on an exponential curve, you often don't notice you're on one until it really picks up. And the exponential curve that we're living on is based on the convergence of four forces. First of all, are the incredible advances in machine learning algorithms that have driven all those large language models and the other things that we're all familiar with. This is the most visible manifestation. But under the surface, this is really a consequence of an unbelievable amount of data on which these models are trained, and an equally unbelievable amount of computational resources that are used to train those models. And then, of course, the growing base of machine learning talent that is able to put those together to generate the models that we're talking about. And so really, those are what we need in order to achieve success in AI, and specifically to achieve success in AI as applied to biomedical data. So it all really starts with the data. The premise of our work at In Citro has been that we really need to first make this happen. And so for the last 5 years or so, we have been building a platform that makes and interrogates very large amounts of data. So first of all, we've built a large and growing corpus of human clinical data that is juxtaposed with an infrastructure to generate, basically print, massive amounts of cellular data at scale. We use machine learning to identify subtle patterns in these data that are often far beyond what a human eye can recognize. And then finally, we leverage the power of human genetics to give us actionable insights on therapeutic interventions that hopefully will causally modulate the course of the disease. So as I mentioned, we collect data in both humans and cells. We believe that that combination is very powerful. Cells gives us an intervenable system. The connection to human gives us the bridge across the gap of translatability. We believe that human biology is very complex, and you can't really get a grasp on what's going on by considering only a single data modality. And so we interrogate human biology both in cells and in humans across multiple complementary modalities, and we put those together to create a picture of what's going on in human biological state. But importantly, no matter how much data we currently are able to acquire or print, it's not going to be enough for the complexity of human biology. And so we leverage the power of effectively generative AI to create more data from the data that we have. So this is a process called imputation, in which data that are in which measurements that are collected are used to impute or infer measurements that are not made. So all of a sudden you have a cohort that is considerably larger and more powerful than the one with which you started. And I'll show some examples later in the talk. So we leverage our platforms across our 3 core therapeutic areas: oncology, neuroscience, and metabolism. I decided to skip the work in oncology so that I can spend more time on neuroscience, which I know is very much a focus of the symposium. So I'll talk mostly about neuroscience and a little bit about metabolism. So our work in neuroscience is driven primarily via our cellular machine learning platform. And here we use imaging as a foundational modality for understanding biological state. The thing we like about imaging is that it is scalably generated and quite cost effectively. It is a surprisingly high content data modality. I'll show you some examples in a few moments. And it is far beyond what a human can extract from those data in terms of the insights that are available within it. We create these imaging modalities, both in fixed and live cell imaging. We're able to actually impute, and I won't show this today, live cell on top of the static, on top of a bright field image, we're able to impute stains without actually having to do the stains using the power of machine learning. And then we're also overlaying additional forms of omic modalities that can also be, as we'll show, imputed from the imaging data. So the disease with which we started our interrogation is a disease that I'm sure many in this audience are familiar with, tuberous sclerosis complex. It's caused by a sort of monogenic loss of function in either TSC1 or TSC2 complex. It is a rare but not ultra rare disease with 50,000 patients in the US and over 1 million worldwide. It presents as tumors, mostly but not entirely in the CNS, giving rise to refractory epilepsy and often neurological impairment. And the standard course of treatment is everolimus, which is a rapamycin analog, which is an mTOR pathway inhibitor. TSC1 and TSC2 activate, the loss function activates the mTOR pathway, and but it's only successful about 50% of patients. So as an overview of what we've done in TSC, well, we have used our cell-based models, specifically engineered isogenic iPSC-derived cortical neurons that are then phenotyped multimodally using both imaging and transcriptomics. We use machine learning to derive a TSC disease axis that is much finer grained than the standard anchor phenotypes that are used to understand, to interrogate the disease. And then we credential those machine learned disease axes, as I'll show you, to regain conviction that this actually corresponds to a translatable readout. We then use our screening capabilities that allow us to do pooled optical CRISPR screening to identify revertants in those disease axes, providing us with a much broader set of reverting hits than had been previously identified. And then we credential those hits using both human genetics as well as a much more onerous but translatable electrophysiology assay that measures epilepsy in iPSC-derived neurons. And then finally, we've taken at this point 2 of those targets and advanced them towards drug discovery, and are actually quite excited about the progress that we're making towards some of those early drug discovery efforts. So maybe just to talk a little bit about the disease axis and what it means to credential a disease axis. We've created a disease axis using multiple different modalities, coming back to this notion of the multimodal ways of interrogating biology. And we credential the axis that machine learning provides us in order to differentiate between the healthy neurons and those that have disease-causing mutations. We credential that using tools, for example, like testing whether rapamycin, a known revertant of the disease, actually reverts across the disease axis. We are also able, in the context of transcriptomics, to align with human clinical data, specifically from bulk RNA-seq collected from patients, from the brains of patients, as well as the brains of healthy controls. And you can see that the disease axis that was trained on these iPSC-derived neurons is capable of perfectly separating the bulk RNA-seq taken from those patients. So that gives us some confidence that this disease axis is actually clinically relevant. We similarly credential other disease axes. So, for example, this is a machine learning disease axis taken from both fixed and live cell imaging. And again, we use similar tools. In this case, for example, the rapamycin dose response, and you can see that, for example, on the right, there is a clear reversion over time to a rapamycin treatment along the disease axis in a way that is dose responsive to the amount of rapamycin delivered, again, providing some amount of confidence that this machine learned disease axis is clinically relevant. So with those disease axes, we have built a pooled optical screening platform for the imaging readouts. We also have a different pooled CRISPR screening platform for the transcriptomic readout. That one is more standard. It's an approach called Perturb-seq that some of you may have heard of. This is one that we built in-house. It takes a pool of cells, in this case iPSC-derived cortical neurons that are all identical, introduces a pooled CRISPR guide library into that pool of cells at an MOI of about 1, so that each cell approximately gets 1 guide, therefore giving us a pool of genetically diverse cells, each with a different knockout mutation. We then take those cells all together in the pool and leverage all of the imaging modalities that we have at our disposal to phenotype cells at a single cell level using very rich readouts that include cell morphology, protein localization of individual proteins like phospho-S6, as well as live cell dynamics. So what you see here, if you can see on the right, this is our live cell imaging platform, and those little pink dots that are moving are mitochondria moving across axons. This gives you a very rich set of phenotypes that you can measure and then test and create basically a single cell phenotypic matrix. And then we fix and sequence the barcode within each of the cells using in situ sequencing approach. Basically, we had to build a sequencer inside a microscope, so you have a single cell genotype phenotype readout that allows us to really, in a very scalable, robust way, identify the effect of a genetic perturbation on all of those high content, high dimensional imaging readouts. And because of the investment that we've made in assay development and automation, we can take now a disease model to testing these modifier reverting kids in 3 weeks, which is a very important ability in terms of our turnaround time. So between the transcriptomics and the imaging, we ended up with 76 hits that we then sent into, that reverted the phenotype, and then we sent them into various forms of credentialing of the hits or validation. So one form of validation that we like to look at is support in human genetics. I think there is a growing recognition that targets that have support in human genetics on a clinically related phenotype are much more likely to succeed in the clinic. And so we looked at the set of phenotypes that we thought were TSC related, and that includes epilepsy, other neurological impairment, kidney and cancer, and those were the ones that we looked at. And we were able to very pleasingly show that of the 76 hits that we took into credentialing, 55% of them had genetic links to TSC relevant phenotypes, which is a fairly high proportion. Now, if you look at the actual Venn diagram at the top, you'll see that the majority of the hits were actually receiving support from epilepsy and other neurological impairments. This is very different from the TSC revertant hits that had been previously identified in CRISPR screens in the literature. Those were mostly done on immortalized cancer lines. And what we found both in this experiment and in others is that modifiers are very cell type specific. So that the set of things the set of genes that revert a phenotype very much depends on which cell type you're doing the experiment in. We're doing these in iPSC-derived neurons, which is why we've got a very different set of hits than typically is found. The other form of credentialing of the hits that we did is in a functional assay called multi-electrode array. This is a very different orthogonal readout to the 14-day imaging or transcriptomic readout that we used to do the initial screen. Here you have to grow neurons to 50 days until they develop network activity, where they start basically communicating with each other. And it is fairly well established that as these neurons reach this level of maturity, they start firing in tandem, a phenotype called hyperexcitability that is very parallel to what you see in patient EEG data. And so there's reason to believe that this is actually a pretty reasonable surrogate, if you will, to what we see in patient EEGs, and it is also known that rapamycin, in fact, rescues that the phenotype as well. So we took the 76 hits and we tested them using both chemical tool compounds were available, siRNA tool compounds in cases where the chemical tool compound was not available. And what you see here is an example of a dose-responsive reversion of 2 MEA phenotypes. On the left is mean firing rate, on the right is burst frequency. The red line is the TSC knockout, the sick neurons, the green is wild type, the blue is the reversion that you get from rapamycin, and the purple bars is a dose response to the tool compound that we employed for one of our targets, demonstrating that you get a reversion of this phenotype, which is dose responsive and at a magnitude that is commensurate to rapamycin for this particular target. We've since tested the majority of these targets, and most of which are novel mTOR pathway modifiers that you would not see in a standard mTOR pathway diagram, and we were pleased that 40% of them reverted the disease, many of them with an effect size that is at or close to rapamycin. And so this is what gave us several targets, and we picked a couple that we really liked. We're now winnowing out the rest and have sent them into drug discovery, and as I mentioned in the beginning of the presentation.
So this is TSC. In parallel to our work in TSC, we also took on a massive effort to take that same platform and apply it to a much more complex disease, which is ALS. ALS is more complex genetically, and that only 10% of the cases are caused by 25 known genetic drivers. 90% of the cases are sporadic. And I don't think I need to tell this audience about the clinical manifestations of ALS and the fact that there's really no disease-modifying intervention. This is a partnered project with Bristol Myers Squibb. Here we followed the same process, looking at each of the ALS mutations effectively as a monogenic disease, and then trying to see what commonality we were able to find. So we took similarly high content data from these iPSC-derived motor neurons. By the way, cortical neurons are so easy compared to motor neurons. Motor neurons are really a pain to work with. And we were able to show that once again, there's a tremendous amount of information available in imaging on these neurons. So what you see here, for example, is the ability to infer TDP-43 mislocalization, a quantitative trait from pure cell morphology, basically that being TUJ1. At a correlation of 0.725, this is at a single cell level. You're able to look at a cell, staying only for core cell morphology with these two readouts, and able to infer TDP-43 mislocalization, which is the hallmark of ALS. Even more surprising, perhaps, is the ability to actually impute differential expression between mutant and wild type, also in imaging alone. In this case, it's TUJ1 plus TDP-43 that are used for the imputation. But you can see that there is a very strong correlation in predicting basically the gene differential expression between mutant and wild type for these ALS relevant genes. So with this conviction that there's all this information available in our imaging readout, this is in 10-day-old motor neurons, which, by the way, is a surprise in and of itself, because whereas TSC is a neurodevelopmental disease that begins its process presumably in utero, ALS manifests in your 50s. And so the very fact that in immature motor neurons you actually see an ALS phenotype is itself noteworthy. So here we took these are 5 of the common mutations that are known familial drivers of ALS. And what you see here is the ability to predict mutant versus wild type using this machine learning model. And you can see that it predicts considerably better the blue bars compared to the gray bars, which are using TDP-43 mislocalization anchor phenotype on its own. This is done in a very rigorous regime where you're actually predicting across completely different donor backgrounds in a completely different set of wells. So the fact that there is a generalizable phenotype that you can train on one set of donor backgrounds and test on a different set of donor backgrounds. And because the phenotype is so much stronger in the machine learning model, you're now able to do some of those screens that I showed you on the previous in the TSC work in a way that is much better powered, because you have a much larger dynamic range between mutant and wild type, and therefore able to recognize reversion at a much smaller number of cells. So you could actually reduce the number of cells that you need by a factor of approximately 5 in order to demonstrate reversion. So this is the first screen that we ran. A number are still ongoing this year. This is one screen, and you can basically see 2 different disease axes on these 2 dimensions. And what you see here is the change between a non-targeting guide that doesn't do anything, presumably a positive control reversion, and one of our newly discovered hits. And we're quite excited about these novel targets as our partners at DMS, and we're now progressing those into additional credentialing assays as well as drug discovery. So all in all, in order to make all this happen, we have built an incredible set of resources. We have probably the largest known collection of isogenic iPSC lines that span both familial and sporadic ALS genetics, over 200 lines. We have robust automated protocols to generate motor neurons in a very repeatable way. We have all the phenotyping work that I talked about. We have this POSH platform for screening to find genes that revert disease phenotypes, and then the scaled processes such as NEA for target credentialing. And the goal here is to take the multitude of known genetic drivers of ALS and to really sort of identify how they, probably through different pathways, give rise to ultimately motor neuron death, and which are interventions that might end up reverting the phenotype in one or more of these subtypes. How much time do I have? Sorry, I don't have that.
5-ish minutes.
Okay, great, perfect.
7 minutes.
Perfect. Okay, so very briefly to talk about our work in metabolic disease, which I recognize is not a rare disease, but I hope that some of the lessons learned here will apply broadly. Here we take both the clinical and a cellular machine learning approach to really create something that is incredibly powerful. So what we end up doing is we take large amounts of available human cohort data and do a genetic analysis to create a much deeper understanding of the genetics that underlie the disease. That gives us both a set of potentially interesting genes that are involved in the disease, but also if you take a step up and look at a more holistic view, it also tells you a set of biologies or processes that are perturbed by the disease, which helps inform, both of these help inform the construction of a cellular disease model in ways that allow us to more correctly interrogate the disease processes. Out of the screens that come out in these cellular disease models come out hits, which we can then go back and take into our human cohort using processes like FIWAS and interrogate whether those genes actually are in fact disease relevant. So the disease that we started out working with here is a disease that is currently known as MASLD. It used to be called NAFLD, it's upper fatty liver disease. It's the aberrant accumulation of fat in the liver. There is currently a very limited understanding of disease genetics, so they're almost certainly heterogeneous, and there's actually evidence to support that many of those are actually an amalgamation of diverse rare diseases, especially when you're talking about genetically driven lean MASLD. But there's very little understanding of what genes actually drive the disease. So what we did here is we basically tried to say, how can we get a better understanding of the disease genetics? So if you can in fact measure liver fat quantitatively using MRI, but MRI is an expensive, fairly complex modality to deploy, and it's not readily available, especially in cohorts that have human genetics alongside. And so what we did is we took a bunch of assays that are much weaker, if you will, but also much more abundant. So the three modalities that we looked at are DEXA scans, which are much more abundant than MRI. I mean, there are gyms that have DEXA. Circulating metabolites and circulating 37 blood biomarkers. And we asked the question of, can you impute quantitatively liver fat from each of those modalities separately? And we had a training set on which both modalities were measured simultaneously. We used that as a training set, and then we were able to test the performance. And what you see here is that, somewhat surprisingly, from DEXA, which is a bone scan, you're actually able to impute liver fat with an R-squared of 0.63, and what's below is basically predicting people with particularly high liver fat, a dichotomized trait, and what you see here is the area under the curve, and very close to 0.9. From metabolites and biomarkers, the accuracy is somewhat lower, but still quite functional in terms of the ability to get reasonable predictions. And so with that, you can now take this imputed quantitative trait and align it with human genetics, which are now much more readily available. So we took the Manhattan plot that looked like this when we used MRI as a modality. You can see there's very few genes that bubble up to statistical significance, to something that looks more like this, where as your N grows from PDFF with a few thousand to biomarkers with a few hundreds of thousands, all of a sudden you have a much richer genetic landscape, and a bunch of very compelling hits emerge from this, including maybe one that's worth highlighting. FGF21 is a target against which there are drugs that are performing quite well in the clinic right now. There has not been, up until now, any human genetic support for FGF21, which always made a bunch of people feel somewhat queasy about the target, because people are now very conscious of wanting human genetic support. It came out loud and clear in our much better powered human genetic study. So in addition to individual genes, as I said, you also get an understanding of new pathophysiology more broadly. So if you look at the pathway analysis of pathways that could potentially be involved in MASLD, and you overlay the former genetics on top of that diagram, there's very little that emerges in terms of conviction about which target is actually driving the disease. If you take our much better powered GWAS and overlay it on top of the same pathway diagram, de novo lipogenesis, which prior to that had very limited evidence, comes out loud and clear. If you go to people studying metabolic disease and you tell them this, they'll say, we knew this, it's just that we didn't have the genetics to provide the evidence. And so now we do have the evidence to provide. And with this type of result, we can now take and build our in vitro assays, the same ones that I showed you before, but now in hepatocyte cells as opposed to neurons, and basically trade out the standard free fatty acid stressor, which really measures lipid uptake, to a de novo lipogenesis stressor, which measures the creation of fat. And you can see that we were able to both create the phenotype in vitro on the left, we were able to revert it using DGAT2, which is Pfizer's phase 2 clinical asset. And in the middle is one of our novel targets that has a reversion of at a level that is comparable to DGAT2 in this de novo lipogenesis stressor conditions. Just before the holiday, December 22nd, literally, we were able to get results from 3 first targets that we pushed into animal models. This is an OB mouse model. We took 3 of our targets that have the strong genetic support that capture these novel mechanisms that are actually master regulators of DNL, and we were able to show comparable triglyceride reduction in the mouse livers that is, again, at the same magnitude of DGAT2, and this is before we optimize the compounds towards greater efficacy. And these targets are now the basis for programs that we're rapidly advancing using siRNA into therapeutic interventions, and we hope to bring at least one of those into IND-enabling studies later this year. So all in all, we built out a fairly powerful discovery platform that really drives our pipeline. We have capabilities for creating multimodal data at scale across human cells and molecules. I didn't talk about our chemistry platform, but it too exists. As we said, data drives everything. The imputation to do data expansion, which I demonstrated both in the neuroscience world, where we're able to impute TDP-43 or transcriptomics, as well as in the liver case, where we were able to impute liver fat, allows us a much more powerful set of data cohorts, and hence that stronger, more predictive models, which give us high confidence novel insights that give us a pipeline of therapeutic programs that we're advancing across both the therapeutic areas that I demonstrated, as well as in oncology, which I did not. And all of this is in service of our mission, which is to bring better drugs faster to the patients who can benefit most through machine learning and data at scale. And if you boil it up to our three-word tagline, it says pipeline through platform, which is a fairly unusual thing in the biotech world, just to really build a platform that is a goose that continues to lay golden eggs. But we hope this is in fact what we have built. Thank you very much.
While the next speaker gets. Yes, there's a question. Do you have a question? No. Okay. You got a question?
Yes.
Go for it. Oh, you need a mic. Okay.
Oh, there's one here.
We'll just give you the mic.
Okay.
Hi there, I'm not a scientist, but I'm trying to do it expeditedly. But you talked about the cells showing the motor neuron within 10 days, I think. That goes to getting it, expediting the drug discovery process from platform to the patient. So how long would you say one of these ALS, this study takes from start to finish to get these targets? And the reason I ask is because they were asking the question earlier in the previous panel about, barriers. We're talking about 8,000 plus rare diseases. The reality is I think it's cost prohibitive. and time prohibitive to tackle each of these with an IND and IRB for each single gene disease. My son being one of them, he has a rare neurodegenerative disease and he's 4 years old, and there's less than 20 patients reported. So the reality is, unless I do something where an AI-generated platform could take the scientific research and bring it from, using the AI to kind of expedite this process and hopefully get it approved quicker.
Yeah, no, that's a really great question. And I'm so sorry about your son. This definitely provides an acceleration of the ability to discover novel targets, because the first time that we did TSC took a really long time. Relatively speaking, but we're to where we'd like to be, because we had to build a platform at the same time that we were doing the study. Now the platform is up and running. We have since identified a number of other genetic epilepsies that we think the same cell type works for, that the MEA assay is useful for credentialing. So we have the building blocks already in place. Now to go from that starting point towards a target screen is a matter of a few months. That's the good news. The bad news is that once you have a target, you still need to make a drug. And a drug is, depending on which modality you pursue, in the brain would either be presumably small molecule chemistry or potentially gene therapy. And the development time for those is still potentially quite long. Now, there was some discussion earlier of drug repurposing as a way of potentially accelerating the process. That is certainly something that we've looked at as our targets emerge to see if there is a known drug that we could potentially deploy. Sometimes there is, oftentimes there isn't. Chemistry is something that we are working on accelerating using a platform that I didn't discuss, but it's still, making compounds, trying them, and then making sure they're safe for patients. There's still parts of this that are hard to compress, and we've only compressed one part, I think, so far, or maybe more than one, but certainly not all.
Thank you. That was a really good talk. You kind of touched on it in this answer, but when you're dealing with rare diseases, the whole plethora of data that exists for common diseases becomes still very sparse. Yeah. How does your technique deal with that?
That's a great question. And, there's partial but not complete answers. So for example, the models that we have, just to maybe focus on metabolism for a moment, because that is a common disease that we worked with. The models that we have for imputing liver fat work by looking at data modalities that are abundant and I think are likely to be successful for imputing liver fat, maybe not for all rare subvariants of the disease, but for at least a subset. Now, I think the question then is, are you powered to identify novel genetic drivers in that smaller population? Or perhaps, if you want to be optimistic, maybe some of the revertants that are relevant to the larger population are also potentially relevant to the smaller population as well. And we might be able to kind of dual purpose some of the therapeutic interventions. But that's all, it's a hope, right? And we don't have any guarantees that that in fact will work. The imputations are probably the most sort of generalizable thing. I didn't show the imputations that we have in cancer where we're able to impute gene expression from histopath. That imputation is trained and tested pan-cancer. So it works for rare cancers as well as it does for common cancers, actually surprisingly well. So there are places where you have this kind of translatability, but you still have to rely on the fact that you are powered to discover interventions in the rare population, despite its small size, or that interventions that you have in the broader population will translate over to the rare population.
Thanks for your talk. I'm wondering if you use the platform to impute clinically useful PD markers for disease perhaps there are none.
That's a great question.
Especially on what the kind of feedback around that.
That's a great question. I don't think we've thought about this, so thank you for the suggestion. I'd have to, see what was available from a data perspective in terms of collected PD biomarkers, and then one would obviously have to train this. This, I think, is related to the other biomarker imputation answer that I gave a moment ago, which is a lot of that does, I think, translate, but I would have to, I mean, it's on a biomarker by biomarker basis.
Computer science.
Okay.
Early in what you said compute is effectively
Okay, so to be fair, it is a way of relating the concept of imputation to to concepts that are very much in people's minds right now. Imputation is basically a statistical prediction task, so is generative AI. You're generating data from an input, but you're generating data that does not exist. And then you're, presumably testing the quality of your predictions. So at what point does pure statistical imputation using a very simple linear model turn into generative AI? I don't know. You'd have to adjudicate the boundary, but it is on that spectrum because you're not just doing a binary classification problem. You're actually generating a full-blown data modality. So it feels to a lot of people like generative AI.
I have two short questions. One is, where do you get your human cohort?
Yeah, no, that's a great question, and it really depends on the disease. The work that I showed in metabolic disease actually comes from the UK Biobank. We are also in the process of acquiring additional cohorts that would complement that. The work that we are doing in oncology, which I didn't present, some of that comes from partnerships with national cohorts like Genomics England. Some comes or TCGA, which is publicly available. Some comes from individual partnerships that we have with academic health centers and so on and so forth. And some are data that we're generating in-house. So we're making a very big investment, for example, in spatial biology in order to basically print data that one can then use as well. So I think the answer varies.
Okay, and then my follow-up question was, do you have longitudinal data?
People sometimes ask me if you had a magic wand that you could use to generate anything, any data set that you could possibly want. My answer is longitudinal, densely phenotyped human cohort data with human genetics. I wish. UK Biobank has recently done UK Biobank at least does collect some longitudinal data because all the patients are registered as part of the National Health Service. So at least you have longitudinal outcome data. If they got sick, you have some blood measurements and so on. They do not currently have access to all of the measurements. Imaging is standard of care imaging is not currently there. They just did a repeat imaging study to bring back a subset of the people who were imaged the first time. There's 100,000 of them, so that you could have a second time point, which is still only 2 time points, but it's better than nothing. But boy, what I wouldn't give to have that type of longitudinal resource, because I think the really important insight here, which I think is what you're alluding to, is that from an interventional perspective, we don't get to, by and large, intervene in disease inception. We get to intervene in disease progression. And so what you really care about is the genetic drivers of disease progression, which are oftentimes quite different from disease inception. And so that visibility is something that we greatly lack in almost all diseases at this point. Yes. Yeah.
Thank you so much.
Thank you.
Yeah.
Okay, it's now my pleasure to introduce the team from Good Growth Capital. Their presentation is, pre-recorded. And Amy Salzhauer is the co-founder and managing partner of Good Growth Capital. She was an undergraduate at Harvard, grad student at Cambridge, and they got an MBA from MIT. And she's also helped start numerous companies and serves on a number of corporate, nonprofit, and academic boards. Joining her is Lawrence Olenoff, who's an advisor to Good Growth. He holds an MD and a PhD from Case Western, and his career was at Upjohn and Sandoz, and he was the COO of Forest Labs before it was acquired by Allergan. Rob Cutchfield is the operating partner at Good Growth. He was a graduate of the University of Georgia in both, excuse me, computer science and chemistry. And he was at many healthcare companies, including Cardinal Health and US Oncology. And he serves on numerous boards. And finally, Sam Schofield's a senior associate there. He's a graduate of Colby College in Maine in biology. And he was a management consultant after college. And now he does due diligence and evaluation of companies. And he's a board observer of 4 of Good Growth's portfolio companies. And so Sohaila's gonna boot up their, yeah, there's one of them right there. I can tell by the name.
Hello, everyone. We are recording this panel session remotely ahead of the 2024 UCSF Rare Disease Symposium. It's a pleasure to be here today, and I'm joined with my esteemed colleagues from Good Growth Capital. Our panel today is called Investing in Rare Diseases, a Venture Capitalist's Perspective. We will be discussing our experiences with investing in rare diseases, some of the challenges, as well as the opportunities. And with that, I think we can kick off. So I believe the audience should have access to more in-depth bios of our panelists, but I wanted to give everyone the opportunity to introduce themselves briefly. So I will be moderating the session today. My name is Sam Schofield. I'm a senior associate at Good Growth Capital, working on the life sciences investments. Prior to joining Good Growth, I was in life science consulting at Prescient Healthcare Group and then LEK Consulting, and recently joined Good Growth as an investor. I will pass it to Amy, then Larry, and then Bob.
Hi, everyone. Sorry, we're not joining you in person today. I'm one of the managing partners of Good Growth Capital and the founder of the fund. My background really was an interest in how things like rare diseases could be funded, why certain technologies successfully or don't successfully leave the lab. And I have a background in molecular biology and plant sciences, an MBA, and then I starting companies to get data for my doctoral dissertation. So I'm really coming to you as a failed PhD student who never finished writing her dissertation. But 11 companies later, I feel like I do have a little bit to add on this subject. And we've just been so excited here to be able to fund some rare disease companies at Good Growth Capital. We'll tell you a little bit more about the fund in a moment, but why don't I pass it first to Larry and then to Bob.
Hello, my name is Larry Olenoff. I'm a clinical pharmacologist by training. Spent my career in the pharmaceutical industry. Having retired from there, transferred over to the Medical University of South Carolina, where I serve as an advisor to the Technology Transfer Office. I serve as an advisor to Good Growth Capital, which is my connection to Amy and her colleagues. I've been on the board of a number of public pharma companies and now smaller private companies in the biopharmaceutical space. A number of which have pursued orphan drugs even before they were called rare diseases. So I've watched that whole area evolve and have a pretty reasonable understanding of some of the hurdles and blocks we face in moving these projects forward.
Hi, I'm Bob Crutchfield. I'm an operating partner for Good Growth Capital. I've been in healthcare my entire career. Started out in the more traditional way of selling surgical products and moved into provider services. Took a company public early in my career and then did build and grow work as a VP GM for Cardinal Health and then US Oncology, where I built and ran their pharmaceutical services division. When it was sold to McKesson, I got into venture capital where I served as a general partner for a firm in Birmingham, Alabama called Hart Growth Partners. That's where I met Amy and Good Growth. And a few years later, I found myself with an opportunity to build and the Bright Edge Fund for the American Cancer Society, and did that. And then today, I live in Charleston and have reconnected with Amy and her team, and I'm very fortunate to be part of their organization as an operating partner.
Great. With that, I think first question, Amy, but I think it would be great for the audience to hear a little bit about the genesis of Good Growth Capital and perhaps, how Larry and Bob are involved and other, associated members of the team.
Sure. So we had, in my career, I'd worked on really transferring different technologies out of academic labs. It's a challenging process and challenging to bring together a team that really has the technical and operational expertise to be able to fund those kinds of innovations, especially at deal sizes that start smaller at the beginning. It's easy to put together a team that can put in $50 or $100 million to a software company in the first round. But that's not usually what we're up to here when we're funding these kinds of innovations. So we felt like it was a really important mission to see if we could bring to that kind of technical and operational expertise around the table. We're a women-led impact fund that invests for the highest possible return. So one thing that we also thought was important was to really level the playing field for founders from all different kinds of backgrounds and ethnicities, and for founders that might not meet the kind of stereotype of what makes a charismatic venture-backed founder, but to be able to fund people at the very earliest stage, at the, even if they don't have the business experience, if you really have a transformative science idea, we're going to listen to you and really know how to help take that from just that transformative piece of science to all of the business steps that it would need to take to be able to be fundable and continue to grow. That was really the mission of the fund. We also invest in data sciences and green tech and advanced materials and the intersections of all of those things. It's such an exciting time to be investing in life sciences. So we're really happy to be speaking with you.
Great. I think that's great overview for everyone. I thought it would be interesting to hear just a little bit more about kind of your perspectives as leading a venture fund for as a woman. I looked at some research, and according to the latest research, it's quite grim. 1.3% of investments into VC funds went to female-led VCs. So certainly trailblazing in that regard. So would be curious to hear how that kind of impacts your perspective on investing.
I think that we really try to provide safe harbor for those founders, but we're really looking, and I guess one of the ways we're doing that is that we're really looking at your science. This team is going to look very hard at the intellectual property, at the vision of the company, and then, for better or for worse, we do need to make sure that things actually will return capital to our investors. So the way that venture capital works, and this is going to come up throughout our conversation, I think, is that there really are constraints. We need to have at least a 3x return to our investors, preferably within 3 years. It means that funding rare diseases can be very, very challenging. And, Bob and Larry have a lot more years under their belt of experience with some of those challenges. So, but I'm really happy to speak to anybody from the symposium. Send us an email if you'd like to hear more about the specifics of and challenges of being a female founder or a female venture capitalist. We'd love to encourage you.
Great. I guess just to set the stage for the audience, might be helpful to share a little bit about our investment areas within the life sciences. So we're about split 50% on medical technology, 50% in biotechnology. Within the biotechnology, we invest about a quarter of that, or half of the biotechnology, into therapeutics, and then the other half into diagnostics. Larry, anything you'd like to add in terms of what are some of the areas that we look for, and what are some of the qualities that we look for when we're making investments in life science companies?
No, I think one of the key features of the companies we look at is the quality of the science, the logic behind their development pathway, and clearly the understanding that the product from a development perspective has to pursue an identifiable market. Add those features in to having a stable intellectual property platform, and you have what is the nidus of a company. So I always refer to it as the three-legged stool: the market opportunity, the technical regulatory pathway outline, and the intellectual property which grants that exclusivity to that product. So those are the general features I look at when we evaluate individual companies, be that a diagnostic, a medical device, or a therapeutic. With therapeutics, it gets more complex even from the standpoint of thinking about the target, the validation of that target, and there's a whole number of factors we can look at that are specific to rare diseases or orphan drugs, which I'm happy to comment on further.
Yeah, I guess before we get into that, Bob, anything else to add about what we look for, additional to what Larry has mentioned?
No, I think Larry's kind of laid it out well. I mean, the only sort of perspective that I would add to that is, this type of activity from a scientific development to, regulatory pathway, preclinical, regulatory pathway, clinical, it's an operating business. And so if you can't get your head around the IP and then find your ability to communicate with the entrepreneurs and believe that they have the ability to execute on the thesis, then that's the hard part, right? And that's what we're trying to underwrite to, because these deals are really early most of the time when we get involved, and we're trying to look for an expedited pathway to at least some stage of clinical trial performance, our preclinical performance, regulatory performance, that gives an eye that this is something that will get into the market. And it's a very challenging set of variables that we're trying to figure out.
I'll just add, we have a lot of scientific expertise to evaluate deals. It's very hard to find funds that actually have that expertise. And if you find yourself pitching to a fund that doesn't, that's a clear signal to you about how things are going to progress. And you're much less likely, I think, if you have a rare disease, to end up with actually being funded, because it is very complicated to diligence most of these kinds of investment opportunities. There are we're certainly not the only fund with, I'm not pitching the fund. We're not the only fund that has a lot of scientific expertise, but that's something that you should look for, people who understand really what you're doing, not just looking for metrics in terms of economic metrics or returns.
Amy has made the comment multiple times when we reviewed companies started particularly by young founders who don't really have a lot of experience doing this, moving a product forward through a development pathway, that one of the critical features is finding individuals that are open and coachable, individuals we can benefit from their expertise and the quality of their science, arguably, but they can also benefit from our experience.
We're old. Is that what you're saying, Larry?
Well,
No, he's saying we've been young longer.
That's great. Well, thanks for sharing all those factors that we're looking at. I guess back to you, Larry. Are there specific things when you're evaluating rare disease startups that may make it compelling for you, things that you look out for?
I think from any therapeutic standpoint, as I mentioned, target validation is a key point that one needs to begin with, and directed specifically to the disease of interest. That can be done a number of ways. Popular ways are using knockout models in mice typically, or siRNA type evidence to show that the target really is valid to the disease of interest. But that also evolves around the fact whether or not you have actually a transgenic or some other animal model of that particular disease where the gene has been identified, by example, if it's a monogenic target. And the knowledge of that gene of interest and whether that gene of interest is specific to a very small portion of that population or whether you're looking at a therapeutic that perhaps can address a larger part of that particular rare disease because it's not directed at any specific gene. See, these are all considerations. The animal models can be very interesting in their design. I like to look at models, and a lot of my colleagues in this field like to look at models that not only translate to the gene of interest and potentially allow for biochemical or cellular biomarkers, but also show functional phenotypes that one can extrapolate to the disease in humans. And that's just the beginning point. Oftentimes, the big stumbling block is reproducibility of that model, and not necessarily reproducibility of the therapeutic results, but simply reproducibility of the model's phenotype. And there's horror stories that people tell of moving that laboratory perhaps 25 miles down the road and still not being able to reproduce that model. So that becomes, I think, the first major hurdle in terms of moving these disease targets forward. It's also very helpful if you can recruit a secondary model, which gives you a greater degree of confidence than simply reproducing those effects in the same animal model. And once you have your molecule of interest identified or your therapy, whether it's a biological therapy or a cell-based therapy identified, then, or gene therapy for that matter, then it's understanding the disease and thinking about the clinical program. And that's a whole nother discussion of its own. But people oftentimes refer to doing studies in natural history of disease, which means you need to partner with typically with foundations that have been built around this disease, and oftentimes identifying a consortia of individuals that treat this disease, that see these patients in very selected centers. Because as rare or orphan disease defines, these are not broadly present in the population, and you're looking at a selected or limited number of centers where these patients are seen. And understanding the disease, understanding the variability in the disease is critical if you're going to try to design a clinical trial, particularly in areas where clinical trials haven't been pursued in that particular disease. And if, without getting into any further detail at this point, I will comment that the regulatory agency in here, the US, the FDA, has become more receptive to looking at disease models in the clinic where surrogate endpoints could serve as a point of approval. And that becomes critical in diseases where the clinical endpoints, be it death or disability, declare themselves over a long period of time. It's very hard to anticipate trying to do a trial which takes 10 years to perform, because you've basically eaten away at all your intellectual property and you're relying on the orphan drug exclusivity alone. So, by example, in a number of diseases, particularly in ALS now, there's opportunities to look at biomarkers, which may be of interest here in terms of utility specifically for purposes of getting approval. So I'll end there with my comments.
That's great. Thanks, Larry. I guess would be helpful for the audience to know Good Growth Capital has invested in 2 companies that have rare disease programs. The first was Skyhawk Therapeutics, and their lead program is in Huntington's. And the second one is Tavard Biosciences, which has a focus in Dravet syndrome as one of their various different indications. I guess to start, we can talk about Tavard. So Tavard, this is a Harvey Lodish founded transfer RNA therapeutics company. Good Growth Capital led the Series A round. It was founded with a mission from to parents of children with Dravet to cure Dravet syndrome, which is a rare form of epilepsy. And so I guess, Amy, would be helpful to hear kind of what did you think about in terms of the diligence here, and what got you comfortable in terms of investing in a company like this?
It's a good question. And in a way, they are outliers in terms of rare disease companies because they both are really developing platform technologies that then are applicable to multiple diseases. I think one of the big challenges, or the primary challenge from a venture capital perspective in funding for any rare disease, is that it's going to cost the same to take that through the clinic as it would to take something through the clinic that then has a very large market and large economic potential. And so when you're choosing amongst deals, we see a huge number of deals, thousands and thousands of investment opportunities, and then we fund tens. That means that we really do need to pick and choose and think which ones have the highest potential to have returns to our investors, at the same time that we are still, even though we're an impact fund, there are public-private partnerships. There are funds like the American Cancer Society, Bright Edge Fund, that have more mission to them than just economic returns. And I think that they can have, an easier time funding these kinds of companies. So we were really looking at the science with those companies. Like I said, we have, I guess we have the luxury of not having to focus on whether an invention or an innovation has a management team associated with it. For example, Bob might, parachute right in there and help things to grow as an operating partner, assume the role of an executive chairman, will help you build a board. But if, as Larry was saying, if the intellectual property isn't there, and the potential to see how you're going to move through the clinic isn't there, especially when these markets are so small, it's almost impossible. So Bob, do you want to comment on that?
Yeah, I think you and Larry have said it well. In businesses that are successful in rare diseases or any sort of, early stage life science, and any therapeutic or diagnostic, the IP has to be strong, but it has to be demonstratable at some level that it's repeatable in terms of animal studies or those kind of things, because things that aren't repeatable, they rarely become predictable, and things that aren't predictable rarely scale. And so, we're looking for that. And so evidence that supports the intellectual property, that there is a method of action that is demonstratable and shows promise in animals and translates to human phenotypes. Those are things that the more data that you can develop using grant funding and other sources of non-dilutive capital, when you then come and present to the venture capital funds that do early stage investing like us, you're checking the boxes for the things that we're going to, sort of deselect around, right? Because our business is about deselecting well, so that we go through all of those, companies that come through our funnel that we're not going to do, so that we're selecting really well the ones that are a good fit. So I think, the one message I would have out of this is use non-dilutive funding, grant funding, for as long as you can to get as far down the pathway as you can before you start, trying to raise venture capital.
Larry, I just wanted to touch on one point that you brought up. So, the use of natural history for some diseases. In some cases, I think, the natural history of a disease may not be fully characterized. And so in those circumstances, how, if at all, is there an opportunity to, move a drug forward? Is that too big of a barrier? Or how would you perceive a situation like that?
I think there are a number of options one can pursue. The most straightforward one is to conduct a natural history of disease study per se without an intervention, or at least without an innovation that's not standard of care, and really follow a set of biomarkers, functional outcomes within reason in terms of timing that you can also translate then to studies with your therapeutic of interest. Those can be somewhat costly, and they can be a barrier simply because of the cost. But if the funding is available, one can work with these disease foundations. They're oftentimes quite capable of helping you set up these studies. And as I mentioned, with the consortium that exists already, where people have been looking through perhaps therapeutics coming through the NIH sponsorship or other previously company-sponsored trials. I think it's also important to keep an eye on the literature and understand what people have used for clinical endpoints or outcome measures. And I would advise people, if they're interested in this area, to spend some time on clinicaltrials.gov. It's a free website. It gives you quite a bit of information in terms of what has gone on in the past. It sometimes even provides results on some of the studies. But one can look at those studies under that indication, get a better understanding what kind of general endpoint or outcome measure is entertained in both early studies, be they phase 1, phase 2, or even in more pivotal trials. The other option that one can pursue is try to if you have a reasonable understanding what you think is an outcome, and one that has stood the test in the past, perhaps for another disease in an FDA regulatory environment, is to employ that outcome and try to validate it in concert with the trial itself. And some people have done that in the past, and it's at least partially successful. So I wouldn't shy away from considering that as well as an option in terms of pursuing a program. But I think what is key is understanding how that outcome measure predicts for the long-term benefit or risk of that disease going forward, and being able to describe that to both the community and the regulatory bodies. I mentioned earlier in terms of surrogates. So one of the surrogates that's become very interesting to the community is neurofilament light, which has been employed now in an approval of tofersen in ALS, but it may also be of interest in multiple sclerosis and Alzheimer's disease and in Huntington's disease. So there may be some overlapping biomarkers here that one can employ, and as I mentioned earlier, look at these kinds of outcome measures from prior trials and perhaps even in related indications.
Very helpful. Thanks, Larry. I guess for the broader group, we've talked a little bit about some of the challenges associated specific to rare disease drug development and investing in those startups. But I'd like to hear from the group, what are some of the advantages of investing in rare diseases, and what are some of the positives that we see here as an opportunity?
Bob, do you want to take that?
Yeah, I mean, I'll start. I mean, you and Larry are truly the experts here, but the thing about a rare disease, that if you're successful with it, the win is very big, both financially and societally, and so there's that motivation that is both intellectually but also emotionally guided, right? So I think rare diseases offer an opportunity to solve a very big problem, get rewarded for it, but also get rewarded by the fact that you're doing something very meaningful for a small number of patients who are suffering from, very, very difficult and often traumatic diseases.
I'll echo what Bob said. I mean, the reason I started on my career path, I guess on my life path, was that I was watching thalassemics struggle to gain access to chelating agents. So I really started doing everything that I've been doing for my professional life because I saw the challenge for patients. I think all of us are doing this because we love it and we want to have an impact on people. When we funded Skyhawk Therapeutics, that founder had previously developed a drug for the Spinal Muscular Atrophy Foundation that was sold to Roche and moving through the clinic. And I met a 6-year-old with SMA who was, you know, up and about and thought that is just amazing. What, you know, what a miracle, but really it's not a miracle. It's science and drug development and all the work that everybody who's at this rare disease symposium, I'm sure, is working hard for. And we're working hard as well. You know, one thing I wanted to say is people often apologize for pitching us, for telling us their ideas, but every venture capitalist, it's actually their business to listen to you and to help you. And I think pretty much everybody, you know, is in it. Certainly anybody who's going to be in this area. I think one prime motivation is, generally speaking, not, you know, earning a lot of money, but having a lot of impact on patients.
When I started in the industry, pediatrics was a rare disease. Most pharmaceuticals were developed for adults, and really, you know, as a pediatrician, perhaps you needed to practice in the context of, well, this is what I think should be a useful dose based on my adult medicine colleagues. So we were, you know, back 30, 40 years ago, we were guessing at how the dose, both pediatric patients in general, but also in rare diseases where we felt perhaps some of these agents that are used in the general population may have value in these smaller populations. It was all trial and error. So now we've kind of reversed things in that pyramid. We flipped it, where rare disease can often serve as a very important proof of principle or proof of concept for a particular agent, as we know that target is specific to the validation of that disease problem. And in fact, if that particular therapeutic is both safe and effective, moving on from the rare disease, we can look at analogs and potentially pursue them into the general population. So in rare disease, beyond serving that community, which is so critical, can often have benefits even beyond that community at large.
That's great. And for sake of time, I wanted to wrap up by asking one final question, hopefully optimistic and forward-looking. So just to share some data, there's been obviously some considerable growing interest from both pharma and investors alike in developing rare disease therapies over the last 15 years. I read a statistic that said, I think 54% of drug approvals by the FDA were for rare diseases in 2022. And partnership deals between pharma and rare disease biotechs have gone up 6x over the last 6x over the last 10 years. So would be curious from the audience, kind of what are some of the trends you see in the market moving forward, and what are some of the opportunities for investors and people developing these types of drugs?
Who'd like to take that? Bob, why don't you start?
Yeah, I'll start and then hand it off to you guys. So this is really a team sport, right? And I think that's what you're alluding to, right? Expertise and experience, as broad as you can get it as you're forming your investment syndicate, or with Good Growth, we have this wonderful team of advisors who have deep experience in just about any, you know, disease type that we are looking at. So, but I think the more knowledge you have around the table in these investments, and the more experience and the more, you know, leverage you have for referencing outside data sources, advisors, et cetera, I think that increases the chances for success, because you're going to reach points along the way that you have to make a decision. And that decision is going to be informed by as much data as you can find, by as much data as you have produced on a retrospective basis. And then you're going to have to trust the instincts and the experience sets of the people around the table that you're trusting to give you guidance. So I do think having, you know, syndicates that are composed of investors and strategics often create a lot of value in these early stage deals. So, I do think, my perspective is that the more people you have around the table and the more experience and the more leverage of resources, the better.
I'm going to let Larry have the last word. I'll just say quickly that when I was starting companies, and I have full sympathy for founders, when I was starting companies, given where I came from and why I was motivated, starting with the thalassaemic patients, I always wanted to find some way that I could make a difference for orphan diseases. And every year, every summer, we would have our interns do the exercise where another one of our partners, who's still my partner, Maureen Boyce, would have the interns look at, why can't Amy start her rare disease company? And it was because it was economically not feasible. It was traditional pharmaceutical development. And I really feel like this is a golden age for rare diseases, just because now we're operating under a different, I'm going to say, a genetic regime instead of just hoping for compounds that show some efficacy. It's amazing. And with that, I'll turn it over to Larry, who has a lot more experience in this than I do.
I think it's always important, as Bob and Amy has said, is to get people around the table, even as consultants are on a one-time basis that have been down this pathway before in general, and understand all the constraints, all the hurdles that one has to face. You're juggling a lot of balls in the air simultaneously, and I think understanding what's in front of you and what will be in front of you is critical to your decision making and how you set up your development path and what are the critical milestones for funding as you go forward. I would, as an example, getting a regulatory person involved early on is quite important. Just getting orphan drug indication or orphan drug designation is a critical feature, I think, as you begin to promote your development program to other funding investors. And that can be done fairly early. You don't need clinical data to get orphan drug designation. Some people believe that's the case. It's not. You can do that with high quality animal work. So I think getting an understanding of where you're going and getting the right people involved early on, there'll be a lot of questions that you have to ask, and don't be afraid to ask them. And don't be afraid to contact people that, as I said, have been in this field before, particularly in academia, who may not know a lot about drug development, but understand the disease, more importantly, understand the population.
Great. And with that, I think we are at time. So thank you to all the panelists for all the great insights. And thank you to UCSF for putting on this great event. And if anyone would like to reach out to any of us, you can email me to start. My email is samuel@goodgrowthvc.com.
Thanks so
very much.
Everybody. Thanks, Sam.
Thanks. Good job, Sam. Thanks, everybody.
Bye now.
Hi, the day is almost at a close, and this is the golden age for cures in rare disease. And on that note, it is an absolute pleasure to invite Becky Beatty here. Please come and join us. She's the CEO of Launch Bio, who are instrumental in building these bridges, and it is my absolute pleasure and delight to introduce her to whet your appetite for the event tomorrow.
Here we go. Thank you, James. First of all, what an incredible day. I am inspired and motivated to keep working. I am incredibly honored to be in the room with all of you, very smart people. My name is Becky Beatty. I'm the CEO of Launch Bio. We are a nonprofit created to support early stage life science entrepreneurs and companies and help them grow by connecting with capital and resources. A few years ago, we noticed a trend where foundations, rare disease foundations, were starting their own venture arms. And we thought, this is interesting. And this is a new way to connect our community with very specialized resources, and understanding that it's like a three-legged stool. You have the startups and their deep knowledge of science, the foundations and their patient base and connection to advocacy groups, and then the venture arms that know how to build and grow companies. So we launched Invest in Cures, and we've done this forum across the country in Boston, New York, Dallas, a few times in Research Triangle, San Diego, where I am based this last August, and James was there. And we're so pleased to be doing it in collaboration with UCSF, which this conversation started 2 years ago by a connection that our friends at UT Southwestern made, which really, again, I want to emphasize the power of collaboration and meeting in these types of in-person forums to look people in the eye and have conversations and exchange business cards if we still do that, or maybe you swap, LinkedIn messages. So really, this is an invitation to return again tomorrow. If you have the time and interest, Invest in Cures will be we'll start at 8 to 9, will be networking and breakfast. We will learn about the advances in disease foundation funding, what we've seen and how it's come along over the last 5 years. We have a really incredible startup showcase, and following along with the ocular and rare epilepsy theme of today, we have 4 companies that we would like to present to you that are absolutely incredible. Again, the potential of AI to revolutionize rare disease diagnosis and development of cures, and then venture philanthropy funds that invest early, and what that means when they invest early and then follow along investment comes. So when we have an example of a company, actually a local company and a venture firm, and how that collaboration worked, it will be an incredible day. It is a ticketed event, but if you'd like to attend and you have the time, just use the code UCSF24, and we have flyers in front. But we'd love for you to join us, and if there's anything that Launch Bio can do for you, or any introductions you'd like to make, that's what we're here to do. We really see ourselves as the connective tissue, bridging some of these gaps and hopefully making a difference. So thank you for the opportunity, and thank you to UCSF and the Foundation Ipsen. It's an incredible collaboration, and we're honored to be here.
Somebody's lost their keys, and that's not a metaphor for solving a rare disease cure situation. If these are your keys, come and get them. Look, I know it's late, but a massive, massive thank you to the whole team at UCSF, the whole team in Paris, this has been a true collaboration. But I think far more importantly, thank you to you all being here all day, throughout this whole day. This is a massive, massive health need. And bringing your genius, your energy, your thoughts, your conversation, your collaboration to this mission is way more important than us being in a room today. But you can hear from what happens tomorrow, this is always becomes the start of something important. So a massive thank you. Please help, please engage, please bring your genius to the table, and thank you for coming today. Thank you.